the time allows for it. So without further ado, Hannah, please.
Thank you, Ivan, and good morning, everyone. Next slide, please. Can we just check that you can see our slides? Sorry.
Can anybody tell us if you see the slides in the room?
Yeah, we do.
Perfect. Thank you so much. So we are, of course, sure that all of the participants today are aware of the scale of the problem that AMR presents, but we would still highlight a few key figures here. So AMR is accelerating, as today 1 in 5 bacterial infections is caused by a pathogen that is already resistant to antibiotics. And as you can see here on the slide, there are also significant human and economic costs for the EU and EEA countries due to AMR. But we know from evidence that there are solutions, as every euro invested in prevention would bring nearly 3 euros in economic benefits. So One Health action is affordable and effective. The European Commission has long recognized AMR as a priority that requires sustained action. And with the new Commission mandate, AMR is also clearly identified as a priority in the mission letter to Commissioner Vahedi. Next slide, please. Before we will present how the Commission is addressing AMR and supporting our member states, we would first like to explain our role in DG Santé. So our unit is coordinating the One Health and AMR 5 across DG Santé and within Commission services, and we are situated in the Directorate for One Health in DG Santé. That means in practice that we are regularly bringing together colleagues from all different kinds of DGs of the Commission who are working on AMR-related files, and we regularly exchange knowledge and input. We are also closely working with 5 of our EU agencies, as our unit is responsible for the newly established interagency AMR working group, which enhances the cooperation between the European Environment Agency, the European Centre for Disease Prevention and Control, the European Chemicals Agency, the European Food Safety Authority, and the European Medicines Agency. Through this working group, the agencies coordinate, share knowledge, and promote synergies on topics such as awareness raising and training activities on AMR. Another work stream through which we coordinate with these EU agencies is the Cross-Agency One Health Task Force, which is a joint initiative of those 5 agencies that goes beyond AMR and enables them to successfully contribute to the implementation of the One Health approach in Europe. Next slide, please. So our main initiative on AMR in recent years is the Council recommendation, which was adopted in 2023, and the proposal was published at the same time as the proposal for a revision of the pharmaceutical legislation, which is currently under negotiation. The Council recommendation provides recommendations to the Commission and to Member States to maximize synergies and achieve a strong response against AMR, taking a One Health approach. The objectives cover the key fields of actions that are necessary to tackle AMR. So, for example, we have actions strengthening national action plans in a One Health approach. We have actions on surveillance, infection prevention and control, stewardship, awareness raising, but also cooperation and global actions. And the Council recommendation also includes some recommendations on research and development. And here we would like to highlight the new One Health AMR partnership, which is a 10-year research innovation initiative that is coordinated by the Swedish Research Council and involves 53 organizations from 30 countries with a total budget of 253 million euros. And the first joint transnational call was launched in November on European Antibiotic Awareness Day, and we invite you to have a close look at that one. Next slide, please. So the most important or novel action in our Council recommendation are our targets on antimicrobial resistance and consumption, which are to be reached by 2030. On the animal side, we also have a target that has been set to reduce the overall EU sales of antimicrobials used for food animals and in aquaculture by 50%. And on the human health side, we have 5 targets, all with the baseline year of 2019, so before the COVID-19 pandemic. 2 targets are on consumption and 3 are on resistant bloodstream infections. And in setting these EU targets, we realized that each individual member state, of course, also has to reach their own national targets. And so the national targets have been linked to the national situation and context, as we know that no single solution fits all, and member states need tailored actions. So every year at the occasion of European Antibiotic Awareness Day, the European Centre for Disease Prevention and Control, they publish the latest results on the progress towards those targets, and the key results for each country are available on ECDC's website. But we have added here, for example, the example of the Greece's progress with the 2024 data. So the latest results show that unfortunately the EU is not yet on track to reach most of the targets. Consumption is overall increasing, and so far only little progress has been achieved in the proportion of antibiotics coming from the access group. Many countries have also shown increases in the incidence in bloodstream infections, particularly with E. coli and carbapenem-resistant Klebsiella pneumonia. And this is also here the situation for Greece, as you can see on the right-hand side. Following these new results, we are working closely with ECDC and member states to understand where further efforts and support are needed. And our goal as a Commission is, of course, to ensure that the information and the support is available for those countries to show some positive progress as soon as possible. Next slide, please. Then there are a number of ways in which the Commission is supporting the member states, and we will continue to do so in the best way possible moving forward. Our main tool is the Joint Action EU Jamrai, which my colleague Ivan will explain in more detail in a few minutes. But there are also other tools and fora in which we are supporting member states, and we have highlighted some here on the slides. So we have, for example, the One Health AMR network, which is ensuring that all information and best practices between member states and stakeholders are exchanged. So this expert group comes together twice a year and brings together member states, stakeholders, EU agencies, and also Commission services. And the most recent meeting took place at the end of November, and the presentations and also the agendas are publicly available on our website, and we have included the link at the end of our presentation, so you're very welcome to have a look. Then in between meetings, we have the EU platform on AMR, where we keep engaging with stakeholders and member states, and we have a weekly newsletter where we publish the latest news and scientific reports related to AMR. Generally, raising awareness among citizens and stakeholders is, of course, very important. So we monitor citizens' knowledge every few years through the so-called Eurobarometer survey, and we will have a new one next year, and are hoping that this will show an increase in awareness about the responsible use of antibiotics. And then what we already mentioned is the European Antibiotic Awareness Day, which takes place every year on the 18th of November. And this is when our awareness-raising activities accumulate. And this year we have focused on member state success stories, showcasing best practices in the implementation of AMR. And we hope that those stories can also inspire others. And we have created a fact sheet that is also available on our website. Finally, we also do targeted awareness-raising campaigns. Lastly, we are focused on younger generations, so we have created the Beat the Bug awareness campaign that produced an interactive video game on Fortnite, and at the same time came out with communication and teaching material for parents and teachers. Next slide, please. We are also supporting member states through evidence gathering and through studies. So, for example, in November, we have published the final report of a feasibility study on integrated surveillance systems for AMR and AMC. The study aimed to support member states in developing integrated surveillance systems, and it also identified enablers and barriers to establish such systems, including, for example, the establishment of independent coordinating bodies or investments in IT infrastructure to ensure data compatibility. And earlier this year, We have also published the final report of the study that was aiming to design a monitoring framework to assess the progress and results of our AMR actions. And this study developed a set of indicators in addition to the targets that we already have. So, for example, we will be using indicators like the level of intersectoral coordination in the implementation of NAPs, or the extent to which diagnostic testing is available in each member state. And these indicators will help us to understand whether our actions are effective and have an impact. Next slide, please. And with this, I pass the floor to Ivan. Thank you.
Thank you very much, Hannah, for outlining the main elements we do at our unit level. And I'm going to talk about the European Union action on antimicrobial resistance and healthcare-associated infections, the so-called EU JAMRAI 2. This is a second version. There was a previous one, also lasted for years. And today we will focus on the work being done so far in this joint action. Next slide, please. So why is this joint action important? It is a tool that the EU Commission has to implement the EU AMR policies that Hannah has described in the previous presentation on the ground. So this tool works as a kind of bridge between the EU strategic declarations already covered by Hannah in her presentation, the country recommendations, the 2017 action plan, and so forth, with the concrete national action plans from member states. This joint action has a budget of 50 million euros coming from the EU for Health project, and also 12 million euros from member states. So it's a total budget of 62 million euros. It covers 30 countries, all member states, the 27 member states, plus other partner countries like Iceland, Norway, and Ukraine. There are 128 partners in total, so it's a big joint action, and it has 10 work packages. The goal of the joint action is to foster a One Health community in Europe. That means covering the three main areas, human, animal, and environmental sectors, to try to move from working into silos to a coordinated way between these three sectors. Next slide, please. I mentioned in the previous slide that there are 10 work packages. I will now briefly summarize the work of the main work packages, work package 5, 6, 7, 8, 9, and 10. And I will start with work package 5. This is the so-called engine room. This is the work package that coordinates the direct support to member states. And the three main achievements of this work package. So I mentioned that JAMRAI 2 started in 2024 and will last until 2027. It's a 4-year joint action. So we are midway, exactly midway through the joint action. And I will explain the achievements so far in the 5 work packages that I'm going to explain. So the main achievements of work package 5 are, first of all, the creation of a network of policy officers. So the 30 participating countries have policy officers. Then there is a support program that has been developed. This has implied the conduction of 27 interviews in the 27 countries to map the needs of the different member states, and also to develop support to the constitution of the roadmaps on the national action plans of the member states. And finally, another of the achievements of Work Package 5 is the allocation of a budget of 1.5 million euros dedicated to training or mentorship, like direct learning from countries, from the best practices of other partner countries, and so forth. Next slide, please. In this slide, I will briefly explain Work Packages 6, dedicated to stewardship, Work Package 7 to Infection Prevention and Control, IPC, and Work Package 9 on access. I will start with Work Package 9 on access. The goal of this work package is to balance the access to antibiotics and the appropriate use of the antibiotics. The main achievements of this work package having to identify vulnerable supply chains in 17 countries, both for human and veterinary antibiotics, and also to develop tailor-made interventions such as joint procurement pilots to prevent shortages of antibiotics. And both in stewardships and IPC, the main goal has been the harmonization of core competencies, both in IPC and in antimicrobial stewardship. And there has been in both the work packages a big effort in training, especially in training tools for professionals of the 3 sectors in 28 countries. Next slide, please. Work Package 8 is dedicated to One Health surveillance. The idea is to close the gap between the 3 sectors, the animal, veterinary, human and environmental sector, to try to have an integrated One Health approach to surveillance. And the main challenge has been to harmonize data, because these sectors are quite different between each other, and the harmonization of data is probably one of the biggest challenges of the core package. The main 2 achievements, for example, in the animal sector, has been the expansion of the EARS-VET, which is the surveillance network for disease animals, and now covers 19 countries. This allows real-time resistance data to veterinary professionals. And in the case of environment, the main achievement is the developing of the EARS environment, which is a surveillance program for AMR in the environment, mainly in wastewater. At the end, the main goal of this Work Package 8 is to develop a unified dashboard for decision makers, connecting the 3 main sectors. Next slide. And the last work package is Work Package 10, focused on awareness and communication. Among the main achievements, we have the promotion of the AMR symbol, which we all have here. We have distributed more than 4,000 of these pins, including at the United Nations General Assembly 2 years ago. And now we are even going further and try to include it in the package of the antibiotics, which is an ongoing discussion at Parliament and Council level. We have also developed success stories, like the launch of the faxes with success stories that has been quite recent. These are linked both to the Santi and Jan Roy websites. And there have been other communication campaigns like AMR art memes through social media, and more recently the AMR sketching campaign that has taken advantage of the Awareness Week to launch a quite successful campaign in social media, mainly in Instagram. And also we have created a network of communicators with all the 30 participating countries of Jan Rai that meets several times a year. And next slide, and the variant, the next steps, we have a General Assembly, which is a major milestone in this halfway of the project. It will take place at the end of January. And the idea, the main focus is that Jan Rai will perdure in time. We create permanent tools that will be used beyond the end of the project. So we are moving from saying to doing, and Jan Rai proves that when Europe acts together on One Health, we deliver results. Thank you very much. Beatriz?
Thank you, Ivan. And I will briefly conclude this presentation and coming back to the slide used before by Hannah, with the different objectives of the Council recommendation on AMR, how we could explain some of the global action we are involved and following. So also, as mentioned by Ivan in his presentation, you are probably aware of the UNGA high-level meeting on AMR that took place last year in September, where a political declaration was agreed between all UN member states. And this political declaration contained commitment and targets across 7 country areas. So many of the targets are similar to the one we have already from the Council recommendation and other EU action, but it has also broader coverage. And for example, it includes also a target in financing to mobilize 100 million of dollars to support at least 60 countries having funding that, because this is something seen in tracks and also with other tools that many countries, well, some countries still are not developed an app, but many others would have developed an app, but then they don't have sufficient financing for implementation, which is a key issue. And after this high-level meeting on AMR and the adoption of the political declaration, there have been some other meetings, but also some of the action we are now following this year, because as also mentioned in the political declaration, the quadripartite, so WHO, WOA, FAO, and UNEP, they were already working together, but there is also a formalization of the quadripartite Joint Secretariat on AMR, and together these four organizations are working to update the Global Action Plan on AMR by next year, and also to establish the independent panel on evidence for action on AMR, which was supposed to be launched by end of 25. But after this 2 months of consultation, we are still waiting to see if it can actually be launched still before the end of the year, or if that will be postponed to beginning of next year. Also, the UN Secretary General will provide implementation report, and there will be another high-level meeting on AMR in 2029. Next slide, please. But before that, in between, let's say, we have also the so-called high-level AMR ministerial conferences. So last year there was one in Saudi Arabia, in Jeddah, in November. So that was more or less 2 months after the UNGA political declaration. And in those commitments, countries agree to translate some of the commitment from the political declaration into practical commitment and action. There is also the formalization of those high-level AMR ministerial conferences that will happen now every 2 years. And the next one will be actually next year in June or in Nigeria. And here you have a link because they have recently published a webpage with some information about the ministerial conference, and there is also a call for abstract to be presented there. Next slide, please. And I will finish with by mentioning some other global initiatives we are supporting and we are involved too. So the AMR Multi-Partner Trust Fund, which is a tool which was also mentioned in the UNGA political declaration. It already exists since several years. I think it was mostly launched after a call from the Netherlands. And this tool is mostly supporting countries in implementing their NAP, mostly low and middle income countries, with an approach to help them to for a tailored NAP and implementation. And we hope that this dedicated fund to AMR, because we know there are other funds that can also contribute to helping countries to tackle AMR, but that one is really focused for this. And there is a call in the UNGA political declaration to increase the number of contributors. So this is something that we hope to see also in the future, but we continue participating to this fund. And for stakeholder engagement, so before in the presentation, you've heard what we are doing at the EU level between member states and stakeholders. Yeah, our network, our platform, also all what is done via Shanghai. But also just for you to be aware, there is this AMR multi-stakeholder partnership platform at global level. It was launched recently with some funding from us. It has now more than 250 members and represent different sectors, so research and academia, civil society, government, private sector, financial institution, and it is organized by action group where interested participants can discuss a specific topic, and it is also organized by cluster depending on your constituency. You can also discuss with peers. And the idea is also to share best practices and enhance knowledge. So in case you want to have more information, you have also the link to this partnership platform. It will have also a plenary meeting just before the ministerial conference in Nigeria next year. And with this, I think that was it. We have also some other global action like participation with colleagues in, for example, declaration of G7, G20, but I will not enter into detail. And on that slide, you have also the picture of the symbols that Ivan mentioned before. And if you have any question, we are happy to answer. Thank you for your attention.
Thank you very much for your presentation. I would ask now if anyone has a question from our face-to-face audience or online.
For your presentation, I would ask.
Sorry, the connection is not great. It's breaking up. Can you hear us?
Well, we have a question here in the audience, so maybe we can start here.
Good morning, Uli Klümper from TU Dresden. Can you hear me?
Yes, but the quality of the sound is not ideal on our end, so...
All right, now I'll try to keep it short. So with the new environmental surveillance coming up, there will probably be a lot of sequencing data and so on. So in before, I think data harmonization was mainly about results, while sequencing data allows reanalysis to go back in time, looking for things that might have appeared earlier and now cause a problem. So is there a plan for a big data sharing initiative or platform to also access like raw data and so on?
Can you switch? I hope I understood the question. This falls within the remits of Jan Rai. Work package 8 on data and IPC. I mean, has the objective to develop a big dashboard where surveillance data from the three main sectors will be shared with professionals across Europe. I'm not so sure about mental health data, but for sure that surveillance data will be found in this dashboard that will be that is still in development, but hopefully will be shared with the general public. And there we should be able to access data from the different professional sectors, the environment, animal, and human sectors.
Thank you very much. We don't have time for more questions. We have to move on to the next speaker. So thanks for your presentations, and I hope you enjoy the rest of the session today. And we're going to move on to Uli Klümper, that he's going to present to us the ecological and evolutionary drivers of environmental antibiotic resistance. He's a senior researcher in the Institute of Hydrobiology, Technical University of Dresden. So welcome, Uli, and the floor is yours.
Perfect. Good morning, everyone here in the room, as well as everyone online. So I have today the opportunity to give a nice little keynote talk about the ecological and evolutionary mechanisms that play a big role in AMR in the environment. So if we think about chemical pollutants, it's rather easy if we think about the dynamics of them. They enter the environment, they might spread around, they get degraded, and then they're gone. However, if we think about AMR, it's very different because it's a living pollutant. So there's a lot of different mechanisms that play a role in how resistant genes are moving through the different compartments that all make up the One Health continuum. From humans and food animals, where a lot of the antibiotics are used, to the more natural environments, to wastewater treatment plants, where a lot of the waste is ending up, then to soils, and back into the human sphere. So in our research group, we are mainly interested in looking at what do these mechanisms mean, how do they actually work. And if we think about the environment, one of the crucial facts is that in the environment, microbial communities are rather complex and consisting of a lot of different members that interact and have certain relationships with each other to keep each other's fitness alive. If now we have bacteria that are enriched in the anthroposphere, that includes both the human as well as farm animal environments, they can enter into these communities. For example, they can be non-resistant bugs, they can be resistant, having the resistance on a chromosome, or they can be resistant with having resistance plasmids. So if we think about the processes that are involved now, I think the first process is obviously invasion. So if something gets released into the environment, how well does it invade into this microbiome? Does it stay there? Does it persist? For this persistence, an important process is selection. So number one, all these bacteria that might be exactly the same except for resistance might compete with each other. Which one has the best growth rate? Which one is most adapted to the environment it's invading? At the same time, they all have to compete with the natural members of the community and see, can they survive in that niche? Can they find a niche that is suitable to them? And then obviously there is horizontal gene transfer. So once they have established themselves, or even if they are only in those communities transiently, they can spread the plasmids to natural members of the community, which in turn can also spread it back to the community members. And all of this is not happening in isolation, but it's happening with a lot of different environmental drivers that determine the rates of these processes. And these can be, for example, biotic interactions, similar to competition or facilitation, but it can also be abiotic factors like temperature. We yesterday talked partly about global warming, which can be stress like pollutants, antibiotics, heavy metals, and so on. And all these might influence how well these processes are working. So today I want to take you through a few of these processes and a few of the studies we did on these processes. I want to start with invasion. So one of the things is we constantly have invasion into natural microbiomes, for example, in aquatic microbiomes, through the release of wastewater. So all these resistant bacteria try to invade, and we obviously don't like that that's happening. So if we want to stop this invasion from happening, one of the things we need to do is understand what determines the success of these invasion dynamics and how can we mitigate it. If we think about a simple river that gets ARGs and ARBs from wastewater, so we have our river microbiome, which is usually in the form of biofilms, the bacteria start to invade. However, in wastewater, we don't have antibiotic-resistant bacteria and antibiotic-resistant genes in isolation. We have them together with a lot of other pollutants like metals, pesticides, personal care products, pharmaceuticals, which cause stress to both the invaders, but especially to the river microbiome that is not usually used to them. So in our case, we did an invasion study to see how does a microbial community resist the incoming foreign bacteria, and what is the effect of the additional stress that is coming in. And we did that study based on resistant E. coli moving into natural river microbiome biofilms. And we used copper as a stressor, mainly because we worked in a project together with an Indian group who has a river around the corner, which is one of the most heavily polluted with heavy metals. So I wanted to see in the German rivers, is it actually similar with regards to effects? So we took two rather pristine German rivers that are usually not exposed to wastewater and looked at their river microbiome. How did we do that? Well, we created artificial exposure units, which basically is a fancy word for we printed some plastic carriers, we put some glass microscope slides on it, we added them on a concrete slab, added a cage over it to protect it, and then we dumped it in a river for 1 month. So on these little slides, we can have biofilm growing, we can thereafter move them to the lab, and then analyze them. At the same time, we sampled a lot of the river water. That's one of my students, Kenyon Bagra, who did all of that. And then we moved our setup to the lab. So in the lab, we have like these artificial flumes, which simulate rivers with water flow. We fill them with sterilized river water, and then we can put these artificial exposure units in there and destructively sample them. So how does that at all work? So we usually put them into these flumes for around a week. After that, they are adapted to the lab conditions and then not really changing in diversity or biomass anymore. And then we can run our experiment. We invade them with either E. coli or E. coli under stress. And we get our 3 treatments where we have a control treatment where nothing happens, one with E. coli no copper, one with E. coli and copper, and we do that for both rivers. We then perform destructive sampling, so we take out 1 slide from each of the artificial exposure units at each time point. We do that initially when we take them out of the river, at day 0 before treatment, at day 1, 2, 7, and 14. And then we analyze them. How do we analyze them? Well, we run qPCR to see how many bacteria are in the biofilm with 16S. We look at how many of them are the E. coli that invaded, and we run 16S sequencing to look for the diversity of the communities. If we look at the initial invasion of E. coli directly 1 day after we added it, we see very clearly, okay, E. coli is able to invade, and since it's log scale, we have approximately 1% of E. coli in the biofilm. So this is without stress. If we now add the stress, what we see is we don't see anything. So basically, the initial invasion is exclusively based on how much area is there to attach to, how many E. coli do we add, and then everything else is stochastic. But what about the invasion success, persistence, and so on? So if we look at what happens over time, we see here for our non-copper stress treatment, it goes clearly down for both rivers. So the E. coli is lost, it's not adapted to live in a river biofilm, not a big surprise. But if we add the stress, suddenly we see that E. coli is increasing in numbers or persisting in numbers, so it is not suddenly outcompeted by the river microbiome anymore. So why is that? That can have 2 reasons. So if we look at the left-hand side on the axis, it's E. coli per 16S. First reason could be, well, under stress, more of the bacteria are dying, so there's less 16S, which means even with the same number of E. coli, we would see this upgoing trend. Also can be that E. coli is actually increasing in numbers. So first thing we looked at was what is the total 16S numbers, and as you can see here, there's barely any difference. There's some variability, because some of the biofilms are a little thicker or not, but overall there is not really any trend with regards to that. So there must be another reason why suddenly E. coli is increasing in numbers. And one of the things we looked at was the diversity of our biofilms. So if we look at our control treatment where we added nothing and where we added only E. coli, you see that the diversity, like I said, we did 7 days of acclimatization. Afterwards, it's very stable with regards to how diverse the biofilms are. But if we look at our copper treatment, we see that consistently the diversity is lowered. So what does that mean for invasion? So if we look at microbial communities, we have one that has no stress and a high diversity, one that has low stress, that has stress and a low diversity. So here it should show low diversity. The numbers are the same, just for illustrative purposes that there's less bacteria, But the important thing is that there's less diverse bacteria, less different colors. So what does that mean if we think about ecology? Well, bacteria like to live in their little niches. If we have a lot of diverse bacteria, we've got a lot of niche occupation. Close to all the niches are filled with a suitable bacterium. However, if we lose some of these bacteria due to stress, we suddenly see that there's empty niches. So now if resistant bacteria invade, that means high competition, low invasion success, and low competition, high invasion success, where the resistant bacteria can suddenly find a niche and don't have to compete with the natural biofilm bacteria that are a lot better adapted anymore. So maintaining diversity can serve as a short-term barrier to the invasion by antibiotic-resistant bacteria and their transferable resistant genes. So from that, we should actually be able to conclude diverse environmental communities should, in the long term, if it's not about recent pulsed introductions, host less ARGs. And that's exactly where I'm going to take you next. So we did a big EU project to test this hypothesis. So what did we do? We went across different European countries. We sampled pristine soils and rivers from forests and from rivers that are not influenced by wastewater influent. Main reason for that being is we need pristine environments so we don't see the ARGs from a very recent invasion, some manure that is applied recently or wastewater being dumped recently, which means here we really see what happened in the long term due to, for example, aerial deposition and so on. We then did 16S RNA-based sequencing and high throughput qPCR of around 30 antibiotic resistant genes. This was a project funded by the EU Biodiversa and it included 7 different countries, and we took more than 200 samples of these pristine environments. So what did we see? First thing I want to show you is how many of the 30 tested ARGs did we detect in the individual communities. And if we look at the rivers, we see we have around 18 plus minus 5.9 ARGs that are detected, and there's no relation with the richness of the community. So I'll show you a few other metrics of diversity later on. So we see no real effect. However, in soil, where we have like a little bit less of a detection of ARGs, which makes sense, that there is this clear trend of the more diverse a community is, the less resistant genes do we actually detect. If we look at the evenness of the communities, so are most of the bacteria appearing at similar amounts, we see that the effect is even stronger. So no effect for the rivers, but a very strong effect in the soils where the more even a community is, so the more different bacteria are there at equal numbers, the less ARGs are there, which makes some kind of sense because for niches to be completely filled, you need a certain critical number of the bacteria to fill it. And then if we look at the Shannon diversity, we see the same trend, which makes sense because Shannon diversity is basically a mixture of richness and evenness. However, obviously, if we think about risks of ARGs, we probably don't need to think about what is there or what is not there. We need to think about their individual abundances. So for each of the 30 different resistant genes, we looked at how does their relative abundance correlate with diversity. And you'll see quite a few of these graphs now. So you see on the very left, all the different resistant genes that we tested. You see down here, the Spearman's row of the correlation. So is it a positive correlation with diversity or is it a negative correlation? If it goes left, negative. If it goes right, positive. And only when they are full, like here, is it a significant relationship that actually means something. And if we see here, similar to how many ARGs we could detect in the rivers, there's not really any effect on the richness. There might be one single type of ARG where it's different, but it doesn't really mean something if it's 1 out of 30. And also, if we look at the other diversity metrics, it's all the same trend. However, if we look at the soil where we saw clear effects of detectability, we also see strong negative effects on the relative abundance of these ARGs. So it seems like in soil, actually, where we have kind of a very stable environment, a stable community, it's not dynamic like in the river where like biofilms are constantly exchanged, ripped off, and newly building. we see this very nice effect of diversity that limits how many resistant genes are there in the environment, which is kind of a nice and positive effect. So these ARGs that now are in the environment, what can happen to them? I want to show you a few of our things that we did on the plasmid transfer, where they might not be able to persist long because of high diversity, but they might leave their resistance load encoded on plasmids behind. I think most of you know how horizontal gene transfer happens. It can be by transformation and transduction, which are both kind of stochastic events that happen randomly. However, what is really interesting and causes big problems is conjugation, because as soon as resistant genes are encoded on plasmids that have low cost and low selectability, then we have a big spread also into the human sphere. One of the things that I did very early on when I started studying all of this is, well, plasmids, they're really small, even smaller than bacteria because they're inside of the bacteria. It's really difficult to study them because they're just not visible, just like tiny pieces of DNA. So one of the things we did is we created this fluorescent-based assay to detect conjugation. How does it work? So we have a donor strain in which we have on the chromosome a red fluorescent gene. We then have a plasmid in there that has a green fluorescent gene. At the same time in the donor, we have a repressor for this GFP. So inside of the donor, M cherry, the red fluorescence is expressed, GFP is not. So if we now add that into a recipient community, for example, an environmental one, we can see how often does conjugation happen by quantifying these green cells, and we're also able to look at who are they, the ones that are permissive towards the plasmid. So if we look under the microscope, we see, for example, here, red donor cells, green transconjugants that took up the plasmid, and around it, it's not emptiness, it is cells from the environment that were not able to take up the plasmid. And so one of the things we did early on was, let's quantify how often does plasmid transfer happen, and who are the ones that actually take it up. So we extracted soil communities, we added them on a filter, added our donor strain, we put them onto soil extract, and we're then able to, under the microscope, look for these green microcolonies, which allows us to quantify transfer events growth independently. And at the same time, we're able to scrape off all of this biofilm, put it through a flow cytometry device, and sort out all of the green transconjugants, sequence them, and look, who are they? So who were they? Well, actually, they were a lot more diverse than what was known in before. So we see here in white our donor strains in the gamma proteobacteria. It was very well known that plasmids can spread to beta proteobacteria, to alpha proteobacteria, so to closely related clades. But when we did our sequencing at high throughput, we saw that all of these different groups also are able to host the plasmids. And one of the big surprises was that transfer from gram-negatives to gram-positives, which have completely different membrane structure, are actually more or less the rule. So this happens quite a lot. And that also explains how, for example, gram-negative integron cassettes end up in these gram-positive bugs. So overall, I recently had a look, since this study was like around 10 years ago, what is actually known by now if we look at these model plasmids. So in most environments, they are able to invade like 2 out of 10,000 different cells. Might not sound like a lot, but if we think about soil, for example, if we take a spoonful of soil, that's like 10 to the 9 bacteria, which means there's 100,000 cells in there that are potential hosts of these resistance plasmids. From the studies that have also used this approach over the years, which are by now over 50, we have 16 to 20 different bacterial phyla that are able to take it up. So these plasmids, they can spread really far, and it's thousands of diverse species, including plenty of pathogens. So plasmid transfer can happen to a lot of different bugs. But what actually is driving how often it happens? We obviously know that chemical pollutants are doing it, but there's also other things. And so we recently did this case study on microbial interactions, which are very common. We can stop pollution. We probably can stop microbes from interacting. And so one of the things we know is protozoa, a lot of them like to eat bacteria. Bacteria don't really enjoy that. So what's happening is protozoan predation leads to bacterial stress. When bacteria are stressed, they look for new genetic information to take up that might help them to deal with the stress. Protozoan predation also can cause aggregation and biofilm formation among bacteria, which means they are in close proximity. If they're in close proximity, they can interact, they can spread plasmids to each other. At the same time, protozoa also use chemicals, for example, heavy metals or natural antibacterials, to lyse the cells that they have eaten up and taken into their food vacuoles. And obviously, those things can also co-select for AMR and increase plasmid transfer if resistant genes against these compounds are co-encoded on the same plasmid as resistant genes. So based on that, we had the hypothesis, well then, protozoan predation, it probably elevates bacterial conjugation rates. So we used our system, same as before, this time with Pseudomonas as a normal environmental bacterium as the donor and recipient. We looked at selective plating. How often does plasmid transfer happen in this single strain environment? And we ran it in a way that we added donor and recipient into a wheatgrass medium, because in there a lot of the protists are quite happy to live. And we used 4 different protozoan predators, Tetrahymena and Paramecium, which are ciliates, which use like a lot of these tiny hairs to move around and to also get the food into their, well, let's call it mouth hole. Then there's amoeba. Amoeba, I guess you know, they build out these pseudopods, little feet, they are amorphous and can move around and eat the bacteria. And then there is one flagellum, which is Poterosporum lacustris. And that has like these long hairs that they also use to move around and can also get food into their mouth and then digest it. So let's take a look what happens to bacterial numbers when we add our different predators. So for the amoeba, very clear, the more amoeba we add, the less bacteria there are in the final assay. Same true for flagellates. 3.8, 7.3-fold decreases. So these protists are very happy to eat the bacteria. Same is true for the ciliates. For the Paramecium, it's a bit more of a steep decline after a certain critical concentration. For Tetrahymena, it's also very clear linear. So bacterial numbers are reduced with increasing predator abundances. Well, and before I said, bacteria need to encounter themselves to undergo plasmid transfer, which means lower numbers should mean lower encounter rates, lower plasmid transfer rates. However, what we see very clearly is we see, despite the lower numbers, an increase in the transfer rates. So for the amoeba, around 2-fold, for the flagellates, around 3-fold, for Paramecium, 5.4-fold, and suddenly for Tetrahymena, this massive spike in increase by like 24-fold, which was a lot more than for the others. So we were thinking, okay, what is it that causes Tetrahymena to have this far stronger effect than the other bacteria, the other protists? And first thing we thought, maybe it causes an increased level of stress. So we measured stress based on reactive oxygen species produced by the bacteria. And you can see here, with increasing predator abundance, we see the bacteria having more and more intracellular stress. That makes sense. However, that was the same for all 4 of the different protists. So couldn't explain the difference. We then looked at the expression of plasmid transfer genes. We see, some of the plasmid transfer genes at medium or high predation are up or down regulated, but it was a very mixed picture, very difficult to predict, and also similar for the different protists and different plasmids. So finally, one of the students came to us and said, I have an idea. I see something weird under the microscope. And what they saw was these little round structures. And what they actually are is, when we went to the literature, is Tetrahymena, other than the other 3, is very regular when they have a lot of bacteria to eat. So there's plenty of food around. They fill up their food vacuoles. They start digesting them. But rather than digesting all of the bacteria in there, they rather poop out some of them and start feeding on the ones that have just recently been filled again. So basically, we have these protist poop particles in there, fecal pellets. And when we looked at them under the microscope, we can very clearly see that there's still a live red donor bacteria in there. And at the same time, also a lot of these green transconjugants that have taken up the plasmid. So this could actually be one of the reasons, because if we think about these fecal pellets, we have close spatial proximity of the bacteria. They get filled in there by the protists. We've got high nutrients because a lot of the other bacteria have been digested, and whatever is in the bacteria that have been digested is probably a very good growth medium for the ones that survived. And we've got selective pressure from whatever the protists used to digest the bacteria. So it's kind of the perfect breeding ground for elevated horizontal gene transfer rates. With that, I think I've taken you through a few of our studies. I want to go to the take-home messages. So one of the things we can take home is once antimicrobial resistance becomes established in the environment, it is extremely difficult to control. We have low costs of resistance, selectivity is very low, and but we have diverse biotic and abiotic drivers in addition. For example, protozoan predation, which can further amplify its spread. That means we can probably never completely stop it. But what is positive? High microbial diversity can act as a natural barrier against the invasion of resistant bacteria and mobile resistant genes. So in addition to limiting selective pressure and limiting exudation of ARGs into the environment, we should probably protect environmental health to maintain diverse communities, because it's crucial to limit AMR dissemination. And that's number one, One Health, where environmental health is aiding veterinary and human health. So what we definitely should not do, and where we should probably reconsider, is, for example, co-release bacterial stressors together with our resistant bacteria, because that erodes this barrier effect. And by that, we suddenly gain bigger problems. With that said, I'd like to thank most of our institute, no, actually all of our institute, including bachelor and master students, ONE Bridge for allowing us to get together here, all of the other projects that were involved, collaborators and so on. And thank you for listening. I'm open to questions.
Thanks for this clear presentation. We have time for one question if someone among our audience.
Roberto Spuri, University of Camerino, Italy. Thank you, Uli, for the nice presentation. Very stimulating. Question about the if the setup that you have shown for the stressor at the very beginning, copper effect, I guess can be replicated with any other kind of stressor. What about a stressor that is found in wastewater treatment plants, like antibiotics? Which kind of effect would you expect from repeating the same experiment under a stress which is tightly correlated with AMR?
So I think if we talk about antibiotics, we purposefully didn't use it in the very beginning because we wanted to be able to separate invasion from selection. If we combine the two so that there's directly an active selection for resistant traits, I think there is two potential outcomes. The first outcome is, well, it will obviously favor the bacteria from the wastewater that are naturally resistant, that can invade. The second option is, it really depends on which community you are invading. So if it's a community from a river that has for a long time received wastewater, it might be already enriched in resistant genes, and by that have good competitive ability to withstand the invasion by new resistant bugs. But if it's a more pristine community, I would expect that the diversity drops would be far higher than, for example, for copper, because heavy metal pollution is a lot more common than a strong antibiotic pollution. So I think releasing antibiotics is probably never a good idea for all of these processes involved, because it selects, it increases stress, by that increasing plasma transfer, it lowers diversity, and so on. So compared to heavy metals which appear in the environment, I think the effects would likely be stronger if it's not a community that has already been so damaged by us that it's basically beyond rescue.
Thank you very much. We don't have time for more questions, but we will catch you on the break. And we move on to the next speaker, which is Sara Soto, the head of the Viral and Bacterial Infections Program of IS Global in Spain. And she's going to talk about AMR as a silent pandemic, the global and EU perspectives of it. Welcome, Sara.
And it's a pleasure for me to be here, at least in a online mode, because I have some family problems. I would like to give you a perspective of the antimicrobial resistance problem, and also some of the global and European perspective in this to solve it. You can see my screen? Yes, I saw that. Okay. What is the problem? The problem is that, as you know, we have a high impact of antimicrobials in health in the 60s and the 70s, but 50 years later, infectious diseases are the second cause of worldwide mortality. This is due to the increased emergence and dissemination of the resistance among different microorganisms, viruses, bacteria, fungi, and parasites. The numbers are very terrific because we have 33,000 annual deaths in Europe, and also 700,000 annual deaths worldwide. And this also costs extra 2.5 million of extra days per hospitalization a year, and an economic cost of 900 million euros every year. I suppose that all of you know this paper published in 2022 that was like the start of this awareness about this problem. That was the global burden of bacterial antimicrobial resistance in 2019. And this problem had two scenarios. One is the deaths attributed to antimicrobial resistance, that is the caused by this infection by antimicrobial resistant bacteria, but also the burden of the deaths associated with antimicrobial resistance. That was the first cause is another diseases, but they are more severity due to these infections. We are speaking about a number like 1.75 million in the first scenario, and 5 million of these in the second scenario. The distribution of this impact of antimicrobial resistance is different depending on the area of the world. No? You have the biggest mortality in the Western Sub-Saharan Africa, and the less one in Australasia. And also it's different depending on the bacteria. We have different scenarios in Central Europe, Eastern Europe, and Central Asia. As you can see, it is more related to E. coli. In high-income countries, it's more related to Staphylococcus aureus, but in Sub-Saharan Africa, it's more related to Klebsiella pneumoniae. It depends on the part of the world. And the expectative are not so good. If we don't do anything, we can expect that in 2050, there is more death for antimicrobial resistant infection than from cancer. And you know the cancer is the biggest or highest epidemic today. No, I suppose that someone had some cases of cancer in the family or friends or something like that. But the scenario is that we have overpaid for 2 million of this. But how we achieve, how we arrive to this situation? Okay, we have done a bad use and overuse of antibiotics in human health. We have now we have bad prescriptions. We don't take the complete treatment. We don't put the antibiotics when the finish in the appropriate place. We take for the water and the blood. They are now in Europe, at least it's good regulated, but in some part of the world it's not regulated at all. We have also a high consumption and abuse and bad use also in animals' health. And they are used antibiotics in animals for growth promoters, also for treatment, but also for prophylaxis. And this picture is one that we had done some years ago. No, the farm animals eat antibiotics in the feed. And also we have contaminated the nature. And all the contaminants as we not discharge antibiotics themselves, we also have some chemical substances like pesticides, and other ones like endocrine disruptor, heavy metal that can also help to the emergence of antimicrobial resistant microorganisms. That we can see that there are association with heavy metals and antimicrobial resistant genes. All these things had supposed to be the cause that we had emerging and disseminated Patras, the bacteria that are resistant to most of the antibiotics that we use in clinical settings. And this is the big problem in this sense. Thus, the World Health Organization had launched a list of priority pathogens on which we had to search new measures such as new antibiotics. We need new antibiotics. We don't have treatment sometimes in our hospital to treat Acinetobacter baumannii, Pseudomonas aeruginosa, Enterobacteriaceae that were pan-resistant, is resistant to all the antibiotics that we use in the hospital. If you see this scheme, this is in the upper part, we have the antibiotic deployment, and the lower part, we have the antibiotic resistant observed. As you can see, in the 30, we have the sulfamides, but in the 40, we have the resistant bacteria to sulfamides. But here in the last part of the graphic, you can see that we have daptomycin in the 2004 and daptomycin resistant in 2005. 1 year later that the daptomycin go to our hospital, we have bacteria resistant to that. And this is increased, the crisis is increased due to that we had no antibiotics in the pipeline. The pharma companies, we don't have to invest in antibiotics because they are not take a lot of money for this. We had a treatment of only 10 days, and the investment that we had to do it to develop a new antibiotic is so high. This decreased between the 80s to now, that for 20 antibiotics more or less to 6 in the 2014. But the market is not is good. We have a market opportunity. It's estimated that the global antibiotic resistant market was valued in 7.80 billions in 2017, and this increased in 5.6% in 2045. This is a good place to develop antibiotics. We have a good market. However, we need new antimicrobials with new mechanisms of action. In this sense, we need to have new families, new mechanisms of action that have activity against the priority microorganism considered by the WHO. I would like to put this sentences from David Cameron, the past former UK Prime Minister, because it reflects the problem that we have. It says that if we fail to act, we are looking to an almost unthinkable scenario where antibiotics no longer work, and we are cut back into the dark age of medicine, in the pre-antibiotic phase, in which, pregnant woman with an infected cesarean could death in this sense. But the race to develop an antimicrobial is very complicated, that's very long. We have to pass all these phases, the discovery stage and the development stage. The discovery stage is formed by the identified target, identified compounds, established activity, select clinical candidates. And the development phase stage that have stage safety, human clinical trial phase 1, 2, and 3, and finally human clinical trial 4, and regulation and implementation in the hospital. We have see that if we have this, we develop antibiotics from the screening phase until the commercialization and pharmacovigilant survey. We have from 10,000 compounds. We finish with one compound and 9 million euros and more than 10 years of development. This is the summary that we had 12 to 15 years of development. The cost of developing a new antimicrobial is 1 or 2 billion euros, and with only 1% of success rate. For the reasons the pharma companies don't want to invest in this antimicrobial resistance problem. And also we have different obstacles. These obstacles had called the Valley of Death, and we had two principal valleys in this race to develop a new antimicrobial. The first one is to pass from the results obtained in the university or in a research institute in academic to the industry, and we can do it by patents, licenses, or to the creation of a spin-off. That is not easy. And we have in the second valley is the industrial technology transfer. We have taken a pharma company that is interested. They finish the development. They do all the tests, but we have to implement it in the clinical setting. And this, we have another big valley that is the clinical essays, the regulation, also the approval for the medical agencies. In the last years, we had several initiatives to increase the support to the pharma industry to develop a new antibiotic. The first one was called the Netflix model. That was the pilot was done in UK and was very successful. That was not to because the industry company, the pharma company, didn't obtain the money for the sales of the antibiotics. If not, the health government establish amount of money, like a subscription to this antibiotic. Then the pharma company obtain 1 million, some samples of €8 each year for this antibiotic. It was used or not used in the clinical settings. This innovative mechanism of funding, in addition to the reimbursement reform that the healthcare taxpayers could reimburse hospitals for antibiotics and after praise, give us the opportunity to have a sustainable antibiotic market. The patients gain access to new and innovative antibiotics and revitalize antibiotic development. This is the Netflix model that was very successful in UK, and now it's done some pilots in some European countries. But we had also to develop another alternative to antibiotics. We had several alternatives, but none of them could completely exclude antibiotics. The first one are phages. Phages are well used and very effective, but have several limitations. We need to know the bacteria. We need to have phages in a cocktail because, as you know, the phages are very specific for each bacteria, not for each species. And if you use in combination with antibiotics, we can be a possible solution for antimicrobial resistance. We have also to develop vaccines. They have a great potential against superbugs, thanks to recent technological development, modification of microorganisms, biconjugates, but many years of testing are needed before they are applied to the public. Also, we had a microbiome that was it's the fashion now, and we had to modify the bacterial digestive tract to stimulate the prevention of acute infection by superbugs, but it's still in experimental phase. And also we have antibodies, but antibodies, they are very, we need to be specific to a particular type of bacteria, and its monoclonal antibody must follow its own clinical development process. But none of that, as I commented, could completely exclude antibiotics. For the reason we need to continue with this development. But in addition to have or to develop new antibiotics, we have to stop the transmission of the antimicrobial resistant bacteria, antimicrobial resistant genes from animals to human and to the environment. We have to close or to stop this dissemination in this One Health point of view. In this sense, we had several European policies. In June 2023, the Council adopted the recommendation of stepping up European actions to combat antimicrobial resistance in a One Health approach. The objective of the Council recommendation, there are no recommendations, there are no obligations, are to strengthen One Health and national action plans on antimicrobial resistance, to reinforce surveillance and monitoring antimicrobial resistance and antimicrobial consumption, to strengthen infection prevention and control, strengthen antimicrobial stewardship and prudent use of antimicrobials, recommend targets for antimicrobial resistance and antimicrobial consumption in human health, improve awareness, education and training, foster research and development, and incentives for innovation and access to antimicrobials and other antimicrobials medical control measures, increase cooperation, and enhance global actions. Several talks that we had, I suppose that we had also to speak about the success for the National Action Plan on Antimicrobial Resistance in Spain, that tried to board or afford all these recommendations for the European Commission. And also, we had an update on the antimicrobial resistance related uses in several environmental possibility contexts. And in the last 2024, in November of the last year, there are a directive for the European Parliament and the Council related to concerning to urban wastewater treatment. And in this new directive, they are including the surveillance and also to treat the surveillance of antimicrobial resistance in urban wastewater treatment. But now it's not clear what is the market that we have to use and what type of tests we have to apply in the wastewater treatment plant to do the surveillance of antimicrobial resistance. But there are a lot of work done that say that wastewater treatment plant are the hotspots of antimicrobial resistance dissemination from human to the environment. But this is a work that we had to do all together. We had to work together, the science, academia, research institutes. We had to work together, the pharma industry, to perform or to develop new antibiotics, to help us to develop the new antibiotics. We had also to work with clinicians, with veterinaries, with ecologists in a One Health vision. We had to have the support of the governments in funding and also in help us to implement these results. And also we had to have the support of the community. We had to work in training in education. We had to work in awareness in the general public, because the action for the people is important in this to stop the antimicrobial resistance problem. All together got to solve this problem. This is the antimicrobial resistance symbol of the prudent use of antibiotics in Spain. And thank you for your attention.
Thank you very much, Sara, for your presentation. Are there any questions in the audience for Sara? If not, I have one. Maybe it's out of curiosity, being Spanish myself, but this part of the economic incentives that you described that UK is implementing for getting or fostering the cooperation with the pharmaceutical companies on the new development of antibiotics, how do you think this could be achieved in a country as Spain?
I don't know if in Spain, because we don't have we are now seeing in another European countries that I don't remember what it's doing now, but I think it's a good opportunity, because most in Spain we had some small pharma companies, some biotech that are very interested in developed antibiotics, and I think it could be a good opportunity, because the problem of antibiotics is you use in a short time. It's not so much money. It's not for a chronic infection like cancer or cardiac, or chronic. And this thing in Spain, it could be, I think the National Action Plan and the government is trying to collaborate and to foster this development of antibiotics. And I think with the resources in UK, it could be possible that we can implement it in Spain.
Good to hear your positivity. I'm glad. And I think there are some online questions, so I give you the floor.
Yes. We do have one question. Well, it was addressed to Professor Klamper, if that's possible. It's from Benedito Toalha from the online participation. The question is, how can One Health frameworks better incorporate environmental AMR drivers into national action plans?
Is it on? Yeah. That's obviously a complex question because it is complex processes that we have to take care of. So one of the things which today will also be presented by one of our postdocs from the group, and that I presented yesterday as an outcome of the workshop, is that we probably need to take care that we survey in the environment in a different way than we would in the clinic or in the veterinary sphere. For example, by looking at what is the mobility or what is the selection pressure that we expose our things to. And I think looking at, if we think about drivers, for example, concentration dependencies, and if we think about looking at ARGs, risk prioritization based on genomic context, those are two things that we can do. And once the data is there, we can create risk-based models, because risk-based models is what's currently definitely missing for the environment to actually quantify what is worth going against. And yeah, so I think those are the things that we need for integration in national action plans. It's creating data and setting up less selection.
Thank you. Are there any more questions online? No? I actually have one more question for Uli, because we have a few minutes from your presentation as well. I was wondering, also following these different stressors that you experimented with, have you considered to replicate these experiments as well, considering climate stressors such as drought or electric storms or so on?
So with regards to heavy weather events, there will be a talk by one of my PhD students later today, so I will not spoil that. With regards to climate change, so we did some studies on temperature and invasion of wastewater. So you would expect in general that wastewater bacteria, they are probably more adapted to warmer temperatures, and by that have higher invasion success. However, we saw something of the opposite. So when we did our flume experiments with wastewater as an invader rather than E. coli, what we saw was in the control flumes, higher temperatures caused increases in the naturally, well, semi-naturally occurring resistant genes in the biofilms. But if we added the wastewater would be lost a lot faster. And that could mainly be explained that competition works a lot faster if metabolic rates are increased. So it seems like the temperature optima benefit is not as big as we think. It's rather that activity in the appropriate niche space of something that is adapted is the main driver of it. So we were really surprised to see that temperature didn't have so much of an effect.
Thank you very much. And I think now we can move on to the next keynote, led by Christos Zafeiridis. I hope I pronounced it right. Second and national expert from the Directorate General for Health and Food Safety and the Directorate for Crisis Management in Food, Animals and Plants, the Directorate for Official Controls, an EU representative of Northern Ireland, liaison group of UK, and official veterinarian of the Ministry of Rural Development and Food, Asclepius, One Health. He will talk to us about the prudent use of veterinary drugs and antibiotics in productive animals and the risk of development of antimicrobial resistance.
Thank you for the floor, Chair. Good morning, everybody. Good morning from Belfast, Northern Ireland. Currently, I'm on a mission with the European Commission here in Northern Ireland for the implementation of the Windsor Framework for the last 4 years. Today, I'm participating to this Congress with my capacity as a scientific advisor of the Mediterranean One Health Center and the Asclepius One Health platform. Okay, my involvement with the AMR topic is that I was, and I'm still, the coordinator of the pilot project of the Ministry of Rural Development and Food project, National AMR Surveillance Network in Animals. So my presentation today, no, excuse me. My presentation today is relevant to the prudent use of veterinary drugs and antibiotics in productive, otherwise farm animals, the risk of development of antimicrobial resistance. Well, firstly, I would like to start with what is a veterinary medicinal product? Any substance or combination of substances that appears to have therapeutic or prophylactic properties against animal diseases, or any substance or combination of substances that can be used in animals with the purpose of restoring, correcting, or modifying their physiological functions by exerting a pharmacological, immunological, or metabolic action. Let's focus on what is antimicrobial resistance. Actually, it's a natural biological phenomenon, the resistance developed by pathogenic microorganisms to antimicrobial medicines. This is the term. Let's have an approach how the veterinary medicines and AMR are involved. Pathogenic microorganisms, bacteria, viruses, fungi, parasites, and protozoa have a remarkable ability to adapt, especially to acquire and transmit the antimicrobial resistance. Excessive and uncontrolled use of antimicrobial substances promotes the multiplication of resistant organisms and therefore endangers the achievements of previous decades in the field of health. Finally, antimicrobial resistance caused by natural factors existed even before antimicrobial substances were introduced into medical treatments. However, it is generally accepted that there is a link between the amount of the antimicrobials in use and the increase in resistant organisms. Please. Okay. Oh, sorry. Excuse me. I don't know what happened. Can you see my screen?
I mean, we do, but there's some rectangular shapes.
I have to recenter again. I apologize for that. Since the discovery of penicillin in 1940, antimicrobial medicines have become essential for treating many microbial infections in humans and in animals as well. Almost 80 years later, their usefulness is seriously threatened due to emergent spread of microbes resistant to affordable and effective first choice or otherwise first line medicines, making these drugs ineffective for treating infections. Increasing global exchanges and travel facilitate the spread of antimicrobial resistance among countries and continents. Therefore, antimicrobial resistance is a source of global public health concern. Here is a diagram of how AMR it can be considered as a European or a global problem, and the sectors that are involved is the medicine, the human medicine, the veterinary medicine, the livestock farming, the agriculture, the environment, the trade, and I could add one more, that it's tourism. At the EU level, as my colleagues mentioned before, the problem of antimicrobial resistance has been recognized and examined for many years. The EU places emphasis on the relationship between the use of antimicrobial substances in veterinary medicine and the development of antimicrobial resistance. So, the increasing risk of antimicrobial resistance, it's starting with unindicated use of antimicrobial substances, the failure to follow hygiene rules, the use of medicines for non-therapeutic purposes, the environmental pollution, with the final result acceleration of the emergence and spread of resistant microorganisms. Serious consequences, a subset of drug-resistant bacteria is responsible for almost 30,000 human deaths annually. Common bacteria that cause, for example, diarrhea or respiratory infections in various animal species have become more resistant to commonly used veterinary medicines, causing increasing suffering and mortality in animals. This, in turn, leads to production losses, additional expenses, and increased occupational risk for farmers. Here is another diagram that we can see the development of resistant strains, Salmonella, Campylobacter, Escherichia coli, and other ones. The ineffective medicines can cause ineffective disease management, and the risk to animal life reduce production and economic loss. The final result is that we have a risk to public health. Additionally, the development of antimicrobial resistance, the pressure, I can't see the whole presentation of mine. I apologize. I don't know what is happening, but I can see only a part of my slide. I don't know if you can help me on that. Excuse me?
Yes. Could you perhaps just stop sharing and attempt to reshare, if possible?
Yes, thank you. I will do that again, and I apologize for that.
No. Same problem.
Okay, now I think it's better. Okay.
Yes.
I was talking about the increasing risk of antimicrobial resistance. Additionally, the development of antimicrobial resistance, the pressure to reduce the use of antimicrobial medicines, the weak market incentives, and the increasing difficulties and costs of developing new effective antibiotics. All that has as a result, the discouragement of investments in the pharmaceutical sector, and the final result is that today only a few new antibiotics are under development. Significant initiatives have been undertaken in the fields of veterinary medicine and livestock farming, the ban on the use of antimicrobial substances for promoting the animal growth, the control of salmonella at all appropriate stages of production, processing, and distribution to reduce the human exposure to potentially resistant salmonella, monitoring of zoonotic antimicrobial resistance transmissible between animal and humans, and the use of antimicrobial substances in animals, requirements for veterinary medicines and other products such as food enzymes, probiotics, and disinfectants with potential impacts on the development of antimicrobial resistance. Despite the actions taken so far, it has not been possible to limit the risk of antimicrobial resistant development. A substantial strengthening of the established measures is required, along with a new set of strict measures. The basis for EU policy design has been scientific opinions on antimicrobial resistance provided by the EU assessment bodies such as the European Centre for Disease Prevention and Control, known as ECDC, the European Food Safety Authority, EFSA, the European Medicine Authority, EMA, the Scientific Committee on Emerging and New Identified Health Risk. Similarly, some actions have been undertaken at the international level by the World Health Organization, the Codex Alimentarius, and some others. So, this is another diagram that here depicts the increasing risk of antimicrobial resistance is the risk management that leads to the holistic approach to the problem, the food and the direct contact with animals that leads to the transfer of antimicrobial resistance from animals to humans. And we arrive that the One Health concept is totally involved with the human medicine and the veterinary medicine plus the environment. New actions have been adopted with the aim to mitigate the risk of antimicrobial resistant development through the proper use of antimicrobial substances in both humans and animals, ensuring the appropriate use across the EU and promoting microbiological diagnosis to determine, as far as possible, the need to use antimicrobial substances. Establish effective measures for the prevention of microbial infections and their spread of, the development of effective antimicrobial substances and alternative medicines for the treatment of human and animal infections, the development of international collaborations to limit the risk of antimicrobial resistance spread through international trade, travel, and the environment, and finally, the strengthened research to develop the scientific basis and innovate tools to combat antimicrobial resistance. Three main actions have been adopted to limit the development of antimicrobial resistance in veterinary medicine. First of all, the prudent use of antimicrobial medicines in veterinary medicine. The second is the strengthening to the promotion of appropriate use of antimicrobial substances in animals, information, education, and training of farmers, veterinarians, and all those involved in the veterinary livestock sector. This is what we did with the farmers in Greece during the implementation of this pilot project that I firstly mentioned. And third, It's the improvement of prevention and control of infections in animals, the establishment of a new regulation on animal health law, and the promotion of proper agricultural practices. What is all about the prudent use of antimicrobial substances in veterinary medicines? That the use in animals only when necessary and after prescription. Use the appropriate antibiotic following a reliable diagnosis and whenever possible after a sensitivity test. Use at the correct therapeutic level, for example, the recommended dosage and for a specific period. Avoid the insufficient dosing. This is a very crucial point. Limit use only to sick animals and not for prevention in all farm animals. And to reduce the overall use of antibiotics, especially those of critical importance, for example, third and fourth generation cephalosporins and fluoroquinolones. There is a need, the conclusion that there is a need to limit the prophylactic use of antimicrobials to cases with clearly defined clinical needs and to restrict the prescription and use of antimicrobial substances for group treatment of animals to situations in which the veterinarian has assessed that there is a clear clinical and, where applicable, epidemiological justification for treatment of all animals. There is a growing concern about the use of antimicrobials in veterinary medicine that are critically important for humans. Third and fourth generation cephalosporins are classified by the World Health Organization as critically important antibiotics for humans. EFSA considered that an effective control measure would be stop or strictly limit their use, allowing it only under specific circumstances. Methicillin-resistant Staphylococcus aureus, MRSA, it's more known, is a major cause of resistant hospital infections. Pigs, on the other hand, are a significant reservoir of a newly emerging MRSA type. A joint report by ECDC, EFSA, and EMA concludes that extensive use of antimicrobials for disease prevention is a key risk factor for MRSA spread. This is another point that we are focused on during our pilot project, this combination and the comparison of the data of AMR that we took from the hospitals and from the farms regarding the methicillin-resistant Staphylococcus aureus in Greece. Prevention and control of infection in farm animals, measures for animal health and welfare, biosecurity, and the promotion of good agricultural practices prevent infection and thus help reduce the use of antibiotics in animals, including in aquaculture, consequently limiting the likelihood of AMR development in animal pathogens and zoonotic agents. A new animal health legislation, known as animal health law, has been established focusing on disease prevention, reducing antibiotic use, and replacing existing disease control-based provisions. We all know that prevent is better to treat, is better than treat. The prevention and control of infection in farm animals, in 2013, the EU issued an implementing decision for monitoring antimicrobial resistance for Salmonella, Campylobacter, Escherichia coli, and Enterococcus in poultry, pigs, and cattle. I was the responsible person for all that years, from 2017 until 2021 for that procedure, and the Salmonella in pigs is the second most important source of human salmonellosis. So, the comparison between prevention and implementation of good agricultural practices could be based on the compliance with animal health rules, vaccinations, cell vaccines, implementation of surveillance and disease control programs, compliance with animal welfare rules in farming facilities, animal density, environmental conditions, etc., and the compliance with hygiene rules in farming, the removal of bedding, the disinfection, the waste management, and some other topics. I talked before about the promotion of good agricultural practices. Special care is needed when raising young animals from different sources as they are exposed to higher disease risk when grouped together. Avoid excessive animal density as large herd sizes can hinder individual or small group treatment and promote prophylactic antimicrobial use. And finally, breeding disease-resistant varieties can reduce the need for veterinary drugs but should not replace proper farm management. So, the livestock and aquaculture sectors should focus on disease prevention through proper hygiene, animal health and welfare, and strict biosecurity measures rather than on prophylactic antibiotic use. This is our conclusion. Here is a diagram that it is all connected. The implementation of good agricultural practices, the reduced occurrence of diseases in animals, the compliance with animal health and welfare rules, the reduced use of the veterinary medicines, and the common scope is the limitation of AMR development. Nevertheless, animals can still become ill, and appropriate treatments and effective veterinary medicines must be available. This leads to that there is a need for education and awareness raising of all stakeholders, veterinarians, farmers, manufacturers, and distributors of medicines. And then with the goal of changing behavior regarding the use of veterinary medicines. Our priority must be the prudent use of veterinary medicines. In Greece, we started two educational efforts. First, to train the public veterinarian, the public servants veterinarians, to train themselves on One Health and AMR topic, and then to train all the farmers in another step and then farm veterinarians on the AMR initiative. Here, the promotion of prudent use of antimicrobial substances can lead to that it depends on changes in attitude, practice, and education, and finally between farmers, veterinarians, and other professionals involved in the distribution and use of antimicrobial medicines. These are all our conclusion after the completion of our pilot project. I was very fast, and thank you for your attention.
Thank you very much. Are there any questions in the audience or online? No. Maybe I have one question for you. After these measures to actually achieve this responsible use of veterinary drugs or antibiotics in this case, some of these measures actually require big investments from the farmer side. What would be your suggestion to actually ensure that this can be implemented?
On the farmers level.
Yeah, exactly. Like, for example, to invest on the infrastructures to make sure there's more space between sick animals and not sick, etc.
Yes, well, thank you for the question. This is our intention to train themselves on how to deal with all that stuff. And we had too many sessions for the farmers and the farm veterinarians, the private veterinarians that they are responsible for the farms, just to be informed and involved in the whole project that it's called our National Action Plan Against Antimicrobial Resistance in the context of One Health. It's a very long story. It started on 2021 and still goes on with between the Ministry of Rural Development, Food, and the Mediterranean One Health Center.
Great, thanks. Yeah, long work, but very necessary. So thank you very much for presenting it. And if there's no more questions, we would leave it here and we can start our coffee break a bit earlier. Thank you very much again for all the speakers and for everyone that joined us. And I believe we can have the coffee break until 11:30, is that right? So, yeah.
Yes, we can move on with the coffee break. Thank you very much.
Okay, good morning, everyone. It is my pleasure to chair this session, which is about antimicrobial resistance. And I would like to welcome our first speaker from Denmark. It's Professor Barth Smets. He's professor at the Department of Biological and Chemical Engineering in Aarhus. And his talk is on the effect of sub-inhibitory biocide exposure on conjugal transfer of antibiotic-resistant plasmids. So thank you, Professor Smets.
Okay, I presume everybody can hear me.
Yes, we can hear you well.
Good, and you see my slides as well. So I would like to talk about this topic. First, I would like to thank the organizers for giving me the opportunity to speak, because I think I have a story or a message that is, I think, quite relevant. And in my presentation, there will be some good news and there will be some bad news. So stay tuned. So I'm at Aarhus University. This work was conducted by a recent graduated PhD student, Jiming, when I was still at the Technical University of Denmark. And I also acknowledge my other co-authors. And we are, at least my group has been interested in trying to understand what happens when these clinical isolates carrying clinical antimicrobial resistance genes, what happens when they leave the clinical setting, when they are flushed down the toilet, go through the sewers, enter a wastewater treatment plant, and or even enter the environment. What conditions are there? What processes are there that still make these genes to be retained either in the isolate itself that survives or in other microbes into which these genes may be transferred. And within that context, we have been looking at what are assumed to be selective conditions for some of these antimicrobial resistance genes, and that is biocides. And biocides we use extensively. Biocides are not don't get the same attention as antimicrobial resistance genes, and a biocide is essentially anything, substance, mostly chemical substances that are intended to destroy, deter, render harmless, or exert a controlling effect on any harmful organism. So they don't have the specificity that antimicrobial resistance genes have, but their purpose is indeed to kill. And so where they are used, they're used everywhere. They're used in industry. They're used in residential settings. They're used in hospitals. They're used everywhere. And at the points where they are being applied, they are effective because they are used at a certain concentration where they kill. Now, what happens when what happens with these streams of biocides? Ultimately, they again enter an aqueous environment and they are being diluted. So we actually put all of these biocides at lower concentrations into the environment in our sewers first. And there, of course, their concentration will be much lower. They certainly will be sub-inhibitory. And the question is, do these biocides have any effect on AMR dissemination? With our focus on conjugation. And so this is really what we are trying to understand, what we were trying to understand in this project, how biocides interact with plasmids and bacteria to affect the fate and transmission of AMR. This was part of a larger project under the JPI Oceans and JPI Water, I presume, which was led by Joachim Larsen, known to most of you, which was called Biocide. And our task was to look at the potential effect on horizontal gene transfer. And so we know that many clinically relevant antimicrobial resistances are oftentimes located on mobile genetic elements, plasmids. Several of those plasmids are conjugative. And of course, plasmids have a certain structure, and important to recognize is that plasmids can be divided into different families, as I will call them, or incompatibility groups, and that depends on whether they share genes related to essentially their transfer, which are the mating pair formation genes or the MOB genes. Based on the similarity between these two, plasmids are put into incompatibility groups. There's other genes related to their establishments, genes related to their maintenance and their replication, and then there's genes, of course, on those plasmids that we are most concerned about, the antimicrobial resistance genes. When plasmids are conjugal, they code for a pilus, and it's through this pilus interacting with the recipient cell that they can bring the recipient closer and transfer essentially one copy or one strand of the conjugative plasmids to another cell. And this cell may be of a very different phylogeny than itself, and in this way results in the survival of the plasmid or the antimicrobial resistance gene. So again, we wanted to see there had been some strong suggestions that biocides had a stimulating effect on horizontal gene transfer mediated by these conjugal plasmids. And here I just list the number of papers, random papers, not random papers. They all say something about promotion, subinhibitory concentration of disinfectants, subinhibitory concentration of heavy metals, pharmaceuticals, even sweeteners, acetaminophen, blah, blah, blah. Many publications have come out that that indicate that have observed an increase of what is called their plasmid transfer. And we were surprised to read that. We decided to reread all these publications and look at this with a critical eye before we started our own experimentation. And that information has been synthesized in a short manuscript in ESNT last year. And here I show one of the figures out of this manuscript where we show for different publications what fold change of transconjugant over recipients has been observed compared to the non-exposed control. So one way that plasmid transfer is expressed is by looking at what is called the transfer efficiencies. There's many different terms that have been used. And in this case, it's the concentration of transconjugant cells observed at the end of the experiment normalized to the cells of recipients observed either at the end or at the beginning of the experiment. A bigger number would suggest there's more transconjugant, so that probably suggests that the transfer has happened to a larger extent. That means to more cells or with a higher frequency. And what you can see here, there's a number of biocides here, and there are some. This is one, so meaning there is no stimulation. Many of these lines, at least for a while, are above one. So that's what you see. So yes, indeed, for a number of compounds, what they claim in their title is correct. What you also see is that the stimulation is not that large. This is not a log scale. This is a linear scale. So at the maximum, let us say 8-fold. Usually it's a 2 to 3 to 4-fold. So it's really kind of at the edge of experimental error, I would say. The other thing we observed is that in these papers and other papers, that if stimulation is observed, it only is observed, of course, not at the inhibitory concentration, but from about 1/10 of the inhibitory concentration down to 1/1000th of the inhibitory concentration. Now, in all these previous works, there has been a lot of emphasis on a certain model system, and it's predominantly using RP4, the RP4 plasmid, and oftentimes using E. coli. So that is one limitation of those studies. One of the things we wanted to see is if this is generalizable across plasmids and strains. And also, we believe that the way that transconjugation or conjugal transfer was measured might have some issues. So we think there is some bias in terms of the quantification. I'll come back to that. So what we wanted to do is look at this in a more integral way, looking at several plasmids, a range of biocides, and not only looking at E. coli, but also looking at other genera in our experiments. So that's what we wanted to do. And so the first thing we spent quite a bit of time on is to find a methodology to quantify transconjugants. And we need to recognize that even though conjugation happens, conjugation is still a rare event when you look at how many donors and recipients there are. So you need to be able to quantify few cells in the background of many other cells. And ultimately, what we ended up with is a methodology. So we also looked at that carefully. That sometimes you have a problem when you do a spread plating on selective media, that you essentially have background conjugation happening on the enumeration medium, which if you don't measure at time zero, you have an issue. So we ended up with a methodology that gave us little noise, but also indeed low signal, which involves enumerating essentially the transconjugates through an MPN method in selective medium. So this is the methodology. We grow our donors and recipients, harvest the cells, wash, dilute to a certain concentration, then do the mating in a minimal medium, but we put some carbon source in there, incubate for a fixed time, and then enumerate on selective media for the transconjugates based on antibiotics on the plasmid and the recipient strain, and then enumerate the positive wells and then estimate the transfer frequencies. And we think it is more appropriate to estimate the transfer frequencies normalized to the product of the donors and the recipients. So a few example graphs. How does this look like? So here I'm looking at matings between an E. coli and an E. coli strain, or a Putera and a Putera strain, E. coli and E. coli, E. coli and E. coli, with either the RP4 plasmid or the R27 or the R1 plasmid. And to put your emphasis here, so after we do these matings in sufficient replication, we enumerate the efficiencies, the transfer efficiencies in the control. That's mean in the absence of a biocide. And this would be in the presence of copper. My apologies in the copper as a biocide. This is for the RP4 situation. So we always have pairs of observation in the control and in the presence of the biocide, the control presence of the biocide, control presence of the biocide. What you can already see from this is that when you compare the control to the biocide, there's only one situation where in the presence of the biocide, the efficiency is larger. So this information we then translate into a mean and a standard deviation or a confidence interval, and then we calculate the ratios, all with the same results. And you can see in this situation, for copper, we only see a value above 1 for the RP4 E. coli case, not for the other cases. So with that in hand, and having convinced us that we have a good methodology in place with where we can actually quantify rare events with limited noise and with less bias, we decided to look at a suite of biocides. Now, this was done as part of a larger project, so we measured MICs of all of these biocides where they were not reported in the literature. That was then done at Gothenburg University for both E. coli and Putida. So we have silver, zinc, copper, triclosan, chlorhexidine, DDAC, benzoic acid, silicic acid, benzotriazoles, ambit, methylparaben, 4-nitrophenol, and then these disinfectants, chloramine B, chloramine T, and peracetic acid. This is just some information about the target organism and what kind of MIC was reported for E. coli. And sometimes, as I said, we measured it ourselves, and the MICs for P. putida. Now, we did still a lot of work with E. coli and a less amount of work with P. putida. So here are some of the results. So here I show you then across copper, silver, zinc, chlorhexidine, DDAC, and triclosan, their effect at sub-inhibitory concentration on the transfer of RP4 from P. putida to P. putida, from E. coli to E. coli, on R27 transfer from E. coli to E. coli, on R1 transfer from E. coli to E. coli. Why do we have these different plasmids? Because they belong to different incompatibility groups. INC-P and most clinical plasmids do not belong to INC-P. They tend to belong to INC-H or INC-F. Nevertheless, we include INC-P. They're environmentally relevant. And then INC-H and INC-F. What you should see here, and again, what I'm showing you here is the ratio of the transfer efficiency observed in the situation with exposure to sub-inhibitory concentration normalized to the transfer efficiency seen in the control experiment. And what you see, there's actually only a few places where it looks like this number is positive. And that is for copper sulfate there, silver nitrate there, and for DDAC there. And only for one of them, it's significant. And that is for the copper situation. And you can see, so only really for copper, and that is for the situation of RP4 between E. coli's, do we see stimulation of transfer. For none of the other situations, whether it's with E. coli or Putera, whether it's in P, Engage, or in KEF plasmids, do we see stimulation. We also done those experiments at 100th of the MIC, and at 100th of the MIC, we never see stimulation. So that's a bit of a surprise compared to what's in the literature. So we saw only stimulation, as I pointed out, with RP4 between E. coli strains. So we said, okay, so let us examine this strain with this plasmid to a larger extent. So we looked at a number of additional biocides, and I won't read them for you, and looked at their effect on the stimulation. And again, we see no stimulatory effect. Coming back to copper, then we said, okay, let us then go stay with E. coli and see if at least this phenomenon is consistent. We had already seen that it works within P, but not in H and F. We wanted to see if is it at least consistent within a group, a family of plasmids. So we additionally looked at PB10, which is another P plasmid, and indeed you see both for PB10 and RP4, we see stimulation in the presence of copper. But for none of the others, for the engaged plasmids, they are consistently not stimulated. When we look at another biocide, the DDAC, it remains consistent. We don't ever see any stimulatory effects. Well, then we said, actually I should go to the next slide. Is at least what we observe consistent what has been reported in the literature? So as I briefly pointed out, in the literature, stimulatory observations have been noted of biocides on gene transfer, and they predominantly have dealt with RP4-like systems. And those studies that have looked a little bit in detail have indicated this ROS-REC-A, or this reactive oxygen species REC-A SOS system, as being responsible, meaning that if you have these biocides seem to result in the accumulation of reactive oxygen species. This reactive oxygen species, when they are around, they will convert the REC-A protein into an active REC-A protein, which then triggers the autodigestion of some repressors, which result into the transcription of some genes, and some of those genes are specifically involved in gene transfer. So we then looked at E. coli versus a mutant that no longer has REC-A. And indeed, what we observe is when we work with the REC-A mutant, we no longer see that stimulation. And that is looking at these comparisons here. So that at least suggests, yes, indeed, it is consistent. And how does this even work? And I'll show this very briefly, is this REC-A, as I pointed out here, should, if this pathway is responsible, then this REC-A recognizes some repressors, and there are some motifs in those repressors, this alanine, glycine, alanine, glycine, alanine, glycine motifs that are involved. And so indeed, RP4, the transfer of RP4 and NP-like plasmids is regulated by a number of repressors, and indeed those repressors have this motif. So it is speculated that indeed the way this works is an activated REC-A actually binds to these repressors, cleaves them, and allows expression of all of the TRA and the MOP genes. And now interesting, the R1 plasmid and NKEF plasmids, where we do not see the stimulation, it actually works differently. It works based on an activator element. And in this case, the activator carries this motif. So we speculate that if in this case, when you have reactive oxygen species presence, you will actually destroy the activator, and the transfer will go down. And that seems to be consistent with what we observe. And here I just show another experiment that where we have then gone a step further and evaluated, well, these reactive oxygen species, is that something that we observe? And again, we work with copper sulfate and another biocide DDAC, and only in the presence of copper, again, do we see an increase in reactive oxygen species compared to the control. So yes, where we see the stimulation of transfer, it is also there where we see the reactive oxygen species, which then likely can activate the REC-A protein and then essentially destroy the repressors of transfer. I want to come back to that. And so in conclusion, what I would like to say, and this is, well, I'm close to the good news, the effect of biocides on the conjugation, and whether you call it rate or efficiency, it is important, but in the experiments we have done, we take it at time 0 and a time 24-hour estimation of transconjugants, so we can express it both as a rate as an efficiency. What I'm trying to point out is that it matters. It's a function of what biocide you use, what plasmid you use, and what bacterial species you have. And in those systems where the transfer seems to be controlled by repressors, you might have a stimulation indeed. But when the TRAS systems are controlled by promoters, you have a negative effect. And at least in our observations, biocides frequently inhibit conjugation and rarely stimulate conjugation. And that is the good news. And to follow up on that, when we see conjugation, it indeed appears that this ROS-SOS cascade and this REC-A requirement is indeed what's at work. And this is something that we are continuing to work on. So this idea that ROS that creates DNA damage will give you an active REC-A, and the REC-A drives an autolysis of repressors in the case of plasmids of the INC-P group. And so the bad news, I think, is that we should avoid what we hear a lot about: stressors stimulate horizontal gene transfer or stimulate retention of antimicrobial resistance genes. I think the bad news is that we really have to avoid generalizing from specific observations on stress regarding antimicrobial resistance transmission. So we have to qualify that, and we have to figure out how we can essentially do this qualification and still say something about biocides as a whole. And we may need to categorize them in terms of the biocides and the plasmid families, whether things are stimulated or inhibited. So indeed, some biocides promote horizontal transfer of some plasmids in some strains. So I would like to close by acknowledging some collaborators: Joachim Larsen at the University of Gothenburg, Frank Schreiber, with whom we have also intensely interacted, and the group of Yongwan Zhu at the Chinese Academy of Sciences, with whom we continue to interact. And I would like to close with that, just telling you, if you are in the water business, and especially in the innovation in the water business, that we at Aarhus University have just landed this large EIT water, which you will hear more about in the future. And thank you for your attention. I'll take your questions.
Thank you very much, Professor Smets, for this very interesting talk. Questions? Yes.
Uli Klumper from TU Dresden. Hey, Bart.
Hi.
I enjoyed the talk. So, I mean, you said we should avoid generalizing. At the same time, we need to generalize, right?
Indeed.
So what I'm wondering is the effect, if we think about concentration dependencies, rather based on absolute concentrations, which would allow regulating against absolute concentrations? Or is it based really on the concentration divided by MIC ratio, so the amount of inhibition that the bacteria experience?
Well, I agree with you. In the end, we're doing all of this so we can say something about the whole phenomenon of biocides regarding all relevant antimicrobial resistance genes. So we need to get to a point of generalizing. So we just need to break this generalization down into a few elements now that we see this. I think you're correct. It's probably related to the ratio. And that makes it a little bit challenging, because this might not be exactly the same for every type of microbe, even though I would imagine some consistency between gram negatives and as a whole and gram positives as a whole. But I think you are correct. It will not be the absolute concentration. It'll be essentially it's based on the toxicity that metal exerts and the mechanism by which it inhibits microbial activity. Yes, definitely. It's going to be normalized.
Any more questions? If I may, I have a question. I'm not quite in the field, but and I might sound naive. Have you looked in any changes or modifications in the cell wall of the strains that you looked at as a mechanism, as a possible mechanism that is related to what you see? I mean, besides the reactive oxygen species that you saw.
Others have indeed looked at membrane integrity as a measurement of indeed by using reactive dyes. And there is an indication indeed that you also have a loss of membrane integrity happening at the same time as when you have reactive oxygen species. So for some of the plasmid and some of the studies, they've also been able to note a membrane integrity effect. We have not specifically looked at it as far as I can recall. Yes, it's also a possibility.
Thank you. Are there any questions? It seems that we don't have any more questions, so let's thank again Professor Smets for his very interesting talk. Thank you very much for being here with us today. And I think we're ready to move on to our next speaker. Thank you.
Good morning. Could you hear me?
Yeah. So our next speaker is Dr. Kyriaki Trifinopoulos. He's assistant professor of clinical microbiology at the School of Medicine at the University of Crete. And she's going to talk about the challenge of antimicrobial resistance in clinical settings. So, Dr. Trifinopoulou.
Thank you very much. Good morning. And I would like to thank you, the organizing committee for the invitation. I am trying during the next 20-25 minutes to share with you the challenges that we are facing in hospitals in Europe, but more specifically in Greece with AMR and the MDROs. So, as we have already heard from previous speakers, antimicrobial resistance is one of the top global public health and developmental threats. And we know the main drivers for this situation is the misuse and overuse of antimicrobials, not only in human sector, but also in animals and plants. Unfortunately, AMR affects countries in all regions and at all income levels. However, we know that low and middle income countries are most affected. And also, in addition to death and disability, AMR has significant economic costs, and the costs are really huge. It's about $1 trillion US by 2050, additional healthcare costs. So, beginning from the global situation, and according to the Global Antimicrobial Resistance Surveillance System GLASS, you see here in this map the situation with resistance of blood stream isolates of E. coli to third-generation cephalosporins. That is indicative of the production of ESBLs. And if we would like to talk about the cumulative resistance rates for ceftriaxone, it was in 2023 over 40%. And you can see that all over the world we can observe a gradients from low to much higher resistance rate. If we are going now to Klebsiella pneumonia bloodstream isolates, the situation is rather different. We have much lower global cumulative resistance rates, about 15% in carbapenems like meropenem. However, we can see that there are several parts in the world with much higher resistance rate. And the situation is really even more difficult for Acinetobacter baumannii resistant to carbapenem. We talk also for bloodstream isolates, and you can see here that the resistance rates at global level is above 50% resistant to for imipenem. So all these resistance percentages have clinical consideration. And poses difficulties in everyday clinical practice. Firstly, for selection of an effective empirical therapy. And in settings like Greece and our Greek acute care hospitals, selecting effective empirical therapy is quite challenging. And I'll show you in a few minutes the situation. More importantly, we have resistance to carbapenems in both Klebsiella pneumonia and Acinetobacter, and all this emerging resistance to carbapenems, along with resistance to colistin, a reserve agent, complicate further the treatment decision in our settings. And all these are related with mortality. And we know that the drug-resistant infections are threats for all of us, but of course, we are not in the same situation all over the world. And unfortunately, as you can see here, several parts of the world, sub-Saharan Africa or South Asia, faces the most difficult situation regarding mortality, both associated but also attributable to AMR infections. And this is from last year, very nice presentation in Lancet about the global burden of bacterial antimicrobial resistance from 1990 to 2021, with a systematic analysis and also forecasts for 2050. And we can see here the situation and the death rates attributable to AMR in all ages in 1990 and also in 2021, but also a not optimistic forecast for 2050 if something will not change regarding AMR, when we can see these red and dark red. That means very high mortality rates will be our future. And if we would like to be in more details regarding Europe, you can see here that the forecasting for the Balkan Peninsula and more importantly Greece is really very worrisome. So in order to address all these things, WHO has uploaded the list for bacterial priority pathogens in order to guide research and development, investment, and also global policy toward tackling the most urgent antibiotic resistance threats. And as we all know now, this updated list has 15 pathogen drug combination with 3 categories of critical, of high, and of medium priority. And the carbapenem resistant Enterobacterales, as well as carbapenem resistant Acinetobacter baumannii, are in the critical group, while Pseudomonas aeruginosa carbapenem resistant, along with other pathogens that are very important pathogens in hospital settings like vancomycin resistant Enterococcus faecium or methicillin resistant Staphylococcus aureus, are still of high priority. And going from the global to our continent in Europe, regarding, according to the EARSnet for 2024, the antimicrobial resistance surveillance report in EU, the data shows that AMR levels remain high in EU in 2024. There is a well-known gradient on AMR. That means that the southern and central and eastern Europe has generally more and higher AMR rates. And also, this is very important that in each year, more than 35,000 people die in the EU as a direct consequence of antimicrobial resistant infections. So, regarding the carbapenem resistant Enterobacterales that is in the critical group, what we are facing in Europe since 2019 is a really worsening epidemiological situation, because we are facing an increase in the incidence of carbapenem-resistant Klebsiella pneumonia in our hospitals, and also we are facing a convergence of virulence and resistance in Klebsiella pneumonia. That means we have already some cases of hypervirulent Klebsiella pneumonia ST23 carrying carbapenem-based genes. And furthermore, we have newly Enterobacterales species carrying carbapenem-based genes. We have a lot of plasmid-mediated spread of carbapenem-based genes causing outbreaks in hospitals and across the healthcare networks. And finally, we are facing an increasing detection of isolates of E. coli of high-risk clones carrying carbapenem-based genes. And this is a real threat for the spread of carbapenem resistance also in the community. So based on this situation, the probability, the CDC in its rapid risk assessment considered the probability of further spread of CRE in the EU high, with a high impact because of the attributable mortality from these bloodstream infections are really high. So a high probability, a high impact, that means that all these two makes the risk for further spread of CRE in the Europe to be high, very high. And also in the third point prevalence survey of healthcare-associated infections and antimicrobial use in the European acute care hospitals that took place in 2022-2023, the data from a huge number of EU hospitals were indicative of a high prevalence of patients with at least one healthcare-associated infection at 6.3%. And here you can see the distribution of types of healthcare-associated infections. The respiratory tract infections are the first, both during the day of admission or after 2 days of admission, followed by urinary tract infections, surgical tract infections, and bloodstream infections. And regarding the pathogens of healthcare-associated infections, we see that among gram-negative bacilli, that the third-generation cephalosporin resistance in Enterobacterales was high as almost 35%, while the carbapenem resistance was reported as 9% in Enterobacterales, being the highest as expected in Klebsiella pneumonia, almost 25%. Also, carbapenem resistant quite high levels was reported for Pseudomonas aeruginosa, or almost 30%, and Acinetobacter baumannii 83%. With gram-positive cocci, methicillin resistant was reported in the 24% of Staphylococcus aureus, while vancomycin resistance was higher as expected in Physium, almost 29%. And here you see that there is the cumulative percentages that I told you before. However, the known gradient in resistance of E. coli in third-generation cephalosporin or carbapenem resistance in Klebsiella and Pseudomonas aeruginosa and Acinetobacter baumannii is the constant. That means that the South and Eastern Europe is facing the main problem of healthcare-associated infections with very high percentages of resistance to carbapenems. I don't know why. Sorry. Sorry for this, but I don't know. Okay, so the results of this point prevalence survey confirmed that all these both things, AMR and healthcare-associated infections, represent a significant public health challenge for EU. And the estimation is about 4.3 million patients acquire at least one healthcare-associated infection per year in EU. As for the situation in Greece, we can see here that the prevalence of healthcare-associated infections was almost a double. It was 20% in comparison with 6.8%, the EU median prevalence for high. And also, this is quite important and difficult for us, that the composite index of AMR, that means the percentage of antimicrobial resistant isolates, was as high as 68% in comparison with 22% of the EU median. In Greece, you can see here that during this point prevalence study, carbapenem-resistant Klebsiella pneumonia was found in 64% of the Klebsiella pneumonia isolates that were the main pathogen for a healthcare-associated infection, while in Pseudomonas and Acinetobacter baumannii, the resistance rates were higher, Pseudomonas 65% and Acinetobacter 95%. So you can see that the macrobiological spectrum of bacteremia in our Greek hospital setting includes to a large extent multi-drug or even pan-drug resistant pathogens. So the backbone of the empirical antimicrobial therapy for bloodstream infection should include broad spectrum last line antibiotics. So, and we have specific national guidelines for this. You can see here, this is from the last year, and they have been updated for 2025 from the Hellenic Society for Infections. So for carbapenem-resistant Enterobacterales, the empiric treatment can include even one of the newer beta-lactamase inhibitor combination. Of course, we try to be very prudent and very strict to whom will given this empiric treatment. And also there is guidelines for the use, the gradual increase in the use of some rapid diagnostics in order to detect the most common microorganisms and along with the most common genes related to antimicrobial resistance, like genes CTXM for ESBL production, several carbapenemase genes for carbapenemase production, and so on. And there are really very nice molecular point of care tests that we can use in positive blood culture and targeting 43 targets in 1 hour, and we can inform our clinicians about the pathogen, and also if there are some of the most important antimicrobial resistance genes. However, in order for all this integration of all these sophisticated and rapid molecular syndromic diagnostic tests, it's very important to have a close collaboration, the clinical microbiology labs and our clinicians and infectious disease practitioners, in order for to secure that the appropriate test will be used for the right patient, the right time, in order to define the appropriate therapy. And that means that it should be an active diagnostic and antimicrobial stewardship program in every hospital. However, because all of these new technologies are quite expensive, we cannot afford to use rapid diagnostics, molecular diagnostics in every positive blood culture, and we are facing antibiograms like this one with the Klebsiella being resistant or being susceptible only in the newer beta-lactam or beta-lactamase inhibitor, or we are facing situations and antibiograms like this one where you can see that we have a Klebsiella pneumonia from bloodstream infections without any in vitro susceptibility. We use in everyday clinical practice, immunochromatography. That is it's much cheaper, it's quite affordable for everyone to use. And you can see, for example, in the first case, we have a KPC, Klebsiella pneumonia, while in the second one, we have an NDM, a metalloenzyme producing Klebsiella pneumonia. And this is also a very common profile of Pseudomonas aeruginosa in the Greek hospitals. You can see Pseudomonas aeruginosa carbapenem resistant with susceptibility only to colistin. So regarding the definite treatment options, it is very important to know the lab and also to inform clinicians about the type of carbapenemase enzyme produced, because you can see that, for example, in metalloenzyme, the NDM producers, the options are very limited. It's only cefiderocol and aztreonam-avibactam. And you can imagine the challenge that our clinicians face. Still, both drugs are not yet available in all settings. And also, you can see here, Acinetobacter baumannii, this is a really difficult pathogens to deal with. As you can see that the therapeutic options are very limited, and the guidelines are for combination drugs with colistin or tigecycline or abicillin sulbactam. There is a new combination, sulbactam durlobactam. However, this is not yet available in Europe. In order to help and in order to focus and study the circulating carbapenem resistant Enterobacterales, an initiative from ECDC has begun back to 2012 with the USCAPE study, and then ECDC has organized other two studies in the European level in order to perform genomic surveillance to study the spread of carbapenem-resistant clones in European countries. So these studies have shown that the spread of high-risk lineages of Klebsiella pneumonia producing carbapenemase gene within and between acute care hospitals was the main driver of all this increase of carbapenem resistance in Europe. And there are specific high-risk lineages in Klebsiella pneumonia, like ST11 or ST15 or ST258. And also with whole genome sequencing, which is the typic method with the ultimate discriminatory power, it was very easy to find out that there are frequent transmission events within hospitals. So regarding the situation in Greece, the carbapenem-resistant Klebsiella pneumonia, we know that they belong to 5 high-risk clones, with ST258 being the dominant for many years, followed by ST11 and ST39. However, we now are in a position to know the distribution and the time trend of carbapenem-resistant clones in our hospitals, and we can observe this gradual decrease in ST258, and from the other hand, a gradual increase in ST11. And this is very important because ST11 was found to be related for the most transmission events within hospitals. And what we believe is that ST11 has since it is very closely related with NDM, has an advantage in our hospitals because in our hospitals, due to KPC, there is a lot use. There is an increased use of ceftazidime bactam, and the selection pressure favor NDM producing clones. So this is what we believe for the gradual increase and dominance of ST11 in our hospitals. The good news is that our multi-drug resistant Klebsiella pneumonia isolates are not they are not so virulent ones. That means that we have not seen until now hypervirulent clones like ST23 harboring carbapenemase genes. And also regarding Pseudomonas aeruginosa, we know that we are facing a shift also in the molecular epidemiology from VIM producers to NDM producers. And what we also have seen after the first detection and after the first introduction in the country of specific high-risk clones like ST308 or 773, we are facing a rapid reversal of carbapenemase producing Pseudomonas aeruginosa that produce nowadays NDM and not VIM in both northern Greece and also in Athens. Finally, I will show you two very nice studies on attributable mortality to bloodstream infections caused by different carbapenem-resistant bacilli. The first one is from a nationwide study in Italy, which faces quite same difficult situation with carbapenem-resistant gram-negative bacilli in their hospital settings. And as we can see here from this study, compared to carbapenem susceptible gram-negative bacteremia, the MBL producing Enterobacterales, as well as carbapenem-resistant Pseudomonas aeruginosa and Acinetobacter baumannii, were significantly associated with 30-day mortality. Moreover, the attributable mortality rates were much higher for MBL, for the metallo-beta-lactamase producers, Enterobacterales, and also that, and you can see here that it was far more, much higher the mortality, the excess mortality for MBL, 35%, instead of 5%, for example, for KPC producing Enterobacterales. From our settings and our team, this is a mortality due to carbapenem-resistant bacteremia study, a 5-year cohort study in ICU patients. And you can see that both in the in-hospital death due to carbapenem-resistant Acinetobacter baumannii bacteremia was statistically associated with in-hospital death, also with prolongation of length of stay. So the framework and what we should do in our hospitals is definitely the one pillar is antibiotic stewardship in order to save save our antibiotics, because they are not we have no any other options. Antibiotic stewardship means to prescribe antibiotics when they are truly needed, to prescribe the appropriate antibiotic, to use the shorter duration needed for the specific infection, and also to de-escalate from the empiric therapy as soon as possible when we have the final result from AST, of course, when this is the case. It is very important from the clinical microbiology labs to support with the surveillance of AMR and healthcare-associated infections. And of course, of ultimate importance is the hand hygiene, the infection prevention and control measures, mainly with contact precautions, and also the environmental clinic in order to prevent and control further the healthcare-associated infections due to MDR pathogens in our settings. Because healthcare setting, acute care hospital, and the healthcare facilities in general are only one part of the puzzle on AMR. So we know that we are facing a global crisis. There is no time to wait, and the sustained One Health response is what is the priority. It is our need now. So with all this, I would like to thank you, and I am at your disposal for any questions. Thank you.
Thank you very much. We have time for a few questions. So, yes.
Roberto Spurio, University of Camerino, Italy. I would like to ask you a question about surveillance systems or monitoring system and tools, sort of a technical question. I know that throughout Europe, there are some hospitals that now are equipped with sophisticated next generation sequencing system that they use this high throughput sequencing, focusing on the resistome, of course, in parallel with standard microbiological techniques, those that you have shown on your presentation. So I would like to know which is the state of the art in Greece, if what is going on, because this would be the next step in terms of technological advancement and also support for all kinds of analysis and control of AMR.
Thank you very much for your question. Yes, we know there is a lot. There are many sophisticated new technologies nowadays. But I think that the situation in everyday clinical practice is that only university hospitals can afford and can use some of these first of all, rapid molecular syndromic tests for a fast recovery of both pathogens and genes. However, whole genome sequencing of important hospital pathogens is not yet used in everyday practice. For example, in our hospital, the University Hospital of Iraklio, we are using whole genome sequencing in order to study the molecular epidemiology and the circulating clones of difficult to treat Pseudomonas aeruginosa or carbapenem-resistant Klebsiella pneumonia. But all this, we are far away from use it in a way that we can help in the management of a specific patient, This is for us to know the epidemiology and the molecular epidemiology of the circulating clones in order to predict. It will be very nice to use all this data in models in order to predict from the phenotype or from some specific traits for each clone. But I think that we are in our baby steps yet.
Thank you. Any more questions? Okay, we don't have any more questions, so thank you very much again, Dr. Trifonopoulou. And now we're ready to move to our last speaker for this session. Dr. Asma Bashir is Assistant Professor at Zahid Zulfikar Al Bhutto Institute of Science and Technology in Pakistan, and he's going to talk about combating antimicrobial resistance through integrated One Health policies, bridging the gaps between human, animal, and environmental sectors.
We have a technical problem. Okay.
Hello, good afternoon.
Okay, great. Welcome.
Dr. Bashir, we're
Yes.
ready. We can hear you, and we're ready for your screen sharing.
Yeah, I'm trying to connect the screen. Just a minute. Yes, can you see?
Yes, yes.
Okay. Actually, there is a little problem with the internet over here today. Okay. Hello, good afternoon, everyone. I hope I'm clear to everyone now.
You can go ahead. Yeah, we're ready.
Okay. My presentation, I'm Dr. Asma Bashir, Assistant Professor in the Department of Biosciences, Faculty of Life Sciences, Zabist University. I'm a microbiologist by profession, and it's been more than 15 years I am working on antimicrobial resistance. So today I'm here to discuss a crisis that is often invisible, but acts as a foundational threat to modern medicine. My talk is on a One Health perspective of antimicrobial resistance, bridging gaps between human, animal, and environmental systems.
Screen sharing.
Yeah, I'm trying to connect the screen. Just a minute.
Yes, yes.
Is it connected? Okay. Actually, there is a little problem with the internet over here today. Okay. Hello, good afternoon, everyone.
You can go ahead. Yeah.
Okay.
We're ready.
Yeah. My presentation, I'm Dr. Asma Bashir, Assistant Professor in the Department of Biosciences.
Dear Professor Asma, you have been
Yes,
connected
sciences,
with...
Zabist University. I'm a microbiologist by profession, and it's been more than 15 years I am working on my.
Professor Asma, you have been connected with two different links. So we have a problem with the sound. Can you hear us? Can you hear us? You have been connected twice. That's why we have a problem with the sound.
Yeah, I'm trying to connect the screen. Just see.
Yes, yes.
Is it connected? Okay. Actually, there is a problem with the internet over here. Okay.
It seems that you have a very bad connection, and your voice is breaking up, and we can't see your presentation.
Maybe there is a problem with the Wi-Fi. Mrs. Asma.
Uh,
Dear Professor.
sorry, Professor.
Yes, can you?
Okay, can you hear me now, Professor Bashir? Can you hear me? Okay, just bear with us, just a few minutes to solve this because there was a bad connection, and I think Professor Bashir was connected to the YouTube channel, which has latency, and that's why we kept hearing different voices. So let's allow just 1 minute, if that's okay with everyone. So since I don't seem to see that Professor Bashir will join now, maybe we can use the time to see whether we have any questions or comments.
Unfortunately, we can do that. So we either proceed or how long do we have to wait? Because I need to go back to the lab, and I also have teaching. So,
I mean,
so is there a possibility that
we don't have to wait? It's just her Professor Bashir's presentation was scheduled from 12:30 to 1.
Okay, that's what I'm saying.
So
So
if we can allow 2 minutes or 3, or if we have a question.
No, we can wait and
yeah,
for the connection.
Yes, okay.
But
it's not the problem of the conference. It's the Wi-Fi, perhaps, of Professor Bashir. So we have to.
Hello?
Oh, let's
Hello, can you hear me?
wait for a minute. Okay. So we have an update. I don't know if we can connect now.
Hello, this is Dr. Asma Bashir here. Can you hear me now?
Yes, we can hear you.
Okay, let me. Shall I start my presentation?
Yes, you can
Sorry,
start your
actually,
presentation,
the internet was.
but due to time restraints, please can you keep it short? Yes, thank you very much.
Yeah, sure. I'll just take your 10 minutes.
Yes, thank you.
Okay. So good afternoon, everyone. My presentation is on a One Health perspective of antimicrobial resistance, bridging gaps between human, animal, and environmental sectors through integrated strategies. I am Dr. Asma Bashir from Karachi, Pakistan. So I'm here to discuss a crisis that is often invisible but act as a foundational threat to modern medicine. And my talk will bridge the gaps between the human, animal, and environmental sectors. I will move beyond the basic definition of resistance and then integrate strategies to mitigate what is now being called the silent threat. So, this silent pandemic, to understand the urgency, we must look at the hard data. This is not a future threat, it is a current reality. So in 2019 alone, bacterial antimicrobial resistance was associated with 4.95 million deaths globally. And to put that in perspective, that is a mortality burden higher than HIV and AIDS or malaria. So the future projection of this is continue with business as usual. Models project this toll will rise to 10 million annual deaths by 2050. And if you talk about the economic loss, the financial stakes are equally catastrophic. The cumulative cost to global economic output is projected to reach $100 trillion by 2050. And this data confirms that AMR is not just a health issue, it is a development and economic crisis. Our research framework is built upon the One Health triad. Historically, medicine has treated human patient in isolation. However, resistant mechanisms do not disrespect species barriers. If you talk about human health, focuses on responsible use of infection control. Animal health involves sustainable agriculture and veterinary oversights, and environment, the critical area of waste management and pollution. We argue that effective mitigation requires simultaneous action across all three domains. Ignoring one sector undermines efforts in others. So, let's deep dive into the first sector, that is human health. Here we face a massive stewardship gap, and the primary driver is misuse. Overprescription, we see a prevalence of antibiotics prescribed for viral infection like flu, where they are clinically useless. Infection control, hospitals have become reservoirs. Poor hygiene leads to healthcare associated infections involving highly resistant strains. And when we talk about diagnostic delays, a critical operational failure is the lack of rapid diagnostics. And without immediate test results, clinicians often prescribe broad spectrum drugs just in case, fueling resistance. Coming to the patient adherence, when patients fail to complete their course, we create a survival window to resistant bacteria to thrive. And coming to the animal sector, from farm to fork risk, moving to the animal sector, we encounter farm to fork risk. It is a stark reality that in many regions, more antibiotics are consumed by healthy animals than by humans. Growth promoters, antibiotics are frequently used at low doses for growth promotions rather than therapy. And this chronic exposure is the perfect recipe for selecting resistant strains. Zoonotic transmission, the resistant bacteria do not stay on the farm. They travel to humans through the food chain and direct contact. There is a policy gap, and there is an urgent need for strict veterinary oversight to ban the non-therapeutic use of these critical drugs. Then we move forward to the environmental reservoir. The third pillar is the environmental reservoir, which we identify as the forgotten component. The environment acts as a genetic bridge. Wastewater, farmer-run agriculture, conventional treatment plants are often not designed to filter out antibiotic residues or resistant genes, releasing them into the waterways. If we talk about the farmer runoffs in manufacturing herbs, discharge creates hotspots of extreme resistance.
We also have the problem of environment in agriculture sector. Manure containing excreted antibiotic runs off into soil and groundwater, fundamentally altering the environmental microbes. Integrated surveillance system. How do we monitor this complex threat? We propose integrated surveillance system, and currently surveillance is fragmented. We need to harmonize data streams, clinical data, veterinary data, and ecological data. When we talk about the clinical data, standardized reporting from hospitals to track human infection trends. Talking about the veterinary data, monitoring sales and resistant patterns in livestock and aquaculture. Ecological data, this is crucial. Why? Because we must sample water bodies and sewage to identify environmental hotspots before they spill over into clinical outbreaks. Global governance. Governance is the glue that holds these strategies together. As the WHO states, AMR poses a fundamental threat to human health, development, and security. We advocate for the implementation of the Global Action Plan, superheaded by the Tripartite Alliance, WHO, FAO, and WOH. This requires every nation to develop a national action plan that is legally anchored in One Health principles, ensuring that agriculture regulations match medical standards. If the mortality rate are not enough to drive policy change, the economic argument is undeniable. This chart shows the World Bank's projections for a high AMR scenario by 2050. Healthcare cost an additional $1.2 trillion in annual healthcare expenditures. Productivity loss a staggering $3.4 trillion lost annually due to labor supply reductions. And coming to poverty, this economic shock creates a poverty impact that could push 28 million people into extreme poverty, and the cost of prevention is a fraction of the cost of inaction. So there's a strategic policy interventions should be done based on a review. We recommend 4 specific strategy interventions. Number 1, regulatory framework. We must ban growth promoters in agriculture and enforce prescription only access to antibiotics for humans globally. Number 2, water, sanitation, and hygiene. Improving basic sanitations in healthcare facilities and farms is the most cost-effective way to reduce infection rates. The third one is the incentives which R&D should conduct. The antibiotic pipeline is dry. We need to push mechanisms like grants and pull mechanism like market entry rewards to make antibiotic development financially viable again. And the fourth and the most important one is the education and the awareness. We need public campaigns to shift the cultural expectations of antibiotics as a cure-all. And if we talk about innovation and the path forward, while we preserve existing drugs, science is racing to find alternatives. And the future lies beyond antibiotics. And the future can be the phage therapy, using bacteriophages, viruses to target specific bacteria without harming the host, rapid diagnostics, technologies that identify resistant profiles in minutes, enabling precise treatment immediately. And then the third one is the vaccines, the ultimate prevention tool. So the prevention is better than cure. Reducing the infection burden reduces the need for treatment entirely. And the last one is the microbiome modulation, using probiotics to strengthen the host natural defenses. And I myself has done my PhD in probiotics using bacteriocin as an alternative therapy to antibiotics. Coming to the conclusion of my presentation, addressing antimicrobial resistance requires a unified commitment. As we have demonstrated, no single sector can solve this crisis alone. We must dismantle the silos between global health, food security, and environmental protection. The One Health approach, that is bridging animal, human, and environmental, is not just a theoretical framework. It is the only viable strategy to secure our future against the silent pandemic. As I'm living in LMIC, that is low and middle income country, and Pakistan is among one of them. So low and middle income countries face unique challenges. Unregulated sales, we have antibiotics which are often available over the counter without a prescription. We have sanitation problems, lack of clean water and hygiene, accelerate the spread of infections, increasing the demand for antibiotics. Then a very important one is the cost issue. The older, the broader spectrum antibiotics are cheaper, but drive more resistant than targeted therapies. So again, the education and the awareness among the public awareness regarding the dangers of antibiotic is required. And this plays a key role And this is the main thing which we need to cater. Thank you for your attention, and now happy to take any questions.
Thank you very much. Are there any questions? So, Dr. Basir, I have a question. You talked about in your slide, which was entitled Beyond Antibiotics. So, do you see any efforts from the pharma industry to detect any new molecular targets and develop new inhibitors, or let's say new antibiotics beyond the known and targeting beyond the known classical molecular targets in order to?
Yes. There is basically a research going on around 2 decades back. Pharma industry has created a new domain known as pharma biotics, where they are basically looking for the alternatives. Like they are working on the probiotics, they are working on the proteins released by probiotics, they are working on bacteriophages, they are working on bacteriocins, they are working on antimicrobial peptides, but yet still a long way to go to cater this problem. And here, if I talk about Pakistan, actually the problem is that we take the drugs over the counter, we don't consult the doctor much, and there is an overuse of antibiotics over here. So hopefully pharma industry is working on an alternative, so let's see where we goes.
Thank you. Any more questions? Okay, thank you very much, Dr. Basir. It seems that we don't have any more questions, and that concludes our session. Thank you all very much for being here with us today. And now it's lunchtime.
Thank you very much. Now we have lunch until 2 o'clock.
So welcome back, everyone. I'm gonna share that session because we swap with Uli, who will share my session tomorrow. So 3 speakers will speak in a few minutes. So 2 will be in hybrid modes, and 1 will be here. And for the last one who will be here, it's not Andristos Nikolaos. I'm not sure about the right pronunciation. I tried my best. It's gonna be Antonia Matagarka, who will do the presentation on the same topics that in the program. So let's start with Dimos Tenis Choklakis, who is a researcher of the School of Medicine of the University of Crete, and gonna talk about the AMR in wastewater samples, some first results from the application of different methodologies into WWTPS in Crete. So whenever you are ready, you can start.
Sure. Thank you very much. Can you hear me well?
Yes.
Good.
Yes.
Okay, I will share the entire screen just to make sure that we don't have any drawbacks present during the speech. So perhaps, give me a sec. So I guess that now you can see the full screen. Is that correct?
Yeah, we can see everything.
Good, good. Okay, fine. So first, I would like to thank you for your kind invitation, and I would like to express my gratitude for the excellent level of all the speakers who had the opportunity to watch the presentation before. The level is quite high, and the results are more than interesting. So as you mentioned, we will be talking a little bit on AMR and some first results that we got here at the University of Crete and some preliminary experiments that we have carried out during the past 2 months. So before we begin, I'm pretty sure that you know all this stuff, but it's just to prepare the terrain before we enter into the results. So it's a global crisis. You know that. You don't want me to remind that to you. We work on the surveillance of AMR because more than 5 million deaths have been associated with resistance in antibiotics during the last few years. And the overuse of antimicrobials is not a concern of just humans, but it's of animals as well. Why do we bother to survey wastewater? Because it's not biased. I mean, if you go out the influent or the effluent wastewater of a hospital, you will get an image of what the case into a hospital, which is the image that you get, for example, when you got the records of your patients. On the other hand, if you try to survey wastewater of the community, then the big advantage, apart from the fact that you're not biased, is that we can capture data from the sick individuals and from asymptomatic carriers. That was the case with SARS-CoV-2, if some of you have worked with that. The big difference was that you get the whole image, not just a snapshot of the image. This is the tricky part because when we study AMR, we need to make sure of what we need to study for, what we need to look for. If we are interested in human health, then perhaps we can study the wastewater of the effluents or the influence of hospitals, of wastewater treatment plants of large cities. If we need to make sure that we do not leave animals behind in case where the waste from farming animals ends up into the pipelines of the community. We need to make sure that this does not happen, because if it does, the whole picture is completely different. And the other big question that we need to answer beforehand is that whether we are interested in studying what comes into the environment, because what comes in at the end of the day into the environment will return to us one way or another, either through the consumption of food or through irrigation purposes or through the reuse of treated water, etc., etc. Right, so the benefits have to do with early warning, as we said before. One of the crucial concerns is that we have the chance to detect new resistant genes before they become widespread in clinical settings. The pretty obvious part and pretty crucial part is that we can record the trending, which is crucial because there may be fluctuations throughout the year, or there may be an increasing slope if we are targeting to certain genes, and this slope may gather us towards the implementation of certain measures. And of course, in case we find something that concerns us, wastewater surveillance may give us an idea of whether intervention is successful or not. So, the surveillance of wastewater is not something that has come into place during the last few years. It has been into place during many, many years ago. We sort of worked more in a more depth due to the pandemic from SARS-CoV-2. And to be honest, we took the advantage of the knowledge that we gained from the pandemic to apply into the surveillance of AMR. This is the first part. And the second part, of course, is the European Union directive itself that maintains AMR monitoring in urban wastewater for every member state. So there is a need to do something, and this is the first part. And there is a need to do something that may be as most as common among the member states as possible. Of course, apart from the EU, there have been a number of initiatives worldwide who have been working on AMR resistance during the last few or many years, either under the umbrella of WHO or through the Transatlantic Task Force, which is a collaboration between EU and US, or even through the initiatives gathered from the United Nations. Right, so this is what we know. So what did we get? Before we begin, the initiative was to take advantage of the knowledge that we had gained from the SARS-CoV-2 pandemic. They gained that with the knowledge from the treatment of wastewater. And at the end of the day, the ultimate goal is, well, maybe optimistic, but we have some faith that we come up with a common SOP that we can disperse among the members of the national network of the country. So that was the whole idea. But there are several questions that need to be answered beforehand if we want to set up a protocol that will be well established. It can be applied to different wastewater treatment plants. It will answer questions and not raise more questions. Then we need to make sure that we have answered not every, but most of these questions. And these questions have to do, for example, with which sampling points are we interested in? And this has to do with what are we interested to survey for? Population, resistance in the environment, because as we said before, the treated wastewater that gets into the environment will return one way or another to humans. Are we interested in collecting samples from wastewater treatment plants only, from hospitals, or from both? And in case of wastewater treatment plants, are we interested on what's coming in, on what's coming out? And in this case, are we interested on samples that have been treated one way or another? Are we interested in samples that have been treated and they are just one step before they are dispersed into the environment or whatever? And the same counts for the hospital units. Are we interested on untreated samples or treated samples for the patients. Are we interested in sampling once per year, twice, monthly? Do we expect any fluctuations throughout the year? If yes, why? Would that be because of an outbreak? Would that be because of rainy conditions? Would that be because of a touristic season coming up? Are we interested in collecting samples instantly, or are we interested in collecting samples on 24-hour basis? These are crucial questions that need to be answered beforehand, or at least we need to play with all these tricky parts before we end up with a protocol. What about the target of the genes? At the end of the day, as we said before, the EU directive urges towards the surveillance of a marker, which may be at least E. coli, and some genes. This may be common among the member states or less common. In any case, we need to find out which genes we're interested in. We will do it based on patient results existing in the place we're serving, on the bibliography, which may collect data from other countries as well, but may not be applicable to our country. Do we need to apply metagenomic analysis? If we want metagenomic analysis, are we sure that we can translate the results that we'll get there from? Or we need bits and pieces from every one of these? As regards the extraction of the genetic material, are we interested in live cells? Are we interested in data coming from the whole genome sequencing? Are we interested in applying several pre-processing conditions before the extraction method? Are we interested in providing a combination of all the above? Again, we need to play around. As regards the EU directive, what is sure is that we need to survey, we need to record the presence of resistant genes on E. coli. But is that enough? Yes or no? Perhaps in some countries it is enough. In case of our country, where resistance is sky high, perhaps we need to introduce some other targets as well, such as Pseudomonas, Enterobacter, Klebsiella, and Ricocai. And perhaps we need to isolate these targets and apply whole genome sequencing on these targets as well. We talked about the genomics. It may be interesting. It may provide the results. It may be misleading if we are not sure how to translate the results that we get at the end of the day. We certainly need to use quantitative PCR because it's not an on-off situation. We are not interested on just the presence of a gene. As we said before, we are interested in the fluctuations taking place throughout the year. And that is why we need to harmonize and normalize the results we get. We need to get spiking for the recovery testing. We need to make sure that all these conditions are not greatly affected by other inhibitors or other situations that may take place, for example, increased dry conditions or increased dry conditions, etc., that may lead to fluctuations which may not have to do with the resistance itself. Digital PCR is very nice, it's very simple, but we need to make sure that we do not have false negative results because of the limited saturation levels. And at the end of the day, we need to make sure that we use reference materials if we can. In the case of ARG panels, we need to make sure that we are okay with the sensitivity and specificity of these ARG panels, because in most cases, these ARG panels have not been dedicated to work on wastewater or on samples that are full of inhibitors. So we need to be double sure about that. What about chemicals? Does chemical composition play any role? Yeah. If yes, which chemicals do we need to test for? This is a huge question. I'm not going to get into that. I'm just raising the question. And what about biofilms? Great subject. lots of work that has been done and is currently done on biofilms. We will not touch it now in our speech at all. So, based on all these questions and the questions that we initially have, we decided to focus on two cities, the two largest cities of Crete, Heraklion and Chania. They represent roughly half of the population of the island of Crete. And we also focused on the university hospital situated in Heraklion, which is the largest hospital of the island of Crete. So we got these two wastewater treatment plants, and we decided to test everything that we could. So we had samples coming into the wastewater treatment plants and leaving it, either before chlorination or following chlorination or following dechlorination just before the dispersal into the environment. And that was the case with the hospital as well. We collected and tested samples getting from the hospital before and after treatment. The experiments took place in October, November, which is still is a dry period for the island of Crete. We intend and we anticipate to repeat every experiment we have made on January, when we will have loads of water hopefully coming into Crete. We need to make sure to see whether the wet conditions may or may not affect the result that we get. And we decided to work with instant samples instead of 24-hour samples. We chose to initially work with Promega, which is the environmental total nucleic acid kit. And the reason behind that is that we have been working almost 3 or 4 years with this extraction method. It's the common method that all the labs in Greece that have been associated with the surveillance of SARS-CoV-2 work with, because we need to have something in common. And we decided to initially go with this one, and at a later experiment that we will apply, we will go with another kit that we will be talking about it later on. So we followed the extraction methodology that the manufacturer suggests, and we also applied some nanotrap microbiome particles, which are being used for microbiome studies. And we also applied the microbiome kit, which uses benzonase to destroy the cell-free DNA. And we also used some extra certifications where we had the chance to pick up the pellet of bacteria, break them down using big beating, and then apply the amplification analysis. So we play around with the extraction methods to see what was the case based on the extraction method and on the sampling point. So how did we amplify our genes? We used a panel that mainly targets beta-lactams. We used hybridization, which, as you can see, can target a number of genes corresponding to several different antibiotic. And we also applied 16S RNA. We will be talking about why we did that later on. We also applied whole genome sequencing. I will not give you any results at the moment because we are still on the way of processing them. Apart from E. coli, we decided to play around with all these 4 bacteria as well: Acinetobacter baumannii, Klebsiella pneumonia, Pseudomonas aeruginosa, and Rheumatophilus faecium. We isolated by filtration. We counted them, so we had a rough idea of what was their number on the effluent and on the effluent samples. We identified them, and we processed them by whole genome sequence. So, what did we get? Regarding BLA, you can see BLA-MOX and BLA-FOX, and you can, from the very beginning, you can see that we have different results when we are dealing with different genes. What we can also see is that was the case all over the course of our study was that we got the majority of the results that we got had to do with influent samples. Influent, you can see influent. Disregarding the methodology and the extraction that we used, we got major results on the influent samples, not on the effluent samples. And that was the case with, sorry, let me just move this from here. Yeah, because it's a little bit annoying. We had this condition with blah CNY. Again, we had some nice results on the influent samples. We had something on the effluent sample, but had to do with the hospital, and that is why at the very beginning of the speech, I told you that we have to decide which sampling points we need to consider on the testing. You can see that we got on this gene target, we got results not from every place that we tested for when we use promega or microbiome or beat beating. On the other hand, when we targeted the same gene by hybridization, we detected this similar gene on every sample. What does this mean? This means that we need to make sure that the sensitivity of our method on our panel is what we are looking for. Otherwise, we may end up testing for genes, having a sensitivity that is greatly affected by inhibitors, for example, and getting false negative results. And this is very crucial. Moving on to BLAST-CTX, we have some very, very poor results. And again, we had results only on the influent samples. Regarding IMP, that was pretty annoying because we found it on the effluent sample as well. I won't say on that any longer because that was very annoying for us, to be honest. Moving forward into BLA-VIM, and we are now playing with carbapenems. Again, we had in this case, sorry, we had most of our results being present on the effluent samples, not on the effluent samples. And we can see that we moved from one target to another, and at the same time, we moved from one sampling point to getting positive results from a different sampling point. And this is crucial as well. This may mean that we should not focus on a specific sampling point only. We got poor results for Blaoxa, as you can see, and pretty much poor results for Blaoxa 2440 as well. And that was the case with the other OXA targets that we tested for. The nice part, if we can speak about the nice part, is that we rarely saw any positive results on the effluent samples. Here we got the results from the effluent sample, but this comes from the hospital. We didn't have any results, or we hardly had any results from effluent samples coming from the water treatment plants. This means that either we have failed to detect the genes or that the water treatment points, they do their job very nicely, and what they disperse in the environment is pretty much clean. That was the case with black KPC and blind NDM. We had some poor results, and they mostly relied to influent samples as well. I will skip gas because we got it pretty much as where it doesn't seem to work very much as a target that we can rely on. We got SULA and SULD, which correspond to sulfonamides everywhere, but only through hybridization. That was a different panel, as we mentioned before. And that was the case with quinolones, QNRS, and that was the case with CATB3, which corresponds to chloramphenicol resistance. And that was the case with AAC that target aminoglycosides. That was not the case with VAN-A and VAN-B that target vancomycin. We got results from influent samples only. Yes, so the take-home message from the very preliminary results and the 2-month study that we have carried out is that we need to be very careful. If we want to apply ready-to-go ARG panels, we need to be very careful on how these panels work on different samples from different sampling points that may carry or may carry not a different amount of inhibitors. So that was the first case. Then, as I mentioned before, we did some 16S. Why? We need to figure out what was the profile hiding behind the samples that we were targeting for. We need to find out whether the extraction method does affect the final result, or whether the profiles that we get have to do with the sampling point or the sampling sites. So if we focus on the whole 16S profile, then we can clearly see that the profile taken from the different samples, whether the samples corresponding to Hanya or Heraklion, pretty much gathered together. On the other hand, the effluent samples may look likely similar, with the exception of the effluent sample from the hospital, which is, of course, something that we expect to happen. If we play around with the 16S profile on the whole, but we focus on the methodology used, then we can see that the effluent samples gather together, regardless of the methodology used. On the other hand, only microbiome and promega extraction methods work well with effluent samples. And that was the case with the profiling. You can see that every effluent sample pretty much resembles previous one, disregardless of the methodology used, and this is not the case with the effluent samples. On the other hand, if we focus on this 16S based only on the 5 target, on the 5 bacteria that we are interested in, then the profile and the arrangement of the results on a 3D basis is completely different. It's completely mess. We cannot expect to find any sort of association. So we need to make sure that at the end of the day, we've got the correct sample, which may give us the correct true positive result if we are interested in targeting selected bacteria. We are not ready to show you any whole genome sequencing results. What I can tell you is that we have some bacteria that carry resistance, but I will leave it there. So, as a conclusion, different wastewater treatment plants produce different results. That is the case, and this is very crucial, because if we want to apply a common SOP, we need to make sure that this can be applied horizontally. Different extraction kits produce different results. Very, very crucial, because at the end of the day, if we got different results and we got positive results from an extraction kit and positive results from a different extraction kit, then at the end of the day, we may need to apply more than one. Nanotrap seems to be promising. We're not sure whether rain will affect the results. As I told you before, we will repeat the experiment on January. We're not sure whether seasonality may affect the result, and this is why we will continue repeating these kind of experiments when the tourists anticipate to arrive in Crete. That's the case late April, early May, for example, because they may affect the profile of resistance in our wastewater. As I said before, we anticipate to repeat the whole experiment. We anticipate to complete the 16S and whole genome sequencing analysis. We will bring the case into case the DNeasy PowerSol Pro kit. We need to make sure that we can cover as much of the gaps that may be present there. We need to make sure that our results are close to reality, and that is why we need to try more than one extraction kits. We will be collecting samples during the rainy days. We are not sure whether we will be doing any metagenomic analysis. we need to make sure that if we spend some thousands of euros, we are capable of providing a nice translation of these results. We will set up the qPCR because at the end of the day, we cannot target for so many genes. We will try to do something machine learning on the results. At the end of the day, it's not just a matter of targeting what we know, but it's a matter of unrelieving of what will come into play over the next few months or few years. In any case, the million-dollar question, if we record an increase in resistance, what are we prepared to do in terms of lowering, and how much intervention can we afford? These questions are more questions to be answered from politicians, not from scientists. So I'm just putting them on the table and leave them to you. Our quick advice is that if you want to work on AMR resistance on wastewater, please seek advice from other people in other countries. They may have done something that you don't need to invent the wheel from the very beginning. And this is why I need to place some special thanks to Professor Clementina Cucuzza from Italy, who was very keen on providing us with her expertise, some major stuff that they have done in Italy. So we didn't have to start from the very beginning. This is a whole research team. If you want to go for the surveillance, for the proper surveillance of AMR in wastewater, you need medical doctors, such as we have Kyria Trifinopoulou. She gave you an excellent speech later before, because they can give you the true idea of what's happening into the hospitals. You need mathematicians, you need bioinformaticians, you need biologists, biochemists, and you need someone, of course, that like Professor Anzolotti in charge of all that, because you need someone to connect all these people. So this study is in the frame of EU-WISH joint action, which is funded by HADEA. And I will finish up with a sentence: wastewater surveillance acts as a small detector for the community. So at the end of the day, it's not just playing around, it's providing a ring bell of what will come in the future. Thank you very much for your attention, and I hope, I think I'm on time. So any questions, happy to answer.
Thank you very much for your presentation. So if anyone here have a question at the right moment to do it. Yeah, Uli, please.
Hey, Uli Klum from TU Dresden. Thanks for the talk. I believe that, maybe more of a comment, one of the problems with your effluent samples, and based on our experience of doing also wastewater surveillance for quite a while on AMR, is that the 40 milliliters do not give you a representative sample ever. So I think what's necessary is to go for at least a liter, so that you cover enough of the diversity to get representation. And that might lead to a lot of the randomness you see in your data. So as a recommendation, maybe, since you said you wanted to continue it, we usually run with filtering and then extracting from that, because that seems to also decrease the randomness of which bacteria are pelleted and resuspended in the pellets. And I think that might actually dissolve both the sensitivity issues as well as the accuracy issues with some of the results.
Excellent. You're to the point. You're to the point. We decided to go with Promega. We receive on a daily basis, or almost daily basis, 1.5 liter samples from the wastewater treatment plant for the sake of SARS-CoV-2 surveillance. So we said, okay, we'll go with Promega, which has a larger sample than the other kits. Our concern is what exactly you mentioned. We need to make sure that we are very close to reality. And yes, increasing volume will decrease discrepancies such as the ones you mentioned. we will be playing around. We need to make sure that, you know, at the end of the day, the more you spend now, the less you will spend later on, and the better result we will come on a later basis. So yes, your comment is right to the point. Thank you very much.
Thank you for your answer and your question. Anyone else here? I have one question for you. Do you have any idea why you have so much differences between the two kits you use for the extraction?
Nice one. I guess it has to do with the ability of the different kits to get rid of the inhibitors present. You know, the tricky part with wastewater treatment plants, especially the one we have in Chania, is that it receives, and in Heraklion, but mostly in Chania, is that it receives wastewater from the industry. You know, industrial samples tend to have heavy metals, soaps, etc., etc. So it's a very nasty, sample to play with. It's not like a sample coming from a hospital, for example, where you can only find feces from patients and nothing more. Pretty much nothing more. So my best guess is that the sensitivity of these two kits is completely different. And this is why and based on the suggestion of Clementina, the professor in Italy, we decided to get into play with the kit from Kiagen. Perhaps it can solve us some queries on whether we can get rid of these inhibitors. We anticipate to have some, let's say, better results when we do the whole experiment from the very beginning on January.
Okay, thank you very much.
Thank you.
So I think we can go for the next speaker, unless if there are any questions online. No? Okay, so we can go to next speaker. So thank you again very much.
Thank you. Thank you very much.
And so now Poppy Karaolia a researcher from the University of Cyprus, will talk about tracking antibiotic resistance again, but across a Mediterranean city and through wastewater. So whenever you are ready, you can start.
Thank you so much. First of all, I would like to thank you for the very, very kind invitation to present our work here at this very nice conference. So I would like to start by presenting the title of our work, which is Tracking Antibiotic Resistance and Pathogens Across the Mediterranean City of Larnaca. And that will provide some insights from wastewater surveillance and risk assessment done in this city. So my name is Poppy Karaolia from the Nereas International Water Research Center of the University of Cyprus. And I would like to start with some background, as was previously presented very nicely by Demosthenis. Antibiotic resistance has been linked to around 4.7 million deaths in 2021, and it is projected to increase to 8 million deaths by 2050. Bacterial resistance is now growing faster than the drug discovery, and the multi-drug resistant bacteria specifically are very common and harder to treat. Then we have sewage effluents, which are sources of both antibiotics as well as antibiotic resistant bacteria, which are contributing especially to the environmental dimension of the antibiotic resistance crisis. In Cyprus, we have very serious problems with antibiotic resistance, as we have the highest percentage in Europe on MRSA or methicillin-resistant Staphylococcus aureus infections among hospitalized patients. And we also have some very high infection rate, especially for third-generation cephalosporin-resistant E. coli, among others. So this is a very important reason for us to proceed with wastewater surveillance, specifically for antibiotic-resistant pathogens. And wastewater surveillance has been previously shown to capture both pathogen and antibiotic resistance determinants very nicely at real time and from entire communities. It is a very anonymous and very non-invasive process, which is cost-effective for population level monitoring, as with one sample, you can get a lot of information for whole communities. It can be used to complement different public health data, including clinical monitoring, early warning systems, as well as facilitate and check the effectiveness of public health actions. And it has the capacity to detect hotspots of infection and outbreaks of known as well as emerging or new infections within a specific community through, for example, hospitals or airports. So wastewater surveillance is systematic testing of wastewater for public health benefits. And in our study, in the case of Cyprus, we have used it to monitor both pathogens as well as antibiotic resistance genes. The objective of this 2-year work were to systemically investigate the abundance of pathogens and clinically relevant antibiotic resistance determinants through the city sewage network in 10 different sampling points, which I will present in a while, to understand the partitioning of antibiotic resistance genes between the solid and liquid fractions of wastewater in order to be able to really understand how we can utilize wastewater samples and what methodologies are the most suitable for this kind of surveillance, to explore the taxonomic diversity in each fraction through whole genome sequencing, and understand which sampling locations are suitable for specific wastewater surveillance programs. So the novelty of the study is that it is one of the first studies to fully utilize the sewage network of a city through 6 different pumping stations spread around the city, the hospital wastewater, airport wastewater, as well as the influent and effluent of the wastewater treatment plant of Larnaca. And we also aim to contribute to the knowledge regarding refinement and harmonization in the future of the laboratory methods which will be available for antibiotic resistance wastewater surveillance. So here you can see Larnaca in Cyprus. It is the 3rd largest city. It is located in the eastern coast. It has a permanent population of around 72,000 and approximately double that population during the summer and the touristic period because of the inflow of people for tourist reasons. And it's also the host of the Larnaca International Airport, which is the biggest airport in Cyprus with approximately 11.6 million passengers in 2023. So this system is a combined sewer system, meaning that it collects both stormwater from rain events as well as the sewage drains that collect all the sewage from urban settings. An average approximately 10,000 cubic meters per day estimated in 2023. And to do this project, we have employed 24-hour composite sampling through automated autosamplers in order to have averaged and time proportional samples, which are suitable for a robust temporal and spatial comparisons throughout the city. So we have done 2 full years of bi-weekly samplings or every 2 weeks through the 6 pumping stations, airport, hospital, influent and effluent of the wastewater treatment plant. The autosamplers were refrigerated and the samples were 24-hour composite samples. Here you can see the network of our sampling points across the city and how each is connected with the other one. So the methodology that we have utilized is collection of samples followed by sample preparation and extraction. We have conducted solid and liquid separation of the two fractions of the samples in order to be able to evaluate the usefulness of both fractions in wastewater surveillance programs, to evaluate ARG partitioning, antibiotic resistance gene partitioning, to see the taxonomic profiles that exist in both fractions, and try to understand the resistome across these two types of wastewater. We have done targeted qPCR for a panel of priority markers based on some prioritization exercise that we have conducted in the framework of the program. You can see the targeted antibiotic resistance genes, mobile genetic elements, and 16S here in this list. And we have also done shotgun metagenomic sequencing for whole genome purposes. So the methodology followed to be able to understand the removal of ARGs and pathogens after the wastewater treatment plant process is shown here. We evaluated the removal efficacy across the wastewater treatment plant, which follows membrane bioreactor followed by chlorination. And this equation was utilized in order to understand the percentage removal of all the targets investigated. So for the quantitative microbial risk assessment, or QMRA, we have used a metagenomic risk assessment, which examines basically the full spectrum of genetic material available in the wastewater samples. We have done the 16S whole genome sequencing, so we have proceeded to do the risk assessment based on that information. This type of metagenomic risk assessment can provide two types of information. So here, as you can see, first we have the human health resistome risk, which is basically providing information on the likelihood that antibiotic resistance genes or pathogen genes can affect human health, either through the transfer by human relevant bacteria or through specific routes of exposure, including inhalation, drinking, ingestion of crops, for example, irrigated with wastewater or treated with water that could be used for irrigation purposes. And then we have the ecological resistome risk, which is basically giving the information on the potential of ARGs to persist and spread in different environmental compartments. And this type of evaluation basically uses the abundance of ARGs that are found during the metagenomic sequencing, their association with different mobile genetic elements, and their potential to transfer between bacterial species, increasing horizontal gene transfer probability. The software used is called MetaCompare, and this is the 2.0 model, which provides a multi-layered analytical pipeline for potential mobility of ARGs. The clinical importance is taken into consideration, as well as the pathogenic relevance of ARGs. For example, we are looking at how relevant are the ARGs that we found inside our samples to specific pathogens that could be of importance to human health. So let's start with the results of our work for the 2 years that we have done this from 2023 until 2025. Our work is just finishing now. First of all, we have examined the prevalence of the escape pathogens. These are the 6 most relevant bacterial pathogens to clinical settings. We have Enterococcus faecium, Staphylococcus aureus, Klebsiella, Acinetobacter, Pseudomonas aeruginosa, and Enterobacter. Here you can see the results for 5 out of the 6 bacterial pathogens. You can see that the absolute concentrations that we have measured in our wastewater samples across the 10 sampling points. And here you can see that Pseudomonas aeruginosa had the highest absolute prevalence or results as gene copies per liter. While in the bottom figure, you can see the relative concentration of the pathogens. The relative concentrations are basically the gene copies normalized as to the 16S RNA found inside the samples. When we're looking to different types of information, for example, absolute concentration compared to relative concentration to the bacterial load found inside the samples, we have two different pictures. For example, for absolute concentrations or gene copies per liter, we see Pseudomonas aeruginosa as the bacterium with the highest concentrations. But when we normalize our information as to the bacterial load, then we have a very different picture. And we also have some very high relative concentrations found inside the treated effluent, which is very interesting because this is not shown during the absolute concentration evaluations. So we have proceeded to evaluate the different types of escape bacteria that we have found inside our samples. We have seen that Klebsiella pneumonia and Acinetobacter baumannii were found at the highest concentrations among the different types of bacteria found. And they were found in all types of samples, including solids and liquids, and especially in solids, and throughout all seasons, regardless, of course, of touristic flows or any other type of differentiation. So when we proceeded to evaluate the different solid and liquid fractions of our samples regarding the antibiotic resistance genes, we have found that the solid fraction holds the majority of the ARGs regarding absolute concentrations. And this gives us the indication that the antibiotic resistant gene loads are very strongly spatially structured, bound or sorbed onto the solid. And we also have airport, hospital, and some pumping stations showing systematically higher absolute abundances. And this is very interesting because it gave us an indication that solids is a better indicator and a better tool on which to perform wastewater surveillance programs when dealing with methodologies. And then we looked at the absolute abundances of different target ARGs. We have found that among the 10 sampling points that we have evaluated, the absolute abundances were highest in the airport wastewater, as 4 out of the 9 indicator targets were highest in the airport wastewater. Then we have found that the urban pumping stations had the highest concentrations for Vanay, Sul2, and Bla Oxa, while we found that the hospital wastewater was not the leader in ARG abundance, with 2 out of 9 targets being the highest among all sampling points. This gives us an indication that different locations may have different dominant genes and sector-specific resistance pressures, which may be also environmentally relevant. Then we have evaluated the relative abundance of these ARGs according to the bacterial load. Interestingly, we have found very similar relative abundances across all pumping stations, hospital, airport, and effluent. However, the effluent gave a different picture. The conclusion from this investigation that we have done is that some ARGs remain proportionally high compared to the other ARGs that were quantified, or are even enriched after treatment. And this is the case with the effluents. As the relative abundance of different genes has changed in the effluent, this gives us an indication that there is some treatment-associated selective pressure for specific genes in the treated effluents. And we have a residual resistome, which remains even after specific treatments which are effective for E. coli, for example. And these types of ARGs are released in downstream environments and may be found in the reclaimed water that could be used for irrigation, for example, of agricultural crops. And then we have proceeded to characterize the community that lives in each location. I will not go into much detail with this. I just want you to have a look at the colored, bold colored phyla that you can see. The bottom line here is that each location has its own distinct microbial footprint. However, we have observed a shared urban core microbiome. As well as some site-specific enrichment. For example, you can see that proteobacteria are found in hospital, wastewater treatment plant effluent, as well as pumping stations, but not in the airport. This is interesting because we may, of course, after more investigation, we can further examine the microbiome in airport wastewater and determine exactly how it is different compared to the other local wastewater samples. Then we have done some beta diversity analysis of our metagenomic samples. Interestingly, sampling location was the main driver of the community or resistome structure. Season, which is something that has been discussed before, was not shown to have a significant impact on the resistome, except only in the case of DNA viruses, because we're examining the 16S fraction of wastewater. Sample type was also important and significant as a driver for all categories that were analyzed, but specifically for DNA viruses. And this DNA viruses category showed some very distinguished results compared to the other categories. And it is the only category showing significant differences in season, and the only target that was mostly partitioning in the liquid fraction compared to the solids. And this is very important because we may want to do some surveillance for DNA viruses, for specific DNA viruses. Therefore, we may need to look into the liquid component of the wastewater instead of the solids to have a more accurate picture of what is really there. Next, we have done some non-metric multidimensional scaling. This is just to show the clustering of the samples. What we have seen is some clear clustering by location, as we have seen before. Pumping station wastewater closely aligns with the influent of the wastewater treatment plant, of course, because all the pumping stations lead to the wastewater treatment plant. While we have some distinguished behavior in hospital, airport, and the effluent. When we normalized to 16S, all of the ARGs that we have quantified, we have seen some weakened association of the samples with each other, but the differences were not erased, with the effluent remaining in its distinct category and clustering the effluent samples by themselves, suggesting that wastewater treatment may have some important reshaping effect on the resistome of the wastewater. Here you can see the viral community structure that we have also discussed before. Importantly, we have noticed that the DNA viruses were more diverse in the liquid fraction compared to the solids, highlighting that this liquid phase may be the more important part of the wastewater for surveillance, but also it could be the main dispersive route for viral particles if it's found in treated effluents and released into environmental compartments. We have also investigated the fungal community structure. We have found that the influent has extremely low fungal diversity, as it is dominated around 97% by human-associated candida yeasts. The effluent also shows this type of behavior. It is candida dominated. It has some more heterogeneous species composition, indicating that this type of treatment may reshape, but not remove this part of the yeast community. So this indicates that treated effluents also contain some fungal and yeast-associated organisms, which should be considered when releasing effluents into environmental compartments. So regarding the removal efficiency that we have investigated, here you can see both the absolute quantification done for antibiotic resistance genes, as well as the relative abundance of these gene targets. We have observed around 90% or more removal for the majority of the ARGs that were examined. However, when we normalize to 16S, removal is much less pronounced, and some genes were shown to be relatively enriched compared to the total bacterial load, showing that we may have a core resistome that persists through treatment and is released into the environment. Next, I will show you some of the risk assessment results that we have obtained. We have obtained the human health risk ratios first using the MetaCompare tool. Here you can see the risk ratios that were obtained. The order of magnification of the risk ratios is shown here. We have the highest human health risk ratios in hospital, followed by airport, pumping stations, influent, and effluent with the lowest one. It is important to note that we had consistently higher risk ratios in solids compared to liquids. However, the solids had much more both spatial and temporal variability throughout the 2 years and within the 10 sampling points. So, however, liquids, even though they had lower risk ratios, they had lower, a more limited range of risk values. Regarding the ecological risk ratios, we had the exact same pattern found in this case as well. And we try to provide some discussion here to understand how each fraction of the wastewater can provide more information and why it is important to investigate each section. Solid fraction is important as it provides a reservoir or a sink of antibiotic resistance genes and potentially pathogenic bacteria. The benefits that the microorganisms get from the solids are physical protection from environmental stressors, of course, high surface area, which facilitates horizontal gene transfer and protection from external enemies. And solids can provide some very dense microbial communities, which may facilitate and may provide the nutritive ground for horizontal gene transfer. However, this is important as we may, in certain countries and in certain conditions, the solids obtained from different sludge, from the sediment or particulate matter from wastewater treatment plants may be reused in agriculture. So this is very important because these solids may carry very high loads of antibiotic resistance determinants. The liquid fraction is also important, not to be neglected, as it provides information on the more mobile or more dispersive fraction of wastewater, where we can have information on how microbial cells and DNA can be transferred and mobile. However, keeping in mind that they are subject more easily to dilution effects, to different disinfection impacts, as well as the transport done in the water. Next, I would like to show you some work that we have done to understand the health status of Larnaca based on the analysis that have been shown until now. We have utilized clinical data from the general hospital, specifically the clinical data from hospitalized cases that have been associated with antibiotic-resistant bacteria. We have communicated with the Ministry of Health for this, and they were very kind to provide this information. Here you can see the heat maps showing the strength of correlations of wastewater data with clinical data in different lag time periods. As you can see here, we have some very good results for sulfonamides based on the clinical data, confirming that we have a very temporally structured coupling between wastewater and clinical antibiotic resistance data. However, we did have some weaker signals regarding quinolone resistance data. with positive correlations, weak positive correlations, showing from 10 days approximately to 2 days before the clinical data onset. However, this gives a nice indication that we could, with further work and deep understanding of the results, a nice early warning system for antibiotic resistance. However, we need to investigate further different aspects of this process, including, for example, the reporting lag that exists between the offset of a disease until hospitalization and reporting to the medical establishments, hospitalization procedures, and how we can get more information through the hospital, but also within urban wastewater on this type of infections, and the true reporting rates. So the conclusions of this work will be presented here. We understand now that location has been the dominant driver of antibiotic resistance in our wastewater samples and pathogen patterns in Larnaka's wastewater network. Wastewater antibiotic resistance trends may anticipate clinical resistance with the right amount and right type of work, providing early warning signals for related infections. And wastewater treatment may substantially reduce the absolute risk, but we have a resilient core resistome persisting after the treatment. Finally, Larnaca has provided a proof of concept citywide One Health surveillance and risk assessment framework, which we further aim to utilize for more work to be done in the future. We have done integrated wastewater surveillance, which uses qPCR targeted analysis, metagenomics, as well as qMRAs, plus clinical data, which can produce very robust, actionable insights into the AMR dynamics at a city scale. This work aligns with EU regulations of wastewater reuse, calling for pathogen and AMR surveillance in urban wastewater. And this work supports embedding wastewater monitoring into routine One Health aligned surveillance, as well as risk management frameworks, both at local and EU levels. So some future work and plans that we're planning to take on in the next year is to scale probably and diversify the surveillance network that we already have in our hands, to dive deeper into the metagenomic and resistome interpretation of our findings, to strengthen and operationalize qMRA that we have already started, to provide some scenario-specific exposure and dose response information, and try to understand further how the exposure to AMR and different doses of antibiotic-resistant bacteria may impact organisms, especially in receiving environments. Try to understand better the correlation of clinical data with wastewater to turn wastewater surveillance into a predictive tool instead of just a monitoring tool. To evaluate treatment and mitigation options, which help to reduce the burden of antibiotic resistance genes in the effluent even more. To integrate this type of work, this integrated network, into policy regulation, as well as decision support systems for public health purposes, and integrate clinical and environmental data collected so far into One Health framework. So finally, I would like to acknowledge the very kind support by the Larnaca sewage treatment plant personnel, the finance authority, Larnaca District local organization that has provided the funding for this work and the support, the technical support to achieve these results, as well as Novogene, who has done the bioinformatics, the sequencing work. And finally, I would like to acknowledge the work of all the team, including Nerea's International Water Research Center, as well as Sotirios Vasiliadis from the University of Thessaly, who has very kindly supported us and worked throughout this work with us, and will continue to work for very nice results and publications. So thank you very much for your attention. Thank you for the kind invitation to the organizers. And I look forward to any questions. Thank you so much.
Thank you very much for your talk. Due to time limitation, we will have time only for one question. If someone have one question here or online, so that's the right time online as well if you want to ask one question. Nobody? So I will ask a very short question then. When you shown your microbial fingerprinting over a location, did you notice any variation over the season? It's always consistent to have that fingerprint, or it's something more variable?
Yeah, so we have observed some variation season-wise. However, the season was not a defining factor for the microbial taxonomic community. So it did not really impact and produce significant changes in the resistome as such. However, the location was the defining factor of and the distinguishing factor, let's say, between the different microbial communities, meaning that season did produce impact, but it wasn't significant enough to provide unique resistome profiles.
Okay, thank you very much for this clear answer and for, again, your presentation, and then we can move to the next speaker.
Thank you.
So it will be Antonia Mataraka. Okay, so. Okay. So Antonia is from Greece. She's a postdoctoral researcher in animal science, and she gonna talk about antibiotic resistance profile linked to Salmonella serovar from egg products. So whenever you're ready, that's fine. The floor is yours whenever you want.
Now okay. Thank you very much. This presentation is identification and antibiotic resistant profiles of clinically important Salmonella serovars isolated from egg products. First of all, I would like to thank the team that worked on this project. Anastasia Hina, that it was a part of her graduate study, this project. Myself, Panagiotas Stathopoulou, and Nikolaos Andritsos from the University of Patras. So, first of all, why Salmonella? Of course, to set the stage, it is important to recognize one more time the significant public health burden posed by Salmonella, where millions of cases occur every year, leading to hospitalizations, economic losses, and severe cases, even death. So among all foodborne hazards, bacteria, viruses, parasites, and toxins considered in the WHO Global Burden Estimates, bacterial diarrheal agents, including the NTS, non-typhoidal Salmonella, accounted for a large scale of for a large share, of foodborne illnesses and deaths. On the global stage, NTS is recognized by WHO as one of the 4 key global causes of diarrheal diseases. In the European context, according to the latest 2022 European White Zoonosis report by EFSA and ECDC, Salmonellosis, the NTS serovars, is the second most reported zoonotic foodborne infection in humans after Campylobacteriosis. The persistence of NTS, notably serovars such as Salmonella enterica, Serovar enteritidis, Entifimurium, in food underscores its global relevance for food safety. So while most NTS infections cause mild to moderate gastroenteritis, a subset becomes invasive, the INTS, causing severe systemic infection associated with high case fatality rates. Like infections, like sepsis, bloodstream infections, etc. So in this table, we see the most recent internationally valid burden of both NTS and INTS. The invasive NTS. For the NTS gastroenteritis, the most comprehensive global estimate remains the 2010 meta-analysis study that approximately 93.8 million deaths worldwide were occurred worldwide each year. These estimates highlight that roughly 80 million cases were foodborne. For the INTS disease, the most up-to-date global figures comes from the GBD, the Global Burden of Disease, from 2021 analysis, which estimate that there was that excuse me, that was in 2025 released, and this global figures estimates approximately 10,000 cases, 62,000 deaths, and 4.74 million DALYs globally to 2021. These data reflect the substantial regional heterogeneity, with the greatest burden occurring in sub-Saharan areas. So, although most uncomplicated salmonellosis cases do not require antibiotic treatment, the treatment is indicated in severe or invasive infections and in vulnerable patient groups. Ampicillin was historically the first-line antibiotic, but the widespread resistance of Salmonella enterica has reduced its usefulness, and in such cases, fluoroquinolones like the ciprofloxacin are generally considered first-line therapy for adults, while third-generation cephalosporins like the ceftriaxone are preferred for children and hospitalized patients. The azithromycin is used as an alternative option, particularly in areas where fluoroquinolone resistance is recorded higher. So, the aim of this study was to investigate the distribution and the AMR of three of the most commonly detected foodborne clinical serovals of Salmonella enterica, the Enteritidis, Fimurium, and Thompson, isolated from egg products. Samples of commercial pasteurized egg products and of raw eggs were collected from the period 2016 to 2018 and were microbiologically analyzed for the detection of Salmonella species. 10 Salmonella strains were recovered from an equal number of samples, and these strains were further biochemically and serologically confirmed as Salmonella species according to the ISO 6579-1 protocol. So, a brief protocol overview. First, we homogenize the test portion and add it to a pre-enrichment medium, BPW, water, at 1:10 ratio. Then we incubated at 37 degrees for 18 hours to revive the stressed cells and allow even low numbers of Salmonella to multiply. Then we use two selective enrichment broths in parallel. The RVS broth at 41.5 for 24 hours, and the MKTTN broth at 37 for 24 hours. Next was the streaking each enrichment into two selecting agar plates, the XLD and the BGA agar, and incubation for 37 degrees for 24 hours. And finally, we examined the plates from the, that were presumptive Salmonella colonies and picked these typical colonies for further biochemical confirmation as we see here. Following the confirmation, the isolates were further processed for serotyping through multiplex PCR, according to the protocol published from Simizu et al. In 2014, for the detection of Salmonella serovars, Erytidis, Typhimurium, Thomson, and Infantis. After this step, we proceed to the screening for AMR against a panel of 8 antibiotics that we see here, the ampicillin, tetracycline, meropenem, norfloxacin, gentamicin, ciprofloxacin, ceftazidime, cefotaxime, all based to the UCAST criteria, European Committee of Antimicrobial Susceptibility Testing, and the version 15 that is the latest version. So, regarding the result, the serotyping of the isolates revealed that Enteritidis was the most prevalent serovar in 4 of 10 samples, whereas Thomson, Typhimurium, and Salmonella species without that did not further proceed to we couldn't identify it further based on the specific protocol, where 30% for Thomson, 20% for Typhimurium, and one sample was unspecified further as Salmonella species. This indicated that perhaps a product can be contaminated with epidemiologically significant Salmonella serovans, and we will see later on. In this table, we see that all but the 2 strains, the 4th and 5th, the AAL 10.11.9 and 10.12.7, revealed some kind of resistance. In more details, all but these two samples were susceptible to all antibiotics tested. The 10.11.9 Typhimurium strain was isolated from pasteurized egg yolk, showed an intermediate resistance to ciprofloxacin, but it was finally deemed as susceptible after determination of its minimum inhibitory concentration, its MIC, according to the UCAST criteria. However, AMR AMR against ampicillin, tetracycline, and cefotaxime was recorded for the S. Thompson strain, the 10-12-7, the S. Thompson, that was isolated from pasteurized egg white, and finally deemed as resistant in 3 classes of antibiotics, penicillins, tetracyclines, and cephalosporins, and thus was classified as a multi-drug resistant strain. So in the set of 10 isolates from egg products, S-enteritidis was the most frequent serovant, 40%, followed by S. Thompson in 30%, S. diffimurium in 20%, and one anti-salmonella 10% of the samples. This distribution is consistent with long-standing surveillance data and identified enteritidis as the serovar that was most commonly associated with eggs and egg product outbreaks in Europe and elsewhere. The EFSA ECDC One Health surveillance reports and annual epidemiological summaries repeatedly highlight enteritidis as the primary agent in egg-linked salmonellosis and outbreaks, and identify egg and egg products as a high-risk food category for the salmonella transmission. Regarding the serotyping results of this study, at the same time, detection of the diffimurium aligns with many egg and egg product surveillance studies that recover S. diffimurium as a common serovar in eggs or poultry environments, although typically less dominant than enteritidis in egg matrices. Focused surveys report variable proportions by region, like, for example, studies from Asia and other countries than Europe that have reported the diffimurium serovar amongst the top serovars recovered from the eggs. The recovery of Thompson is notable because Thompson is less commonly reported as the dominant egg serovar in many large surveillance studies and summaries, yet it has been documented in eggs and poultry and has been implicated in sporadic outbreaks and in transmission between poultry and humans in multiple studies. Several recent studies have specifically documented that Thompson is capable to circulate between farms and humans in eggs and the egg matrices and in poultry, and this is why it was a very significant aspect of this study that is presented here. Most of the isolates were fully susceptible to the 8 antibiotics tested, with the 2 exceptions that we discussed previously. The diffimurium showing intermediate serofloxacin by the disc testing, and the one as Thompson that showed high-level resistance to ampicillin, tetracycline, and cefotaxime. This overall pattern, a majority susceptible isolates with occasional resistant and MDR isolates, is consistent with many studies and reports worldwide. The ethnicity surveillance in multiple egg and poultry studies describe a background where many food isolates remain susceptible to a number of agents, but resistance to widely used drugs like ampicillin and tetracyclines is common. Resistance to third-generation cephalosporins and fluoroquinolones, while still less frequent as tetracyclines and ampicillins, is of particular concern when you observe these. For example, the European Annual Report highlights that fluoroquinolone resistance increased over recent years, while resistance to third-generation cephalosporins remains low. The resistance to ampicillin and tetracycline, as I said before, are among the most commonly reported phenotypes in foodborne salmonella isolates, globally in egg specifically sampled. Numerous studies report that elevated frequencies of these resistances are observed. Resistance to cefotaxime is less common in food isolates, but is clinically important because it may indicate ESBL or other beta-lactamase mechanisms. The EU surveillance flags even that low rate of third-generation cephalosporin resistance is significant because of the therapy implications. The observation of the cefotaxime MIC is therefore a epidemiologically, clinically significant result of this study and aligns with the literature that is available and documents. And this literature documents occasional cephalosporin-resistant salmonella from poultry and egg food sources. The MDR specifically in foodborne salmonella is a recognized public health threat. The MDR strains increase the risk that human infections following foodborne exposure will be harder to treat and may require higher tier antibiotics. The EFSA and ECDC One Health reports and recent surveillance studies emphasized that the public health concern when MDR or ESBL associated phenotypes are detected in isolates from foods or animal origins and is of increased significance. MDR-S Thompson has preceded in the literature and multiple studies have described Thompson isolates carrying excessive resistance determinants, including examples of isolates resistance to antimicrobial classes, even to more than 10 agents. In these reports that are not many reports, but are there and it is of concern. These reports support that S. Thompson can act as a vehicle of dissemination of AMR in poultry and egg contexts. Cefotaxime resistance is also particularly concerning. The cefotaxime MIC is compatible with a third-generation cephalosporin resistant phenotype, which is less frequent among the egg isolates, but has been increasingly reported in foodborne salmonella and poultry chains. Such phenotypes may reflect the plasmid-borne beta-lactamase genes that can spread horizontally, increasing the risk of dissemination between and along the food chain. EFSA and ECDC surveillance stresses monitoring of third-generation cephalosporin resistance because these agents are critically important. So Of course, this study is more to analyze, has many more things to evaluate, and the follow-up investigation will be a whole genome sequence of the MDR, the specific MDR isolate to identify the resistance genes to like the ESBL genes and tetracycline genes and etc. to determine plasmid replicons in order to assess horizontal transfer risk and to perform phylogenetic typing to check relations to human, animal, or environmental strains. Another part of this whole investigation will be a screen of related samples from the same production or processing chain to determine whether this MDR strain is an isolated contamination event or part of a wider farmland reservoir. Finally, based on all the above, the serotyping results, meaning the predominance of esenteritidis with S. Thomson and S. Fimurium also present here, are consistent with surveillance data that identify eggs as a principal vehicle of esenteritidis, also demonstrate that other epidemiological important reservoirs could also occur in egg products. Most isolates were susceptible, and this is the good part of this study. Many isolates were susceptible to the tested antibiotics, matching many of the food surveillance findings that show a majority of susceptible food isolates. However, the recovery of this multi-drug resistant strain, S. Thomson, that was resistant to ampicillin, tetracycline, and cefotaxime, is of particular concern because it mirrors recent reports of MDR S. Thomson serovar and highlights and signals the potential carriage of ESBL and ampicillin C determinants. And thank you very much.
Thank you very much for your presentation. So anyone here have some questions? So over there, please.
Thank you. Congratulations for your talk. Since Salmonella antimicrobial resistance is a huge issue for One Health scientists like myself, vets and medical doctors. I didn't understand well the samples that your strains came for. If you're kind enough to go back to your slides for the samples.
Can I go? Okay, of course. 1 minute. Excuse me. Samples, okay? Here.
Okay.
Yes.
Here we can see that most of the samples came out from pasteurized samples. That is rather disturbing. Can I ask, this is random sampling? This is part of official sampling? This is part of self-control of some companies? Thank you.
Yes. Professor Adritos, good luck. Professor Adritos is the head of investigation, so he says he would like to respond.
Thank you very much. Nicholas Andritsos from the Department of Food Science and Technology of the University of Patras. Sorry for not being able to deliver my speech myself, because originally me was the speaker of this presentation, but unfortunately I just came up and I didn't know if I could make it actually. So thank you for your question, Professor Solomakos. I would like to say a couple of things about it because it's a very to-the-point question. So the samples originally came up from the in the context of self-control. So we had so many samples. I cannot even give you the prevalence because those samples came up from 2.5 years. There were above 250 samples screened. Those isolates came from samples that were shipped to an external laboratory in the context of self-control, several industries. So those samples, those 10 isolates come up from above 250 samples. So the samples, as you can see, as you have noted quite keenly, most of them are from pasteurized products. So yeah, this is something we would expect raw eggs, for example, to have more to isolate, to have more isolates from raw samples. But indeed, we had from pasteurized. So maybe some of these samples come from one industry specific. So that's why Antonia told you about that we could investigate with the whole genome sequencing performed as a later stage if we have contamination scenarios. That's why. So the answer is yes. Those samples come from quite a lot of sampling from the context of self-control of some industries. Thank you.
Thank you very much. Anyone here had exactly the same question? So I had exactly the same question than you. So yeah, that's perfect. So if anyone else have question, even online, so I think we can end that session and go for a coffee break. Yeah, all good for online? Okay. So thank you again very much. And thank you for your answers.
Good afternoon, everyone. I have a pleasure to present the oral presentation. My name is Carol Delgado, and now we begin with the first one speaker. She is Pooja Lakwe from Queensland Islands for Environmental Health Science from Australia. And the theme we treat is like parent, like metabolite. Do antibiotics and their transformation product send comparable selective pressures? And she present online. And if okay, good afternoon, Pooja.
Just a minute. I will
Can you hear me?
share. Yeah.
Okay, perfect. When you are
Yeah, I
ready,
think.
it's possible we begin.
Okay. I think my screen should now be visible.
Yes.
Thank you. Thanks so much. Hi, everyone. My name is Pooja, and I am a PhD student at the University of Queensland and the University of Exeter. And today I'll be sharing a small part of my project that I was working on this past year, which is about antibiotics and their transformation products, where I look into whether the transformation products of the antibiotics can also exert comparable pressure. Okay. Starting off, I think right from the start, we've been talking about how antimicrobial resistance has been a global burden, and we've seen most of our presenters earlier share about how there has been forecasts and how there have been predictions on how big the AMR is in terms of global health. But when we talk about antimicrobial resistance, we also have to understand that not only are we looking into antibiotics and antimicrobial resistant bacteria or antimicrobial resistance genes, antimicrobial resistance has become such a big issue that we have now also started looking into the transformation products. So moving on, for a really long time, I think all of us are aware that antimicrobial resistance, whenever we talk about antimicrobial resistance, the forefront or the poster child of AMR has always been the clinical settings, the hospitals, overprescription of antibiotics, nosocomial infections. But the bigger picture when it comes to antimicrobial resistance has always been the environment, as antimicrobial resistance cannot take place in isolation. It needs the environment where the resistance can evolve, grow, and it can be transferred between different environmental sections. Here you can see that when it comes to environment, we're not just talking about water, we're talking about water, soil, air. There are so many current researches where people are looking into antimicrobial resistant bacteria and genes in air as well, and how they can transfer between all of these environmental factors. So moving on to the main topic of my presentation, which is the transformation products. So transformation products are the breakdown products of antibiotics. But when we talk about breakdown products, especially in environmental chemistry, we think that these metabolites or breakdown products, once they're done being broken down, they do not remain biologically active. But there are more and more researches, more and more literatures where people have seen that transformation products of antibiotics, different metabolites of antibiotics, can also remain biologically active. I think one of the main reasons why this happens is because they can persist more in the environment as compared to their parent compounds. And because they remain biologically active, they also have the potential to contribute to antimicrobial resistance selection. So again, moving on to talking about antibiotic transformation products, these are the breakdown products of antibiotics, which can form through different processes, including biological process, chemical process, physical process. And most of these degradation might happen in the environment, but some of it also happen in human body whenever we take antibiotics. But wherever the breakdown happens, it all eventually ends up in the environment, especially in wastewater, because my main subject here, my main sample here is wastewater. So I'll be talking more about wastewater and antibiotics present there. So this is one of the studies that was done by my research group, where we looked into the presence and persistence of antibiotics and different transformation products in wastewater, especially in influent. And here you can see I've highlighted some of the pairs. We can see that these are the pairs. So for example, the one on our left, clindamycin and clindamycin sulfoxide. Clindamycin is the parent compound and clindamycin sulfoxide is its metabolite or transformation product. And we can see that not only is clindamycin sulfoxide present in all of the 50 wastewater treatment plants that we sampled, it was also present in higher concentration as compared to its parent compound. Similarly, we can also see that roxithromycin, which again is another antibiotic that we look into, its metabolite decladinose roxithromycin was present in 48 out of 50 wastewater treatment plants as compared to its parent compound, which is only detected in 3 of the 50 wastewater treatment plants. And similarly, we can see that most of the transformation products of these antibiotics were detected in either in comparable concentration, or if it was detected in less concentration, it was detected in more wastewater treatment plants as compared to their parent compounds. So that was the data that we saw was from influent. Now, when we look into the effluent as well, we can see that some of these transformation products can persist as well, and they are also present in higher concentration. For example, we saw that roxithromycin was present in influent, but it was not present in effluent. However, its transformation product, decladinose roxithromycin, was present in effluent as well. And again, another example of clindamycin, which was again present in influent as well as in effluent, and its transformation product, clindamycin sulfoxide, was also present in influent and effluent. And similarly, we can see there are a few other transformation products which are present in both influent and effluent. So when we detected these transformation products in higher number, not just in higher number, but their persistence was also higher as compared to their parent compounds. So when we saw that, I wanted to know, because I have a microbiology background, so I wanted to know whether the presence of these transformation products was just ubiquitously present, or do they also have an impact on antimicrobial resistance, like how the presence of their parent compounds do. So that was the main aim of my study. So I wanted to look at whether first they can select for antimicrobial resistance or not, and then second, look at how comparable they are as compared to their parent antibiotic compound in terms of selection. So to answer those questions, I followed a I followed the methodology that you can see on the screen here, which started right from sample collection from a wastewater treatment plant, right up to data analysis, which includes first aliquoting and freezing them and conducting 2 different experiments. First was select assay, which is a growth-based assay. And then based on the results from the select assay, I conducted a 7-day evolution experiment followed by DNA extraction, and qPCR to look at NTI1 and 16S rRNA-G. So starting off first with select assay. Select assay is a high throughput growth-based assay high throughput experiment, which was designed by one of my supervisors, Dr. Amy Murray. It is a 24-hour it's a 24-hour growth-based assay where the microbial community from wastewater, because we're doing on wastewater here, the microbial community are introduced to a serial dilution of concentration of either antibiotics or transformation products. And these concentrations are based on the clinical MIC, and then the highest concentration that we introduce are the clinical MIC, and then we slowly do serial dilution and go down until whichever concentration gets supported by the plate. And then based on that, we look at their growth profiles. So I've put some of the examples of the growth profiles that I saw from my experiment. The first one is, of course, ciprofloxacin and desethylene ciprofloxacin, which ciprofloxacin, again, is one of the most studied antibiotics whenever we talk about antimicrobial resistance. So after the 24-hour growth post-select assay, we can see that, so this is an area under the curve figure of ciprofloxacin and its transformation product, desethylene ciprofloxacin. The blue one that we can see, the blue curve is the ciprofloxacin, and the orange one is its transformation product. And we can see that ciprofloxacin can decrease the growth of the microbial community as the concentration increases, which is quite common in terms of antibiotics. But when we look at desethylene ciprofloxacin, we can see that even the transformation product, which up until now was considered biologically inactive in terms of antimicrobial resistance, it was also able to decrease the microbial growth. Not as much as ciprofloxacin, but when we look at it separately, we can see that it can also gradually decrease the microbial growth. And I also have another example of moxifloxacin and moxifloxacin N-sulfate, which is, again, another fluoroquinolone antibiotic and its transformation product. We can see that here the blue curve is the moxifloxacin, and the orange one is the moxifloxacin N-sulfate. However, here we can see that moxifloxacin N-sulfate can decrease the growth of the microbial community more than moxifloxacin itself. So based on these initial 24-hour growth-based assay, I conducted another round of experiment known as the selection experiment or evolution experiment, where I took the same wastewater sample, the same wastewater influent sample, and then introduced them to a specific concentration of either the antibiotics or the antibiotic transformation product. And then for 7 days, every day, I treated the same concentration of the compound to the same concentration of compound to the microbial community, as it was done to mimic how they get introduced to all these compounds in the wastewater itself. And then once they were done, I took samples from day 0 and day 7, and then did DNA extractions, which was again followed by qPCR of NTI1 gene and 16S rRNA gene. NTI1 gene, as they are the marker of antimicrobial resistance, and 16S rRNA gene for the bacterial marker. So based on this experiment, I got these results. So I conducted these experiments on 3 different antibiotic classes: fluoroquinolones, sulfonamides, and macrolides and lincosamides. So fluoroquinolones and sulfonamides, because they are regularly detected in wastewater, and so are macrolides. Increasingly, they're being detected in wastewater. So we can see that as compared to, so the first one is control, which showed no increase in the anti-1 prevalence, the abundance of anti-1. But when we look at ciprofloxacin and desethylene ciprofloxacin, or moxifloxacin and sulfate, and then norfloxacin, desethylene norfloxacin, and ofloxacin and desmethylene ofloxacin, we could see that all of the transformation products had some biological activity in them where they were able to, where the anti-1 prevalence was higher than that of the control. So the control was day 0, and then the green ones are day 7. So we can see that except for moxifloxacin and sulfate, the 3 other transformation products had somewhat comparable anti-1 prevalence after 7 days of evolution experiment. Again, the same data, but I tried to present it in a different way to see if there was any significant difference between the abundance of the anti-1. We saw that only in moxifloxacin and its pair, moxifloxacin and sulfate, there was a significant difference between parent and the transformation product. Otherwise, if we look at the other 3 pairs, we can see that even the transformation products were able to, even the transformation products had higher anti-1 prevalence. So even when we compare, even if we put all of them in a single bracket and compare them individually, we can see that the transformation product of ofloxacin, which is desmethylene ofloxacin, had higher anti-1 prevalence, higher anti-1 prevalence not only than its own parent compound, but also higher than other fluoroquinolone antibiotics like norfloxacin and moxifloxacin. So again, I did a similar experiment on sulfonamides as well, where I took 6 different pairs of sulfonamides and their transformation products. And again, similar results where most of the transformation products had showed some anti-1 prevalence after 7 days of repeated introduction of the transformation product and its compound. And when we look at how different those anti-1 prevalence were, we can see that none of them had any significant difference. So it was quite comparable. And even in sulfonamide's case, a few of the transformation products actually had higher anti-1 prevalence when compared with other parent compounds of the same antibiotic class. And again, same thing with macrolides and gosamides as well. I had 5 different pairs of antibiotic and their transformation products from this antibiotic class, and we can see that all of them had anti-1 prevalence comparable to their parent compounds. And we can see that there were the anti-1 prevalence when we look at the ones for controls had none. And when we look at the significant difference and try and pool them together as normal compounds, we can see that some of the transformation products like roxithromycin's transformation product and clindamycin's transformation product had higher anti-1 prevalence than other macrolide antibiotics. And the other thing that we have to understand here is that the clindamycin's transformation product and roxithromycin's transformation products, they have higher anti-1 prevalence. And if we go back to sorry, I'm just going. And if we go back to the data of how we detected these antibiotics and transformation products, we can see that these transformation products are they do not just have higher anti-1 prevalence, they're also quite persistent as compared to their parent compounds. So based on the experiments that I did, based on these preliminary experiments that I did, I came to a conclusion that all of the transformation products that I used for my experiment, that I used for my project, I used 15 different pairs. So all 15 transformation products showed some ability to increase the anti-1 prevalence, which again is an indication that they have a potential for antimicrobial resistance selection. And in most cases, these transformation products, like we just saw, they had either comparable or higher enrichment than their parents, which again brings us back to the same thing again, which is that transformation products should not be ignored when we talk about antimicrobial resistance, because from the results that we just saw, we now can say that degradation always doesn't mean that the compounds lose their biological activity. And that brings me to the end of my presentation. And I'd like to thank all of my supervisors at both the university for their constant support, the UQ Exeter Institute, who've been supporting my PhD, the AMR group at both the universities, and also ONE Bridge Health for giving me this opportunity to present some of my work here today. And I'm happy to take questions. Thank you so much.
Thank you so much, Puya, for your presentation. Your team is so interesting. We have the results so interesting. We have one question. Any question on the auditorium, online? No? Yes?
Hey, Pooja, Uli.
Yeah,
like
hi.
your talk.
Thank you.
I have a question regarding where do you think selection is working in these cases? Do you believe it's working on the entity of resistance gene cassette, the entire organism? Why do you think these results are happening, especially in your evolution experiment?
Thanks, Uli, for the question. I think because I'm still running other experiments, I can't really be 100% sure where the resistance is happening, but I'm thinking it's the overall organism where the resistance is taking place, because we haven't really looked into details. We've just been conducting these evolution experiments based on the microbial community itself, but it would really be interesting to look into where the resistance is happening, like you mentioned specifically in future, and I think that would be a really good project in itself.
Okay, thank you so much. Thank you,
Thank you.
again, Puya. And now we start with the next speaker. She's Antonia Mataraga. She's postdoctoral research in animal science from Department of Animal Science, Agriculture, University of Athens, Greece. And she presents an overlooked risk, the role of Mycobacterium avium subspecies paratuberculosis in antimicrobial resistance across the One Health spectrum. Okay, 1 second. Okay, let's just start.
Not open. So, hello again. At this time, we will discuss about MAP, Mycobacterium avium subspecies paratuberculosis, and its role in antimicrobial resistance across the One Health spectrum. So, why MAP is worth our attention? It is the best-known agent of Jones disease. It's a chronic granulomatous enteritis in ruminants. It causes substantial economic losses to the global livestock industry due to the reduced milk yield, the premature culling, and the lower slaughter value. The infection usually occurs in young animals through the fecal-oral route, typically by contaminated milk, colostrum, or the environment. After a long incubation period, the infected animals may progress to clinical disease marked by persistent diarrhea, weight loss, and eventually lead to death. However, over the past two decades, MAP has also been implicated controversially many times in several chronic human diseases, like, and most notably, the Crohn's disease. MAP DNA, antigens, and even viable organisms have been detected in human tissues, in human blood, in breast milk, across multiple studies. In addition, MAP is recovered from more frequently from the intestines of individuals with Crohn's disease than from patients with ulcerative colitis or from people with inflammatory bowel disease. Additionally, adding to these concerns about Crohn's disease, MAP potential has also been indicated and has been associated with a growing list of conditions, including Bloch syndrome, type 1 diabetes, Hashimoto's thyroiditis, and multiple sclerosis. Despite these findings, MAP's AMR profile has been received very little investigation. There is no standardized susceptibility testing protocols. The organism's slow nature and growth and its environmental resilience make the AMR characterization particularly challenging. So we see that perhaps emerging evidence suggests that MAP may represent a silent but a long-term concern that spans all three One Health domains. So why MAP is overlooked? The status of AMR MAP is critically overlooked for not just one reason. The incorporation of the treatment of John's disease in animals is generally not undertaken due to its cost, the chronic nature of the infection, the concerns about generating antimicrobial resistant pathogens, leading to a primary focus on test and cull control strategies for John's disease. When I say that it has a chronic nature of it has a cost and chronic nature of the infection, I mean that it takes up to 2 years to manifest in young animals, so it may be under recognized as it keeps growing in the animal. There is little clinical driver for routine AST of MAP isolates in veterinary diagnostics. The incorporation of supplements like the Mycobactin J for the in vitro growth of MAP it is essential. Also, the use of decontamination protocols and antibiotics like the amphotericin B, naxalidic acid, etc., vancomycin also is necessary to control the contamination from other microbes in vitro culturation. Because the samples are primarily feces and milk. Also, automated systems that exist for MAP culture, like the BACTEC 460 or the MGIT 960, have been adapted for MAP, but due to its delayed growth that may take up to months to grow in vitro, and the differences and difficulties of the medium, it makes all the laboratory work very time-consuming and difficult. The determination of antimicrobial susceptibility in MAPS, therefore, for the reasons that I already discussed, are challenging due to its slow growth and fastidious nature, and the lack of standardized methods and clinically validated MIC breakpoints for MAP further complicate this interpretation of resistance data. But, okay, it is overlooked, but why is it relevant? Despite these challenges, it is highly relevant because the investigation of antimicrobial therapies for Crohn's disease in humans means that human population have been exposed to antibiotics like clarithromycin and rifabutin, creating a potential pressure for resistant MAP strains in the environment or within human hosts. MAP's intrinsic resistance to many drugs is also significant, and it's a significant barrier to treatment success, as it possesses a robust, that we will see later on, lipid-rich cell wall that acts as a permeability barrier, and it can enter a dormant, non-replicated state in vivo, during which it is phenotypically tolerant to many bactericidal drugs. Studies have also demonstrated that MAP can survive intracellularly within macrophages, an environment that can further reduce the antibiotic efficiency. Therefore, even without the acquisition of classical resistance genes, MAP's innate biology confers significant resistant properties that complicated efforts for control and human contexts. So let's frame it with our discussion within the One Health perspective. MAP circulates across animals, environment, food chain, and the human population. It is shed in massive numbers in manure from infected animals, and it persists for 1 to 2 years in the environment, for long periods in the environment, in soil and in water. It is found in raw and pasteurized milk, in cheese, in meat, and it has been detected in municipal water supplies and sewage systems. So when an organism moves this freely across ecological boundaries, and when it possesses the biological capacity for both intrinsic and acquired resistance, it becomes somehow essential to evaluate its AMR potential more systematically. So a brief view on the resistance susceptibility patterns. The in vitro susceptibility testing has systematically characterized the phenotypic antimicrobial resistance profile of MAP, revealing a pattern of broad intrinsic resistance to many common antibiotics, alongside variable susceptibility to a more limited set of agents. So MAP exhibits significant resistance. First line anti-tuberculosis drugs. So studies show high mix from isoniazid and ethambutol, rendering them largely ineffective in vitro. Similarly, MAP displays robust resistance to the cyclic peptide capreomycin and the fluoroquinolone ciprofloxacin. In contrast, MAP isolates often show susceptibility to macrolides, particularly clarithromycin, which is a cornerstone of therapy for other members of the Mycobacterium avium complex. The aminoglycoside amikacin is another key drug to which MAP frequently demonstrates in vitro susceptibility, making them a critical component for therapeutic regimens. Also, the rifamycins show a differential activity against MAP, and while MAP is often resistant to rifampicin, it is typically more susceptible to its analog rifabutin, leading to the incorporation of rifabutin into several anti-MAP treatment protocols for Crohn's disease. The efficacy of antimicrobials can be modulated by the interactions with each other and with host-directed therapies. Studies have shown that the interaction between 6-mercaptopurine and certain antibacterial drugs can be antagonistic, reducing the efficacy of the antimycobacterial agent. And conversely, some combinations like the triple antibiotic combination RHB-104, clarithromycin, rifabutin, and clofazimine have been shown to be bactericidal in culture, informing clinical trials designed for Crohn's disease. Thiopurines like the 6-mercaptopurine and azathioprine only inhibit MAP at high supraphysiological concentrations in vitro. And the 5-aminosalicic acid and sulfapyridine have minimal direct inhibitory effects on MAP. Suggests that the clinical benefit of these drugs in Crohn's disease is primarily immunomodulatory, and that the MAP may persist in patients on these therapies. Furthermore, common immunosuppressants like cyclosporin A have no effect on MAP growth, while rapamycin and tacrolimus show only modest activity, indicating that MAP can withstand a suppressed host immune response. So, the AMR exhibited by MAP is not a singular phenomenon, but a complex multi-layer defense system. It integrates intrinsic non-mutational barriers with acquired genetic mutations, allowing this bacterium to survive in hostile environments, including those created by the antimicrobial therapy. The molecular elucidation of these mechanisms draw from direct studies on MAP and from well-conserved pathways in related Mycobacteria species. So, just a few words about the unique cell envelope, the outer mycobacterial membrane that is composed of mucolic acids, and it's covalently linked to arabinogalactan, which is in turn attached to a peptidoglycan layer. This mycobacterial cell envelope is its first and most significant line of defense, creating an hydrophobic barrier with extremely low fluidity that restricts the penetration of hydrophilic molecules, including a wide range of antimicrobials. Active efflux is a critical complementary mechanism to the passive barrier of the cell wall, and transporters that export toxic compounds, including antimicrobials, from the cytoplasm are ubiquitous in Mycobacteria. Also, MAP can enter a state of profound metabolic quiescence, which confers phenotypic tolerance to most conventional antibiotics. Under prolonged nutrient starvation, map cultures develop a significant population of cells with a spore-like morphotype, characterized by increased resistance to heat and osmotic stress. This dormant state is highly relevant in vivo, where MAP resides within macrophages in granulomatous sessions, often in low oxygen environment. And this anaerobic model also demonstrates that the same drugs may be bactericidal against replicating MAP and are merely bacteriostatic or ineffective against non-replicating persistent populations, mirroring a key challenge in treating chronic non-M tuberculosis infections. Also, when MAP is extremely adapted to these anaerobic conditions, it exhibits a dramatic and significant increase in tolerance to a wide range and spectrum of drugs, including rifampicin, clarithromycin, and amikacin, compared to the aerobically grown cultures. Am I in time? Oh, okay. I will move more. Finally, the bacterial response to stress is coordinated by complex regulatory networks like the SOS response that modulate gene expression to enhance the survival under antimicrobial pressure. Also, as I said before, beyond the individual cell mechanisms, MAP can form organized biofilms, which are communities of bacteria encased in an extracellular matrix. Bacteria with biofilms exhibit significantly enhanced and multifactorial resistance to antimicrobial agents, involving reduced antimicrobial penetration, altered metabolic states, and the expression of specific resistant genes. Finally, MAP can survive free-living amoebae that act as an environmental training grounds, selecting for bacteria that can resist phagocytic killing, oxidative stress, and disinfectants. I will move very fast because of the time pressure. Excuse me for that. So mutations in the RPOB, which encodes a beta subunit of RNA polymerase, have been identified in MAP isolated from Crohn's disease patients who failed the rifampicin therapy and fall within the rifampicin resistance determining region, similar to the Mycobacterium tuberculosis. Macrolide resistance, mediated by point mutations of the 23S RNA gene, the RRL, is well characterized in MAC, Mycobacterium avium species, and while less frequently documented in MAP, the mechanism is highly conserved and plausible. Also, aminoglycoside resistance can arise through mutations in the RRS gene, and variants in the EMBB associated with ethambutol resistance have been noted in MAP and genome as well. I will move forward with I have prepared some comparisons for the Mycobacterium tuberculosis and MAP, but I will move a little bit forward, and also with MAC. Excuse me for that. So as I said before, sources and drivers of MAP, we can find, although the antimicrobials are rarely used specifically to treat Jones disease, they are widely used through livestock production for other infections, creating this selective pressure in the farm environment. Also, this selective pressure shape the surrounding microbiome and environmental gene pool, and indirectly selecting for MAP variants with enhanced stress resistance or the efflux capacity. Also, we see here that it has been found in many studies viable MAP repeatedly in raw milk, in retail pasteurized milk, in powdered infant formula, in cheese, in meat. Also, MAP can protect itself and persist through during thermal processing. And also the survival of MAP in meat products, particularly from advanced disseminated infections, where the organism can disseminate to muscle and the organ tissues, presents another less studied exposure route. Also, MAP can survive, as I said before, in soil, water, and manure for over 1 to 2 years, has been detected in river watermen, catchment areas, from sewage treatment works, indicated its widespread environmental dissemination. Moving forward, the clinical use of anti-MAP therapy in Crohn's disease creates another selective pressure for resistance analogous to the well-documented emergence of macrolide resistance in MAC lung disease patients during prolonged or monotherapy regimens. The detection of RPOB mutations in MAP isolates from Crohn's disease patients who had been treated with rifabutin provides direct evidence that its selection is occurring in the clinical setting. And finally, serological evidence show that MAP exposure is widespread in human population and that immune responses to MAP antigens can be influenced by therapies used from inflammatory bowel disease, suggesting a complex host pathogen and drug interaction that could influence MAP persistence and resilience development. Finally, I will go to the last to conclude with all I saw and all I didn't show, MAP's antimicrobial resistance potential represents a quiet but perhaps significant One Health issue. It links, and MAP doesn't cause dramatic resistance outbreaks, accumulates tolerance and resistance slowly across the environment, the food, the clinical spaces. And this is why MAP could potentially be a hidden AMR threat, silent but steadily evolving in time. Thank you very much.
Thank you so much, Antonia. Okay, we have a little just the time, okay, and in this part we pass with following with the next speaker, okay. The next speaker is Marios Balmas. He is a PhD candidate for Department of Biochemics and Biotechnology, University of Thessaloniki, Greece, and he presents the impact of manure fertilization on soil, fate of manure introduced, and ingenious taxa engine. Okay. Okay, we can start. Perfect. Okay.
So, hello, I'm Marios, and we're going to go through the journey of manure fertilization and its impact on soils. So, sorry, does not change. Okay, thank you. So, it is pretty clear that the antibiotic misuse and overuse has led to the emergence of pathogenic antibiotic-resistant bacteria and antibiotic-resistant genes, and in their dissipation in the environment. One hotspot for these are livestock farms. And possible routes of dissemination is the application of manure in agricultural lands, where ARBs and ARDs can disseminate, can be uptaken by crops, and then go back into humans. In a previous study, it was observed that soils have different responses to manure application, but there was something common, that manure taxa disappeared from soil up to 25 days. For that reason, in this work, we wanted to track and model the behavior of the introduced taxa and the indigenous taxa, but also ARGs and mobile genetic elements in soil after manure coalescence. And so we studied the taxonomic profile of dissipating or persisting introduced taxa, the indigenous taxa. We also studied ARGs and mobile genetic elements in this process. And we also wanted to study the genetic context of these. For that reason, we applied manure with or without antibiotics, which had been incubated for 48 hours at 37 degrees, so we can have priming of the microorganisms and more realistic conditions. Alongside our control, which was water with or without the antibiotics. And we incubated at 25 degrees for 5 different time points. We moved on to extraction. So, and we did amplicon sequencing, shotgun sequencing on some key samples, integron sequencing for amplified with the Oxford nanopore, and qPCR on some specific genes. So, beginning, we first observed that manure had a strong effect upon its application on soil taxa. As it can be seen, these orange, brownish, yellowish is the manure treatments, which are distinct from the control treatments. But we also observed that there was a convergence over time. Besides that, we also saw that the antibiotic application had some effect upon these treatments. So, we first wanted to see what happens to the manure taxa that do not belong in the soil. And we identified some distinct classes of some different taxa. And as you can see, some taxa seem to be increasing after half a day and disappearing, some being more persistent in the beginning up until 4 days, some disappearing quickly, or some being there for 1 day and then disappearing. So, for not tiring you with heat maps and heat maps, we focused on some main clusters. First of all, cluster 1. Why? It contained some genera with some opportunistic pathogens like Clostridium. And we wanted to model the behavior of Clostridium, and that's why we applied exponential decay models. What we see is that Clostridium is a taxon that is pretty persistent over time. And we also see that the antibiotic application, the colored lines, had some effect on its decay rate as it increased DD50. Similarly, Terisporobacter, which has seen to cause infections in surgical cases, seems to be pretty persistent. Moving on to cluster 2, which had the most taxa, we see that in this cluster, the most abundant taxa are Corynebacterium and Streptococcus, also genera with opportunistic pathogens. But they seem to disappear quicker than taxa from cluster 1. But again, we can see that the antibiotic application may have some effect on the decay rate of this taxa. And moving on, we see cluster 3, which contains Pseudomonas, but these genera disappear really quickly from the soil. I also show you what happens to the soil taxa upon manure application, because we see some cases where you see taxa being increased up to 25 days. Sorry. Interestingly, in cluster 7, we see that there is streptomyces, taxa that produce antibiotics, and they also contain resistance genes. And as we see from their pattern, they seem to increase at 4 days, and then remaining stable up to 25 days. Same for nocardioides, which was the most abundant, but it's just taxa that degrade pollutants. Sorry, what happens? Okay, sorry. So, from our metagenomic data, we wanted to see if there are potentially opportunistic pathogens. And that's why we used a database that contains potentially opportunistic pathogens to see how long they persist in soil. What we observed is that there are groups like lactobacilli which disappear quickly, groups like mycobacteria that they persist a little bit longer, and again we see Clostridia which remained for 25 days. On the other hand, we see that there are also some soil taxa which there aren't in manure, which rise at 25 days, and in that case, we again had streptomyces or streptomycota. Sorry, streptomycota. Moving on, we wanted to check out what happens with ARGs. So first, with qPCR, we tested some specific ARGs, like TETQ, DFRA, and the RMF, that they really didn't exist in the soil. And we see that they follow an exponential decay model, disappearing from the soil. And again, we see that the antibiotic application has an effect on the decay rate as the DD50 values increase a little bit. On the other hand, SUL1 seems pretty persistent because the model could not fit. And we see that it rises, it peaks at 4 days, and then it decreases again, but still remains at 25 days. We used then again our metagenomic data to find ARG locus structural variants, and we identified many combinational clusters at manure structural variants that they persisted in soil for more or less for different time points. I'm sorry. So we first see the example of the cluster of the TED-Z ARGs, which goes with MIRA, which also is an ARG, and we see also some mobile elements which have to do with antimicrobial resistance mobility, and some other have to do with activity loss of some genes. We then see the paradigm of TU-Fab, a modified target that is not recognized by antibiotics. I'm sorry, that it's not targeted by antibiotics, being coexistent with TET genes and ribosomal subunits, so we can speculate that the protein synthesis mechanism is protected. We also have the example of vancomycin genes, which coexist, and they coexist with the machinery of lipid metabolism or modification in synthesis enzymes, which is the main route of protection from vancomycins. And this cluster could remain up to 25 days. And then we see the example of CMXA, which coexists with other ARGs, but also we see that there's a really nice structure of Kaki, Delta-1, and SUL1, which usually exists in integrons, the mobile elements. Interestingly, we saw some soil ARGs also being stimulated, but we had much less combination, much poorer clusters. For example, we see the CSA gene, which is cephalosporin resistance, sorry, it shouldn't say defense, coexisting with some mobile elements that have to do with AMR mobility and biofilm formation. And we see also that there is a cluster of Van Aert gene, which is a transcriptional regulator, which also confers resistance to vancomycins. We also wanted to study what happens with integron integrase 1 gene, which is connected to anthropogenic pollution. And we see that even in soil that's been treated with manure or even in controlled soil, the INTI1 gene always increases over time. So we wanted to see what really happens. So we amplified class 1 integrons and we performed Oxford nanopore sequencing so we can see what these integrons really carry. In the case of manure, we see that integrons pass into the soil, but they disappear over time. But these integrons carry genes that have to do with antimicrobial resistance. On the other hand, we also see soil integrons, which either remain stable over time, and they might carry some basic metabolic genes, or we also see integrons that they are boosted over time, they're increasing, but they're mostly empty or incomplete. We also moved into plasmids from our metagenomic data. We saw some 8 distinct relative abundance-based clusters, and one of these, the cluster 1, was purely manure-related as it exists in manure and not in soil. So, for that reason, we're going to also cluster the plasmids into its relative abundance clusters to see which they are. And we see that there is a plasmid, the AA379, which is abundant in every case, in soil, in manure, or in the combinations, in all time points. We see it being passed into the soil from the manure, but disappearing, also increasing when it's coming from soil in some cases, or remain stable. And of course, we also wanted to see which ARGs are being carried from these plasmids. And we saw that higher abundances of ARG carrying plasmids exist in manure, and they are manure-specific plasmid also, but these manure-specific ARG carriers pass into the soil, but they disappear over time. On the other hand, we see that the manure application might also affect or impact the soil ARG composition on plasmids by boosting them. And on the right side, you can see the different antibiotics which the plasmids bring the resistance for them. And we see that some of them are common in both manure and soil. But manure has also some extra resistance. And of course, we're going to check what happens with plasmid host specificity, where higher relative abundances and increased host specificity existed in manure plasmids. As we can see, we can reach up to genus level, which did not happen for soil plasmids. We again see that plasmids from manure pass into the soil, but they disappear. But some soil plasmids again appear while they have really low abundances, they appear like being boosted. So, to reach up to a conclusion, for the manure taxa of interest, we saw 3 main trends: quick dissipators, slow dissipators, and persisters. We also saw that the ARG variants and the INTI1 in manure samples are more rich structured than in soil samples. But that maybe has to do with technical reasons, because soil is a much diverse it has much more diversity, or these might have functional roles in soil, or even have to do with soil ecology. Maybe they are an energetic burden that cannot stand much more diversity. We also can say that quantities on their own, as we saw for the INTI1 gene, are not necessarily sufficient because we see INTI1 being increased, but soil taxa, soil INTI1 integron 1s do not bring resistance genes. We also saw higher abundance of ARGs and different host ranges, but more specific in manure samples again compared to the soil ones, for the same reasons as the ARG structures or INTI1, technical reasons, they might have a functional role in soil, or again, with soil ecology, maybe having too many extra genes, an extra carrier is an energetic burden. We also saw that there are manure ARG associations with antibiotics that are commonly used in farms. Maybe there is a role of the soil-enriched taxa in ARG dissemination and resistance, as we saw with Streptomyces, which are very known to produce antibiotics, so they make this pressure even worse. And of course, the dramatic reduction, the overall dramatic reduction, does not necessarily mean that there is risk dissipation. And I don't want to tire you anymore. I want to thank all the collaborators of the project, especially MPI Sotos Vasiliadis, the organizing committee for this opportunity, the HFRI for the scholarship I've been awarded, the friends and colleagues in the Laboratory of Plant Environment and Biotechnology. And of course, I'd like to invite you in the Ecotoxicomic Conference, which one of the topics will be antimicrobial resistance next year in October in Thessaloniki, Greece. And of course, it's the IDAR conference in Brisbane next year, August, in Australia. And thank you very much for your attention.
Thank you so much, Mario, for your presentation. It's an amazing theme for discuss. But with the time is necessary to following with the next speaker. But thank you so much. It's one, okay, next, go ahead. The Magali de la Cruz Barron. She's research from Department of Hydrobiology of Technical University of Dresden in Germany. And she's talking with a team, a One Health approach to quantify the mobility potential of antibiotic resistant genes by a linkage-based ddPCR. Okay? Okay, ready? Perfect. Yes. Okay, we can begin.
Can you hear me? Yes. Thank you very much for the introduction. And as it was mentioned, today I will present our method for the use of using ddPCR as quantification for the mobility potential of antibiotic-resistant genes. Since yesterday, we have heard a lot about the threat that antimicrobial resistance, AMR, represent for human health. And unfortunately, this is forecast to increase over the coming years. However, AMR impacts goes beyond the human health. It affects animals and the environmental health, and a disruption in any of those domains will have consequences in all of them. Because of that reason, several actions are taking place, beginning with looking for alternative therapies. We heard about them yesterday, the implementation of stricter regulations and update of the current ones, and increasing the public awareness, and also expanding surveillance programs. However, here, even if the surveillance programs are more within the One Health framework, a challenge remains. And this is because even the One Health domains are so well interconnected, the surveillance priorities often differ. And also yesterday we heard that the main reason is because even if they share common drivers, for example, in the human and veterinary, the focus is in the ARGs that are linked to pathogens. And this is obvious because there is an immediate risk for treatment failure. However, in the environment, the surveillance should be more focused on the antibiotic-resistant gene mobility and its association with mobile genetic elements, because the environment can act as a reservoir where the ARGs can persist for longer, and most of the time in non-pathogenic bacteria before they become an emerging threat for human or animal health. And this is why we need to move forward, and we need to find a method or a surveillance that must shift from asking, is the antibiotic-resistant gene present, to a most critical question. Is it this antibiotic-resistant gene mobile and likely to be spread? So here is where our work comes, and we proposed a method that allows to quantify the mobility potential of antibiotic-resistant genes. Our method is based on a droplet digital PCR, ddPCR, and here is a simplified overview of how the method works. Starting with the sample preparation, then we have a partitioning where the sample is divided in thousands of droplets where that PCR will take place. And this is important to note that this distribution is randomly. It means that some of the droplets will remain empty, others will contain single DNA molecules with or without the target of interest, and also it will have very few of them that might contain multiple DNA molecules, as well with or without our targets. After amplification and detection, we can quantify the absolute abundances of our targets based on a Poisson distribution. However, we use the ddPCR in a different way. So for this, we used two targets or using different fluorescent markers. And because we want to know if the targets are physically linked. And we can use the mathematical equations so we can distinguish if there is a true linkage, meaning that they are really in close proximity, or this is due by chance. And that means that a higher linkage between an antibiotic-resistant gene and a mobile genetic element, there is a stronger association, and therefore it's more likely to be transferred between bacteria. To test this, we first need to work with known samples. And here we used linked and unlinked DNA molecules. For the linked DNA molecules, we used a small plasmid vector where our targets, here we use the integron integrase gene as a mobile genetic element, and the sulfonamide resistant gene. So here our targets are really close to each other. And as expected, here represented as the probability of linkage, most of our targets were co-detected, meaning that they were really close to each other, as we already knew. And very few of them were detected as unlinked. And when we analyzed the single DNA fragments and a mixture of them, most of the signals favor the presence of unlinked targets, and very few of them were co-detected. However, after the mathematical evaluation of those results, those appear only co-detected due to chance, not because they were really physically linked. Since the method was correlated well, what we expected compared to what we were having in theory, experimentally and theoretically were correlated, so we decided to move forward and to apply our method to more complex samples. And here we used as an input the DNA from influent and effluent wastewater samples. Here are the results for the influent and the effluent. And overall, we only observe a linkage percentage of around 20% for both type of samples. And even if the detection favors the unlinked target, this lack of association does not necessarily mean that the sulfonamide resistant gene is not mobile or could not be mobile. And here there are two possible options. One, the antibiotic resistant gene in question is not associated with the mobile genetic elements that we were analyzing. And the second one, it's because we have a limitation of our method. So we want to know if it was the case. So then we decide to move to a natural occurring plasmid. Here we decide to use the broad host range PB10 plasmid. And we decide to analyze different antibiotic resistant genes present on this plasmid. So we choose 4 different antibiotic resistant genes and 2 mobile genetic elements. Again, the integron integrase and the CORB as a plasmid marker. Here is a summary of the different combination between ARG or ARG mobile genetic element, and here the minimum distance between the targets. It's important to know that even the shortest distance is already 4.5 bigger than in our proof of concept. And before running any experiment, we decide to run some simulations to have an idea of what to expect. And first, we simulate the fragmentation of our PB10 plasmid. So in a mean length of 5,000 base pair. And then we used the fragments generate to run in silico ddPCR. And as we were expecting, there is a clear trend where as larger the distance between the targets, the probability of detecting them as a linked targets also decreased. Important to note is, like, for example, already for 20,000, for a distance around 2,000, we have a linkage percentage less than the 10%. So we now decide to test the real samples. And for that, we decide to use the circular plasmid, but also a linearized plasmid. And the results were a little bit surprising because, as expected, so for targets that are in close proximity, we have a higher linkage, but it only happened for the circular PB10 that the linkage percentage remained quite high, around 70%, even for distance between 20 and 30 base pair. If you remember, I just mentioned that in the simulations, this was around less than 10%. But it was not the same as for the linearized plasmid, where we saw a drop in the linkage percentage by around 30% for the same genomic distances. So what does this mean? So the method is accurate enough even for longer distance, but DNA fragmentation matters, and we need to consider that if we want to apply this. However, for clinical relevant antibiotic-resistant genes and mobile genetic elements, either for surveillance in clinics or in the environment, those pairs most of the time are quite close to each other. And therefore, the method remains sensitive and accurate and informative. And with that, I come to my last slide. So the linkage ddPCR-based method is a promising for quantify the antibiotic-resistant gene mobility. It can be straightforward and can be applied to whatever combination that we have in mind, and not only related to environmental samples, but also it's part of the JPI AMR Texas project that we want to target multi-drug resistant bacteria using this method. Also, the linkage ddPCR method could enhance our understanding of the spread of the antimicrobial resistance. And definitely, antibiotic-resistant gene mobility is an important risk indicator that should be included in environmental AMR surveillance. And with that, I thank you for your attention, and I'm happy to answer any questions.
Thank you so much, Magali, for your presentation. Any question on the auditorium, online? No? Okay, thank you so much, Magali. The next speaker is Eda Denise Ardam. She is research from the Institute of Hydrobiology of the Technical University of Dresden in Germany. And she presents the fragile innovation AZT suppresses plasmid transfer, but is easily undetermined by resistant and metabolic risk.
Did I turn it on? Good.
Okay, move your microphone exactly down there.
It's not height adjusted. Sorry. Okay. Thank you for the introduction. I'm Eda. I'm a PhD student in TU Dresden, and today I wanted to talk about a project where we evaluated a drug called AZT for its possible use as a conjugation blocker. So today we talked a lot about AMR and how it spread, and how it spread is a huge problem, and horizontal gene transfer is a major driver of that. And you can see, you can see in the photo, there is 4 routes of horizontal gene transfer can take to spread across, to spread AMR across clinical and environmental settings. But conjugation is the main mechanism of horizontal gene transfer. And what happens in conjugation is a donor bacteria that has a plasmid can make physical contact with another bacteria. And then during this physical contact, the plasmid gets replicated and then gets added to the other to recipient bacteria. So as a result, if you have an ARG in your conjugation, in your plasmid, which 39% of all ARGs are located in plasmids, now, so you had 1 resistant bacteria, now you have 2 resistant bacteria. This is why there has recently been a search for conjugation blockers. And when we say conjugation blockers, we mean compounds that can limit conjugation, limits this process. So, yeah. This brings us to AZT. AZT first came to public consciousness during the HIV epidemic in the 80s, 90s. It was the first drug that was approved for antiretroviral therapy. And what AZT is a nucleoside analog. So its structure is a synthetic pharmaceutical. Its structure is pretty much exactly the same as thymidine. So when it is uptaken in the cell, it mimics thymidine. And it's able to, it mimics thymidine, and what it does is that it gets added to the DNA strain, like thymidine, which is able to do that because, it has this hydroxy group, so it can get added, but it lacks the second hydroxy group, so a new nucleotide cannot be added to the DNA chain, meaning the DNA replication is stopped, is halted, and this eventually leads to cell death. One important thing about AZT, that it's a prodrug, meaning that it's in innate form, it's inactive. It needs to be activated inside the cell by an enzyme called thymidine kinase. And what thymidine kinase normally does is that it takes this thing doesn't like me. It takes this thymidine and then it adds a phosphate group, making thymidine monophosphate. And then after, I think, 2 more phosphorylations, this can add it to the DNA elongation. Thymidine kinase does the same thing for AZT. Takes azidothymidine, makes it azidothymidine phosphate, makes it able to get added to that elongated DNA chain. The problem is, thymidine kinase, any point mutation in thymidine kinase can lead to AZT resistance, and this is rapid because, like I said, it's a point mutation. So this has been what I described as the mechanism of AZT for antiretroviral therapy, but it turned out that this mechanism is also effective against gram-negative bacteria. And people were quite excited when this was first found, because this is an already approved drug. So it can be used as an antibiotic, antimicrobial. Then we won't need to do another drug discovery, another approval process. It can just be repurposed as an antibiotic. So there was some clinical trials in 2018 against Klebsiella pneumonia and E. coli, and they were successful. And then another property of AZT was found, which is an antiplasmid effect. And that means it can maybe be used as a conjugation inhibitor. In fact, there was a previous study that showed that AZT leads to reduction in plasmid transfer by 80%. So our research question in this project was like, is this function reliable? So can AZT reliably function as a plasmid transfer inhibitor, especially considering the resistance? If this resistance develops in one or both conjugation partners, is this sufficient to eliminate the anti-transfer effect? Uli discussed a little bit about these strains in the morning, but I'll mention them briefly also. So we have a donor. A donor is donor bacteria and a recipient bacteria. These are both E. coli MG1665, but these are both, of course, susceptible to AZT. So we needed some AZT-resistant bacteria. We did spontaneous mutation assay on the TK gene, and then we managed to isolate some AZT-resistant donor and recipient strains. And to check if we did manage to get resistance, we did the MICs, and we found that for the susceptible strains, MIC is 8 micrograms per ml, but for the resistant strains, MIC jumps to 125 microgram per ml. So we did have resistant strains. Good for us. So then we could move on with the mating assay. So what we do when we do a mating assay is we have a donor and who is tagged with an M cherry and a kanamycin resistant, like an ARG that confers kanamycin resistant on the chromosome, and it also has a plasmid that confers resistance to tetracycline and also has a GFP. But as long as this plasmid is inside this donor, GFP production is suppressed, so this the donor will always remain bright red under UV light. For resistant, we only have a gentamicin resistance. It's chromosomally tagged, and it doesn't shine anything under UV. So once the plasmid moves towards gets transferred to recipient, we end up having a transconjugant that is resistant to gentamicin, tetracycline, and also is bright green under UV lamp. So why am I telling you this? Is because after 24 hours of mating, we use this system to be able to count our donor, recipient, and transconjugant cells using selective plating. So, based on which strain is resistant to which antibiotic, and we also make use of the fluorescence, and then we get our, the numbers for each strain, the donor, recipient, and the transconjugant, and then we use a calculation of conjugation ratio where we divide the transconjugant numbers divided by recipient and then 100%. So for the rest of the presentation, when I say conjugation ratio, I mean this. So moving on to the results, the first thing we checked was that does AZT resistance development leads to bacteria losing on fitness. So for this, you have the donor strains on the right, and then you have the recipient strains on the left. And you can see that the resistant strains and the susceptible strains pretty much follow the same growth curve. So no resistant strains, there was no detectable fitness cost for the after development of AZT resistance. Ooh. Okay. So after that, we wanted to confirm the finding of the previous study with regards to reduction of plasma transfer rate. And so we did a mating assay between just 2 susceptible partners, and we found that AZT does reduce plasma transfer in a concentration-dependent manner. And we have a high conjugation, we have a high correlation row here. And we also found that this is a log scale on the x-axis, but that minus Curie version is actually 0.01 microgram per ml, and 0.001 microgram per ml. And you can see that it drops so much, it drops a lot. And if you remember, the MIC was 8 microgram per ml for susceptible strains. So this is a concentration much lower than MIC, but the effect is very much there. So from there, we started to play with our resistant strains. This part on the right, I already discussed this, the susceptible donors in the previous slide. And on the left, we have the 2 matings between resistant strains, a resistant donor and resistant recipient. And I should mention that A and B here refers to statistical significance between groups. Here we see that when the mating is done between 2 resistant partners, the inhibition effect is eliminated. So AZT does not lead to any suppression of plasma transfer. This result surprised us because when we have a susceptible when we have one susceptible mating partner and a resistant mating partner, regardless which one is the donor and recipient, and without AZT, conjugation frequency is reduced. So we didn't need to add AZT to it, it was already reduced. So what I mean is that I compared this to this, and it's very much lower. When we did add AZT to these matings, there was no additional suppression of transfer though, so still AZT effect is. And especially the initial part, the reduction of the conjugation frequency without AZT made us think that the mutations that leads to AZT resistance probably causes some metabolic instabilities during mating for these strains. So to test this, we made use of another compound called DTMP. And what DTMP is, it's essentially this thymidine monophosphate, and a deoxyribose sugar is attached to it. And because it is downstream from the thymidine kinase action, it is unaffected by the mutation. So its use in the cell has nothing to do with, is not affected by the TK mutations. So first thing we looked was those one susceptible, one recipient mating partners. And then DTMP was able to rescue the reduced transfer, which confirmed that yes, TK mutations that leads to AZT resistance do alter the metabolic mechanism during matings. When we looked at the resistant matings, this thing doesn't work. When we looked at the resistant matings, there was no effect of the DTMP, which is not surprising. But when we had both susceptible partners, DTMP had a similar effect to AZT, also reduced the plasma transmission rate, which was a bit surprising also, but what we think is happening here is a thymidine pool effect, where there is just way too much of one nutrient inside in the mating medium, and then there's just not enough of the other ones, and that creates some sort of metabolical confusion. So yeah. After this, we got these results. We also wanted to know a little bit about the AZT's mechanism of plasma inhibition. So because we talk about its antibacterial effect, antibacterial mechanism, but nothing is really known how it does inhibit plasmid transfer. So we thought maybe it's a similar mechanism. It somehow affects the plasmid replication during the mating because that's also DNA elongation. So to do that, we used this strain. It's a E. coli also. It's susceptible to AZT. It carries the INCP plasmid, similar to the plasmid we use in mating assays. And it has probes that can be tracked by qPCR on the chromosome and in the plasmid, which means we can track the fate of the plasmid and the E. coli separately. What we saw, and then we took the strain and then we grew it in 4 different conditions with just the media, with an antibiotic that is resistant to, with AZT, antibiotic, and AZT. And we saw no impact of AZT on plasmid prevalence in monoculture. So this is not the mechanism. That is all we know for now. So moving on to the conclusions, we can say that AZT can and does suppress plasmid transfer. However, development of resistance in even one mating partner rapidly eliminates this potential. But at the same time, when AZT resistance develops, it causes some metabolic instabilities in the mating partners. So it's a plus minus plus situation. And conjugation blockers could potentially contribute to limiting AMR spread across One Health settings. But when we're evaluating such compounds, we should look into the consequences of resistance development and metabolic dependencies, and not just the altered rates of conjugation. With that, I want to thank my PhD supervisors and Dr. Fang for helping me with the experiments, and the rest of the team to Dresden. These are our funding of our funding agencies and collaborators, and thank you for your attention. If there's any questions.
Thank you so much, Ida, for your presentation. We have for one leader question, where anyone have one?
Thank you very much for your talk. So I have a question, a broad one, because that's a very interesting approach to inhibit plasmid circulation.
Yeah, thank you.
Do you know any other mechanism who are tested in other laboratories who could target the same objectives?
There is one compound that I'm going to mispronounce. It's a fatty acid, short-chain fatty acid, 2HD, I think 2HDA, something. I forgot its name. But it's a short-chain fatty acid. They found that it's also a conjugation inhibitor. It looks promising, but they were also like the last paper that I read did find that they think it's ATPase related. So it's probably the protomotive force. I think it's affecting that one. It's possible that AZT is doing this also. I just haven't looked at that. So I would be interested in looking at it, but I think it could be. I just hadn't thought about that option at the time.
Yeah, and probably something less toxic than...
Yeah, AZT is very toxic, exactly. Yeah. So, and I don't know about resistance development for this other compound. I don't think it's that easy compared to AZT. It was just AZT. I saw the clinical trials at the time, and it was like the drug repurposing was a big deal. And it's not that... people seem to give up on it a little bit, I think, but at the time it was a big deal, and there was people talking about like, oh, we'll drug repurpose it. And even in the clinical trial, they used it with colistin. They saw big success. So we wanted to also explore its option as a conjugation inhibitor.
Thank you.
Okay, thank you so much. Thank you, Ida. Okay, the next speaker is Dayala Konali. She's a research from the Institute of Hydrobiology of the Technical University of Dresden in Germany. And she presents the theme of tile, a cycle of contamination and recovery, how combined sewer overflows increase antimicrobial resistance in our urban stream. Okay, perfect. With us. Perfect. Okay, we
Can you
can
hear me?
start.
Can you hear me? It's okay.
A little more near. Yeah.
I have a very whispery voice.
Okay, in this.
Okay,
it's
is it
good.
okay now?
In this
Am I good?
distance, yeah.
Okay. Thank you for the introduction. My name is Diala Kunyali. I'm a PhD student in the TU Dresden. And today I will talk to you about my project. And we were looking at how combined sewer overflows increase antimicrobial resistance in an urban stream. And we were studying an urban stream in the city of Dresden. But first of all, let's see what exactly are combined sewage. So it's a system, it's a sewage system where it can, there is a, hold on. It's a sewage system that can take sewage from domestic, commercial, and industrial sources, and it can also take up rainwater runoff from storm drains. So it mixes both of these, both of the sewage and the rainwater runoff in one pipe, and then it takes it to the wastewater treatment plant. So in light rain events or in moderate rain, this is all okay, it all works fine. However, in heavy rain events, and in floods, for example, the system can get overwhelmed. So this mixture of sewage and rainwater can get released into receiving water bodies like streams or rivers, or in the case of Greece, into the sea. And in fact, this is what one type of combined sewage looks like. This is what the inside of the pipes looks like. I was inside of it, and it doesn't smell good. And yeah, so again, yes. So what is actually the problem? This urban drainage system can impact the stream quality, especially during heavy rains. During the heavy rain events, we're gonna call them CSO events, CSO, combined sewage overflow. And these CSOs introduce pollutants from the sewage, like bacteria and antibiotic resistance genes, and they release them into surface water. And what we wanted to see in this study is how a stream that is receiving these CSOs, how it acts during the event and how it recovers from it, or if it ever recovers. And this is our study area. This is the administrative area of the city of Dresden. This is the Elbe River, our main river. And this is the stream that we are working on. It's the Lockwitzbach. I'm just gonna say the stream. And anyway, this is the area that we're working on. It's inside of the city of Dresden. As you can see, there are multiple combined sewage overflow pipes, and there are multiple stormwater runoff pipes. And we chose 2 locations in the stream to study and to sample water from. It's the MS4 and the MS6. The only difference between these 2 locations is that MS6 has a lower flow rate, and then MS4 has a higher flow rate, and it has that because all of the tributaries from the Lockwitzbach, they reach the MS4 and they increase the water there, and also all the drainage, and before it reaches the Elbe River, it has a higher flow rate. So this is the only difference between them. They both have influence from combined sewage, and they both have the same kind of, whatever. Anyway, so as I said, we were sampling, and we sampled from January till September, and we did seasonal sampling, and we sampled CSO events. So for the seasonal sampling, we started for 6 months. We did seasonal sampling every 2 weeks. We got water samples, and then we did targeted sampling during CSO events, and we sampled throughout the entire event. Like we got time resolved samples, and we were able to sample from 2 CSO events. And of course, we continued with the seasonal sampling, and we also did the seasonal sampling between the events. And what we did is, after we got our water samples, we filtered them, we extracted the DNA, and we did 16S sequencing to find out the community composition. And we did high throughput qPCR to quantify our ARGs in our samples. And here is what we found. So we'll start with the bacterial abundance. And if you can see, for example, let's start with MS6. If you can see, there is kind of a bacterial surge during the CSO events, which makes sense. It's kind of, there is a sewage runoff. There is sewage that is being released into the river. So the bacterial abundance kind of rises, and then it kind of normalizes. We will see later. However, in MS4, there is also bacterial surge, but there is these, it's not as significant as MS6, of course, and there is also these random events where there is, during the season, there is a higher abundance of bacteria during the season. We can say that CSOs drive massive increases in bacterial loads. Next, we looked at the community structure, and here we can compare the communities from CSOs and from the seasonal samples, like our baseline samples during the normal times, the dry weather times. And we can see that in MS6, there is a significant difference between the CSO, the communities in the CSO samples and the communities in the seasonal samples. However, we don't see that in MS4. And it kind of makes sense because in MS4, there is a lot more dilution happening. So the effect is a lot more reduced. Next up, we took our sequences and we compared them to a comprehensive list of bacterial pathogens. And we took the list from this paper. I'll leave it here if you want to look, if you want to search it or something. And so we calculated the number of pathogens that are found in each of our samples, and we found kind of the same thing. There is a surge of pathogens during CSOs, and it's significant in MS6, it's insignificant in MS4. So we can say there is enrichment of potential pathogens in MS6, and that is the monitoring station that is upstream. So the effects gets more diluted the more you go downstream. Next up, we looked at the ARG abundance, and we can say that there is, again, a surge of ARGs in both in MS6, and it's increased significantly, and there is an enrichment of ARGs from all of the antibiotic classes. So if you remember our monitoring stations, I talked about MS6 and MS4, and these are combined sewage stations. However, we wanted to see, is this pollution coming from rain runoff or the sewage? So we chose a monitoring station. We set up shop here, and then this is a stormwater monitoring station, and we sampled the water directly from the pipe. So this is just the rainwater runoff, no sewage, no combined sewage. And what we see in here is that, so if you look, this is the stormwater from MS5, and these two are the CSOs from MS4 and MS6. And we see that there is a significant difference between MS5 and the CSO and the CSOs. So we say that stormwater had significantly lower ARG abundance than CSO samples. And if we compare now the baseline samples, the seasonal samples, there is no significant difference between them and between the stormwater. By the way, this is ARGs. I'm talking about antibiotic resistance genes. Yeah, so the stormwater profile was indistinguishable from the river's baseline. And from this, we can conclude that CSOs are the definitive drivers of ARG contamination. It's not the stormwater. And to this overwhelming slide, so if you remember, I showed you all of the total ARG abundance, and in here I just put all of the genes that we studied, and it's every gene that we studied in both stations, and it's their profile throughout our study period, how it increases, how it decreases during the season, during the CSOs. And if now I can just remove the ones that are insignificant, the one that did not show any increase during CSOs, so there is no spike in the CSOs. we are left with this, which makes sense. In MS4, there wasn't a spike during CSOs of ARGs. There wasn't a spike of ARGs during CSOs. So we are left with this. And now if we look at the ones that had significant recovery, that means the ARGs that went back to pre-CSO baseline levels. And we have this. And as we can see, there is significant recovery in most of the ARGs in MS6. So most of the ARGs in MS6 went back to pre-CSO baseline levels. And in MS4, the ones that were significant, most of them went back to pre-CSO levels. And we can see kind of a similar trend in pathogens. And if we want to look at the recovery of the stream, in MS6, so this is the baseline levels, the pre-CSO baseline levels, the normal levels, and this is during CSOs. So we can see that it's significantly decreased and it recovered, it kind of recovered and it went back to the baseline levels. However, in MS4, this wasn't even significant. And so it doesn't matter if we see a recovery or not, there is nothing happening in MS4. Finally, so I showed you that CSOs introduce untreated sewage into the urban stream, and CSOs were the main polluters. It wasn't the stormwater runoff. And as I showed you, it takes some time, and especially in the upstream areas where it's not as diluted as much. It takes some time for the recovery to happen. I was taking samples every 2 weeks. So in this time, the rivers are unsafe. So this can act as a bridge for resistance between humans and the environment. And I just want to say, like, these combined sewage systems are a very old system. I think it was the last century or something. It gets every couple of, what is it? What is 10 years in English? Every 10 years or something, they change, they add something to it. They make the retention tanks bigger. But if we're gonna keep having these heavy rain events or the flood events, our water bodies are gonna be filled with sewage. Finally, I would like to thank my supervisors and my colleagues and everyone at the Institute of Hydrobiology and all of our funding agencies.
Thank you so much, Diana, for your presentation. We have any question in the auditorium or online? Yes, we have one.
Thank you very much for the very clear presentation. So, what is how to say, how the sediment nature, the nature of the sediment in the river will promote the carryover of the infection in the river following the flow through of the infection?
Mm-hmm. You mean how does
depending on the nature of the sediments
yes.
in the river. So can you guess how it can carry over the infection in the river, and is it lasting longer depending on the sediment? Is it something that you are looking for?
We did sample sediments for all of our time points. But to be honest with you, I didn't see much difference between the sediments and the water. But I mean, I think the way the stream is, because also we're taking the upper layer of the sediment, it makes sense to see something regarding like heavy metals or chemicals, but for ARGs and bacteria, it doesn't, it doesn't function that way, I think. But yeah, I mean, from the samples that we took, I didn't see a lot of difference between it and between the water.
Okay, thank you very much.
Thank you so much, Diana. Yeah. We have the other questions. No. Perfect. Online, we have one.
Yes, we do have an online question from Maria Chimonopoulou. She asks, for how many weeks urban rivers are unsafe after rain?
So because I sampled every 2 weeks, I can say it's up to 2 weeks. So I'm not so sure about this time period. It could be less, but I didn't sample before that. I'm very sorry.
Okay, thank you so much. Okay, with this presentation, we finish the oral presentation in the day 2 for this Congress. I expect that you have a good time, and I wish good luck them with the PhD student in your project. Okay, and see you in the next session 5. Thank you so much.
Thank you. Now we will end the online session, but we will continue with an in-person only workshop with Angelina Mchadziedoniou and Konstantinos Valias. The workshop is called Bridging One Health and Lifestyle Medicine, an Interconnected Path. So please bear with us because that will be very interesting just to finish, the day. Thank you very much.